Assalamu alaikum wa rahmatullahi wa barakatuhu. A very good morning to our respected chief guest, distinguished guests, honorable speakers, faculty members, researchers, professors, students, ladies and gentlemen. It is my great pleasure to welcome you all to the International Symposium on Sustainable Agriculture under Climate Change Scenario. From seeds to biopesticides, biostimulants, and biofertilizers. We are truly honored by your presence today. Thank you for joining us and being part of this important scientific gathering dedicated to promoting sustainable agriculture and innovative research. To begin this blessed gathering, I would like to invite Mr. Uzair for the recitation of Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان والأرض وضعها للأنام فيها فاكهة والنخل ذات الأكمام والحب ذو العصف والريحان فبأي آلاء ربكما تكذبان صدق الله.
Subhanallah. Now I kindly request everyone to please rise for the national anthem of Pakistan. Thank you, everyone. Kindly be seated. Once again, I warmly welcome to all the distinguished guests, speakers, researchers, and participants. Your presence has made this symposium truly special, and we are sincerely appreciate your valuable time and support. I am Amber Basit from the Syn Forensic DNA and Serology Laboratory, and it is my privilege to be your host for today's symposium. To formally begin today's symposium, it is my honor to introduce Professor Dr. Mohammad Raza Shah, the Director of iccbs. Professor Dr. Mohammad Raza Shah is one of Pakistan's leading scientists in chemistry, specializing in nanotechnology, material science, and advanced drug delivery system. His outstanding work in nanomedicine, particularly in developing nanocarriers for targeted drug delivery, has earned him international recognition. I request him to kindly deliver his.
Okay, Auzubillah minash shaitanir rajim, Bismillahir Rahmanir Rahim. Let me first thank Shahid Masood for arranging this whole gathering. And I also warmly welcome our friends, colleagues from Islamic Energy Commission and other parts of Pakistan for attending this lecture, and dear students, ladies and gentlemen, Assalamu alaikum. You know that I would just discussing with our guests about the sentence. I asked them whether you have visited this research institute, but they told me that they have seen this from just on the road. They were passing through, but they have not entered and have not seen the facilities. But then I thought that's good to just rather than just discussing about the topic, and I'm not an expert on that topic, so there are many other people who know better than me, so they would be just more talking about that topic. So I thought that's good to just introduce the center. You know that the center is established by very great personality and developed also by legends of Pakistan. Starting right from Professor Sri Muzamas Kirti, who just established the new class, and Professor Rahman. They led this center now for more than 5 decades. So he's been with us very actively. And Professor Ebad Choudhury, he also saw this organization for a long period of time and contributed to the development of this center a lot, along with Professor Rahman. And then they handed over to me that torch. And now you just take over the torch and just guide in the center of this building center. So this is actually our various buildings. You know that we are actively working our self-government. So many of these centers are related to biological sciences, and the self-government have adopted these centers. Actually, these centers were established by us from various funding agencies, with the help of various funding agencies, but still the self-government have adopted these centers. And we are actively just working with them on various projects. This center is the only OIC Center of Excellence in chemical sciences. It is the only one UNESCO Center, a Category 2 center in the country. We do have a UNESCO chair on medicine and bioorganic nature product chemistry, which is a unique honor for the country as well. It is also the Gross Center of Excellence. It is WHO Center for Pesticide Analysis. It's the only center in the Muslim world which were declared as this research institute twice by Islamic Development Bank. It's a Gross Center of Excellence where we are training a large number of students. This center is one of the most decorated centers in terms of awards and honors that include also international awards. If you just go to the history, you will find that in our history there are only 4 FRS, and 2 belong from this center. And Professor Sri Musamas Kirti, Professor Rahman, Mustafa Peace be upon prize, Professor Choudhury received that prize. Then again, UNESCO Science Prize is the only one in the Muslim world, which again in honor for us, Professor Rahman, he received the same prize. And this is again a highest level award from the President of the People's Republic of China. that is also in Pakistan. We have two and many of others. And if you just look at the civil awards, so we have like on the Shah Imtiaz 1, Hilal Imtiaz 5, 13, Sitar Imtiaz, and 13, Tamar Imtiaz, and kind of performance. So these are various kinds of, that just reflects the quality of the faculty which we have. So our faculty is internationally renowned faculty, and that is what we always feel proud of them. Similarly, the research productivity of this center is also unmatchable, considering the number of the faculty which we have in our strength. So faculty is not that big one. It is a small one, but the most productive one, and that is just shown here. Similarly, we are very also good in earning international and national research grant, especially international ones. As you can see that this it was big. I was just discussing this with Professor Rahman a couple of weeks ago. So he told me that this was the funding which was received back in 1970s. And this is actually from the German government, and then it started. As you can see that back in 80s, we received 23 million US dollar from the Japan, and many other international projects. So that shows that you know that you need to have to just generate funding for the research, because without research funding, you cannot just continue your research programs. We are also linked very closely internationally with all continent of the world. We have signed in MOUs and agreements, and we have active collaboration. Over 200 MOUs have been signed with various international and national institutions. These are some of our academic partners. And there is something very much unique in our center, that we have just established laboratories here, and a mirror laboratories of such kind is also there in China. So we are very much closely working with our Chinese colleagues due to a strategic relationship between China and Pakistan. Similarly, we are equally involved in traditional Chinese medicines, and wanted to just have an alternative way, an alternative means of treatment. And we realized that this traditional Chinese medicine is having a thousands years of history, and these are quite effective. Over 92% people in China are just using traditional Chinese medicine. So we realized that it's important that we should check these medicine on Pakistani population and establish the efficacy and safety on Pakistani population, and then that would be an alternate source of medicines for the Pakistani population, and that's what we have been working with them. Our facility is a state-of-the-art facility in this part of the world. It is better than many universities of Europe and US. Like we are having 13 superconducting nuclear magnetic resonance machines, 20 mass spectrometer, state-of-the-art imaging facility. This is again a very important, especially for the cutting-edge research. We have like atomic force microscope, scanning electron microscope, and many other facility here. Our industrial analytical center is again internationally accredited center, one of the best in the country, providing services. We value the quality of our work and our research, and that's the reason that is you see that our center is accredited 9001, 17025, and so on. So this is actually you need such kind of certifications and such kind of acknowledgement from the regulatory body and the quality management system in the country which are in place. So that's what we also invested a lot of our resources and energy. We are also providing additional services because we consider ourselves as a premier institution of the country. So we should lead various projects which are of national need, like during the pandemic, we converted our virology lab to COVID-19 testing facilities, and we conducted almost 500,000 PCR confirmed tests. And that was the time when there were no facilities available in the country, and nobody was knowing that how to just test this virus. So that was one of our contribution. Then we conducted phase one clinical trial of the Sinopharm vaccine. And that was actually the phase one clinical trial is considered to be the most difficult one, because that is the trial when you just translate from animal to human beings. So that is normally most of the research organization or the clinical trial site, they used to they are reluctant to do that. And here in Pakistan, only there are two organizations who have received that license to conduct phase one clinical trial, and one of that license is with us. Though we are not hospital, we do not have any hospital, but still we have a great trust on the regulator, and they have given us the phase one trial license, because they know that we value the importance of the quality. Similarly, our St. Forensic Data and Serology labs have solved thousands of cases for the Sindh province, and also some cases from the Balochistan province. The purpose of this lab is dual. We are not only just providing testing services and solving their cases, but we are also training the judiciaries and other law enforcement agencies officials to just they are bit more knowledgeable, and that is what we are doing here. Then we also conduct these other these are some of our other services which we provide to the public. Industrial Analytical Center is state of the art. We are providing services to 600 industries in Pakistan, and these are some of the clients. So these are the features. These are some of the features of our Industrial Analytical Center. We are also having halal testing laboratory. In our laboratory is the only. We have two. Like one is we are also halal certification like authority. We can, our facility can provide a halal certification based on Sharia compliance. But at the same time, our laboratory is the only one in the country which can just provide certification based on science, based on scientific evidence, and that's and our laboratory was declared is a reference laboratory for the rest of the country by Pakistan Halal Authority. So that is also something. This is what I talked about. As you can see, that the confidence of the regulators. So our center have received full license from Drug Regulatory Authority of Pakistan. Similarly, the Sindh Healthcare Commission have given a certificate to act as a primary care facility in category 3. So that is, though it is not an hospital, but still they have given us the license, and that is the only one in the country. There is no any other organization who have been declared by any healthcare commission as a center where they can just conduct clinical trials or deal the patient. Similarly, Pakistan Accreditation Council, these are some of our other facilities in the same centers. Our Plant Biotechnology Center is also very active one. We are planning to just make it a more robust one and just do some more work here. So many of people who are sitting here, we will be happy to just work with you here, and just it's also state of the art, very good facility, and you can have a benefit from this facility. We do have technology incubation centers, and many of our incubators have already received several prizes. Like just a couple of weeks ago, one of our incubators in the Bonzo province, they received 11 million US dollar for a project which they are conducting in our incubation center. So they competed internationally in the Guangzhou province, and they received 11 million US dollar to just carry on the projects here in our facility. So many of our incubators are performing very well in this center as well. We also have many scholars, and we are also like providing training to international scholars. More than 1,200 scholars from developed and developing countries. We train in our center. Yesterday, Professor Rahman just told me that now you concentrate on bringing students from Germany, France, and Europe, because in the past we were receiving a large number of students. Due to security situation in the country, that discontinued. But now our next target is that we should have students and researchers from the advanced countries as well. We are receiving currently students from the Africa and Gulf and Central Asia. But we would like that people from the Germany, France, and Turkey and United States. They were coming here in the past, but they should also again start coming here in our country. These are various kind of scholarship which we offer to the international students. And so these are some of our international students who used to stay with us in the past. And with this, let me thank you very much for listening me. And I wish you a pleasant and productive day ahead. Thank you so much.
Thank you so much, sir, for your inspiring welcome remarks and a brief introduction of iccbs. Our next speaker is Dr. Shahid Mansoor, HEC recognized national professor and one of the Pakistan leading agriculture biotechnologists. His remarkable research on cotton leaf curl disease and crop biotechnology has made significant contributions to agriculture science and sustainable farming. Now, I would like to request Dr. Shahid Mansoor to come to the stage to deliver his topic.
Thank you very much. I think it's a very brief 3 slides presentation just to set the stage that why we are first of all. So basically, I joined this institute in November 2024, 2022, and then we thought that what we can do productive work here. And the focusing was on one of the neglected areas, in Balochistan and dry areas, Punjab, and also human and well-being that are compatible with the sustainable development goals, zero hunger, good health, well-being, climate action, and partnership for the goals. So the action that we started to do is to development and selection of crops suitable for dry areas. And I'm very proud that we did some very interesting work, technology for sustainable economy, health and safety by pesticide formulation. So one of the major issues that we have in the export of our commodities from Pakistan is the pesticide residue and aflatoxin. So we started to work on developing this technology, and I'm very glad that we received some funding from Higher Education Commission, and we started this work. And also trial and development of biofertilizers. So we are not directly involved, but we are specificating different institutions of Pakistan Economic Energy Commission and other institutions and industry to come up with this work. And sustainable livestock, I will talk a little bit more about this and national international collaboration. So the first thing that I did was that networking. So I have some friends here, and some of them are sitting here. And then what we achieved so far is that we worked on cotton varieties and 3 cotton generation in a year. So that was the first achievement that we had, that we were able to establish a system where under national conditions we could grow 3 generation of cotton in 1 year. And this was possible because that the while mild climatic condition in Karachi, and we developed some essentially dry varieties of cotton, Nayab Kiran, Nayab, and then also new varieties, which is double gene and triple gene varieties, and also we are coming up with another variety which is has also multiple resistance against multiple sucking pests. This project under TPF, we have filed 4 patents so far, and biofertilizer to replace DAP, which is called BOP, and we are working with SM industry in Uttar Pradesh. And then we have also found very interesting data. We have collaboration with King Abdullah University, and we developed we used this Professor Salim Al-Babi, and then we have Dr. Asha Jalal sitting here. We tested this biostimulant throughout Pakistan and found very interesting data about 30-40% yield increase. And also one very important discovery was that we had high accumulation of zinc and iron, and probably this was because of the extensive root system that we have because of this biostimulant. Genome edited tomato, one of our PhD students is working with this. We have collaboration with MIT on vitamin D high, because vitamin D deficiency is very, very important. BNIB, DW2 mutant, and wheat genome at wheat, we are doing some trial and finding some very good data. So what we are expecting is that there will be a big impact that we can generate by working on bispecific technology. And some of the inspiration of this work came from Professor Veena Siddiqui, who is sitting here. She developed neem-based biopesticide, and I hope that we are extending the horizon of this technology. And one of the targets is to reduce the aflatoxin and completely eliminate use of chemical pesticide. Now, this is one interesting case, as we can say this champion. She's a lady from Brazil, and a student of Professor Johanna, who used to come to Nayab and Nibji, and she created an impact of $250 billion just by using biofertilizers. And she got the what we call as the World Food Prize in 2025. And why this is all important, that when you are using less chemical fertilizer, you are helping the environment, and this is equivalent to 230 million metric tons of CO2 equivalent. And I hope that this institution, because of their huge work on natural product chemistry and all the work that we are doing with me, Professor Mushadaf, we are hoping that we will have a new generation of biopesticide which will help to increase our export. Thank you very much.
Thank you, Professor Dr. Shahid Mansoor, for your valuable presentation. Now it is my great privilege to introduce Professor Dr. Ataur Rahman, founder and patron-in-chief of iccbs, fellow of the Royal Society, and recipient of Pakistan's highest civilian award, the Nishan-e-Imtiaz. His visionary leadership transformed iccbs into a UNESCO Center of Excellence and has greatly advanced scientific research and higher education in Pakistan. Ladies and gentlemen, please welcome Dr. Ataur Rahman to deliver his talk.
Assalamu alaikum, everyone. It's my pleasure to welcome all our guests here. We have Dr. Shakir Abbas from the Atomic Energy Commission and the colleagues from the Atomic Energy Commission. as well as other participants. So special welcome to the Institute. And I'd like to thank Dr. Shahid Mansoor for organizing this excellent symposium. So, thank you. I'd like to just begin by showing two slides before I go to my talk, since there are many new people here. This is Pakistan's ranking, international ranking, published by Saimago. In the year 2000, in most subjects, we were ranked 90, 60, 50, 40. All of the changes that I tried to introduce back in the year 2000 to 2008 as chairman at HC, our present rankings are in different subjects are like the, for instance, in agriculture, if you're interested, we are 48, now we are number 15 in the world, and so on. So something remarkable has happened in Pakistan in the last 20 years. People are not aware. The entire landscape of higher education, science, and technology has changed. And what is important is that we have to continue in this direction with HEC and support and with the government's vision. The real challenge for Pakistan is to tap into our real wealth, it's our children, through quality education, through science, technology, and through innovation. And this is a slide which shows Pakistan versus India over the last 20 years, how things have changed. Back in the year 2000, till the year 2000, we were about 400% behind India in per capita research output in public, according to Web of Science. And then this changed, and now we are about 10 or 15% ahead of India. We overtook India in 2017. This is data from Web of Science. The Scopus data is also very similar. So Pakistan is about the same or slightly ahead of India on a per capita basis. In actual numbers, of course, we are about 6 or 7 times behind India. But previously in 2000, we were about 30 times behind India. But on per capita basis, we have caught up with India, which again is not a small achievement. Now, let me take you to the topic of my talk. I'll be focusing on the new horizons, and I'll focus on two areas. One is the advent of advances in genomics and how they can have a major impact in discovery of new bioactive natural products. And the other is artificial intelligence, which is now permeating through every area of human activity, including research. So let me talk about this approach. In HEJ, in iccbs, we have also, over the last 50, 60 years, what we've been doing is we have been taking plants largely, we have been extracting substances, we have been trying to do NMR, mass spectroscopy, and so on to discover new compounds. And often at the end of 4 or 5 years, the students end up with 3, 4, 5, 6 new or novel compounds and with a large number of known compounds. And it takes years and years and years. In many cases, they find that they don't have any new compounds. There are known compounds which are being discovered again and again. And so that it results in a lot of wastage of time and effort. But in the last 3 to 4 years, there has been a complete transformation which we need to be aware of. And that is where people like Dr. Ishtiaq and Dr. Shahid Mansoor and other colleagues come in. And this is a challenge I'm putting to the two of you, especially Dr. Ishtiaq and Dr. Shahid, to introduce this DNA first approach that I'm integrating, which means that you have to have training workshops, train our students that there is a completely different approach now to discovering bioactive natural products. And that is the approach I'm going to talk about. The advantage of the DNA first approach is that you don't have to spend years and years in extracting and discovering known compounds. You can, in a matter of 1 month or a few weeks, actually just by looking at the DNA of the molecules from the plants, the biosynthetic gene clusters to be exact, you can tell whether this plant is going to produce new compounds or known compounds. And it is so from the identification of the biosynthetic gene clusters, which can be done very easily now, you can tell whether this plant has potential or not. So we need to make major changes in natural product chemistry, and we have been the leaders, among the leaders in the world in natural products, starting from late Professor Sulaiman Siddiqui to now, alhamdulillah, we have been one of the best institutes in natural product chemistry. Now the horizons have changed, and this is what I would like to happen with this institute in the future. So here the DNA first approach, not plant isolation, extraction, and so on. DNA first. That is the approach which I'm talking about. Miss Sorry. Should I talk to this? Okay. So the DNA first approach can lead to identifications as you are in the field of agriculture. It can lead to discovering a large number of molecules of which are such as biostimulants, biopesticides, and others, biofertilizers, using this approach. Now, what is this approach? Well, the DNA first approach basically, in summary, it is to first of all to read the genetic code of the organism, do a computer analysis, a laboratory validation, and this leads to the final product. This is just a summary. But let me talk to you a little more in detail what we are doing. So instead of first extracting chemicals from organisms, we are now studying their DNA to predict which useful natural products they can produce. So the first step is collecting the organism and extracting the DNA. That's fairly simple. You collect the organism, extract the DNA, check the quality of DNA, and this is ready for sequencing. So this is just a very simple, straightforward thing. And the sequencing, of course, is done by normal sequencing machines. So the first step then in that case will be once you have the DNA, you have the DNA sample with the right quality, you sequence it, and then the sequencing machine, which may be Illumina or whatever sequencing machine you have, produces millions of DNA reads, fragments. And these are then assembled using computers to build the whole genome. So you assemble that the complete genome is then ready to be searched for special gene clusters. Now what you're looking at is clusters of genes which produce the active metabolites. So does the plant or animal organism or whatever have biosynthetic gene clusters of interest or not? That is the question. And so the complete genome is searched for special gene clusters, and the genome mining process is then carried out using specialized softwares. And there are softwares available such as anti-smash or the deep BGC or GECO. These are all softwares available to find the biosynthetic gene clusters. And once you discover the biosynthetic gene clusters, now there are different type of clusters which produce different classes of metabolites. So you may have clusters which produce polyketides. You may have other clusters which produce non-ribosomal peptides. You may have clusters which produce terpenes, alkaloids, and others. You classify the clusters. Okay, this plant or this organism has these kind of clusters, and it is likely to produce these kind of compounds just by looking at the biosynthetic gene clusters. We have not gone into any extraction or purification or NMR or anything like that. So the next step is that once you know what kind of cluster this plant has, once you know what kind of cluster this plant has, for instance, if it has PTS genes, it's going to produce polyketides. If it has NRPS genes, it has peptide antibiotics are produced. So similarly, there are different types of clusters for terpene synthesis, for ribosomal synthetic peptides and saccharide genes for complex sugars. The likely product can be predicted once you know what kind of cluster that has, and what these clusters are identified using the software which I showed on the previous slide. So then next step is, does it have a novelty in it? जो cluster मिला है उसमें कोई novelty है या नहीं, उससे कोई चीज interesting मिलेगी या नहीं मिलेगी, ये अगला step है. Which is again very easy. अगर वो cluster ऐसा मिला है जो कि पहले identical है किसी और compounds में उस तरह के cluster मिल चुके हैं, 99 let's say 99% similar to
other organisms. इसका मतलब ये है कि इससे कंपाउंड वो मिलेंगे जो कि सिमिलर होंगे, जो एक्सट्रैक्ट हो चुके नॉन कंपाउंड आइसोलेट होंगे. लेकिन अगर क्लस्टर, इफ यू गेट अ क्लस्टर which is only 40% similar to similar biosynthetic gene clusters isolated from other organisms, that means that it has a huge potential to give completely new organisms. तो 4 से 6 हफ्ते में आप खाली ये प्रोसेस करके आइडेंटिफाई कर सकते हैं कि इसके बायोसिंथेटिक जीन क्लस्टर्स ऐसे हैं जो दूसरे जो बायोसिंथेटिक जीन क्लस्टर्स पहले दूसरे ऑर्गेनिज्म से आइसोलेट हुए हैं उनसे काफी डिफरेंट हैं. तो फिर उसके मेटाबोलाइट्स भी जो होंगे वो खासे डिफरेंट होंगे. फिर जो है एक और चीज जो है कि बहुत सी दफा बायोसिंथेटिक जीन्स होते हैं लेकिन वो साइलेंट होते हैं और वो पोटेंशियल होता है उनमें कि वो दूसरे मेटाबोलाइट्स प्रोड्यूस ना करें, लेकिन बिकॉज दोस जीन्स आर साइलेंट therefore those metabolites are not produced. So you need to then check and see if you can activate silent genes present in that biosynthetic gene cluster को देखते हुए through the use of a number of techniques which includes promoters होते हैं, strong promoter replacement कर देते, sorry, you replace the promoters, sorry, yes, so you do either a promoter replacement or you have activator proteins, transcription factors या small molecule inducers होते हैं या modified भी आपके epigenetic modifications के आप activate कर सकते हैं silent genes को. तो ऐसा biosynthetic gene cluster जो कि of interest है लेकिन वो compound produce नहीं कर रहा जो आप चाहते हैं, लेकिन उसमें potential है produce करने का. इसका मतलब है उसके genes silent हैं, on gene silent को turn on करना पड़ेगा, they are turned off, and that can be done through various activation mechanisms. उसके बाद आता है कि ये आपने बायोसिंथेटिक जीन क्लस्टर को आपने आइडेंटिफाई कर लिया, तो उसके बाद कंप्यूटर एनालिसिस से यू रीड द सॉफ्टवेयर एंड प्रिडिक्ट हाउ इस क्लस्टर से किस किस्म के कंपाउंड्स प्रोड्यूस होंगे. बिकॉज यू नो देयर इज अ प्रोसेस इन विच जो बायोसिंथेटिक जीन्स हैं उसके थ्रू फिर you have RNA production and then you have protein production and then you have metabolites production. There is a process. So उस process से, so does this have the potential? So the software जो available है वो Prism है, Anti-Smash है, these are routinely available softwares which read the genes and predict कि इन genes से जो building blocks बनेंगे, जो small building blocks, मिसाल के तौर पे आप terpenes ले लें तो मेवालोनेट का बिल्डिंग ब्लॉक होगा. इस तरीके के जो टर्पीन सिंथेसिस के अंदर जो बिल्डिंग ब्लॉक्स होते हैं वो सारे बिल्डिंग ब्लॉक्स बनेंगे, वो आइडेंटिफाई हो सकते हैं यहाँ से उससे आप पर, और उन बिल्डिंग ब्लॉक्स को सामने रखते हुए देन यू हैव सॉफ्टवेयर टू प्रिडिक्ट इससे जो नेचुरल प्रोडक्ट्स बनेंगी वो किस तरह की बनेंगी. सो देयर इज दिस इज ऑल डन बाय सॉफ्टवेयर. इट इज वेरी इजी, इट कैन बी डन इन अ मैटर ऑफ मिनट्स, इट डजंट टेक आवर्स. फौरी तौर पे बता देता है कि इससे ये बिल्डिंग ब्लॉक्स बनेंगे और इन बिल्डिंग ब्लॉक से ये कंपाउंड बनने की के हाई चांसेस हैं. एंड फाइनली once you have done this you insert this gene cluster जो हमने चूज किया है उसको आप वेक्टर के अंदर या प्लाज्मिड के अंदर इंसर्ट कर देते हैं और ये फिर आपने उसमें इंसर्ट कर दिया प्लाज्मिड के अंदर और फिर इसको किसी होस्ट में ट्रांसफर करते हैं which can be either Escherichia coli, or Baker's yeast, or Bacillus, or Streptomyces, some suitable host, जो कि फिर fast growing host organism होता है, उससे फिर आप natural products produce करते हैं. So actually you're using those very gene clusters from the plant, transferring those genes into a suitable host, and then producing the desired new novel compounds that will be produced from the plant itself. तो इससे बहुत सारे जो बायोएक्टिव कंपाउंड्स हैं वो बड़ी क्वांटिटीज में आप हासिल कर सकते हैं एंड प्रोड्यूस कर सकते हैं. और फाइनली फिर ये आप उन स्टेप्स पे आ जाते हैं जो कि बताया, जो कि आप सब जानते हैं कि उसमें फिर एनएमआर, मास और फिर सेक्स, फाइनली स्ट्रक्चर एलुसिडेशन, दैट आई डोंट हैव टू टॉक अबाउट, यू नो इट वेरी वेल. और इसके लिए भी बहुत सारे स्ट्रक्चर आइडेंटिफिकेशन के लिए NMR और मास के लिए अब बहुत सारे AI टूल्स आ गए हैं जो कि अब बहुत सारे यहाँ इस्तेमाल नहीं हो रहे. And here this will be a challenge for some of our people working in NMR and mass to train our students how to use these softwares ताकि वो मास स्पेक्ट्रा सीरियस को इस्तेमाल करते हुए फॉर इंस्टेंस मास इंटरप्रेट कर सकते हैं. इसी तरह दूसरे सॉफ्टवेयर जो हैं एमजमीन है और GNPS है, इससे मास स्पेक्ट्रोस्कोपी से आप आइडेंटिफाई कर सकते हैं. एंड फाइनली यू डू हैव अ वैलिडेशन वगैरह करते हैं इसके साथ. तो पूरी डिस्कवरी से टू न्यू मेडिसिन ये कंप्लीटली न्यू अप्रोच है टू प्रोडक्शन टू आइडेंटिफिकेशन, एंड आई होप दैट दिस विल बी अडॉप्टेड हियर. The second part of my talk, and I'll only take 5 minutes, is artificial intelligence. How to integrate artificial intelligence into all this. So there are lots of benefits of AI now. It is being used for almost excuse me, for almost all areas. ये तो मैंने आपको बताया अभी कि BCC वगैरह बायोसिंथेटिक जीन क्लस्टर्स के लिए किस तरह इस्तेमाल होता है. Excuse me. Thank you. NMR आइडेंटिफिकेशन के अंदर ब्रूकर, आपके पास NMR है, हमारे यहाँ ब्रूकर लगे हुए ज़्यादातर. तो ऑटोमेटेड तरीके से जो मुख्तलिफ सॉफ्टवेयर अवेलेबल हैं, जो कि जैसे कि अश्योर NMR AI है, ACD लैब स्पेक्ट्रम AI है, एमनोवा AI है, जिससे पूरे के पूरे स्ट्रक्चर को आइडेंटिफाई होता है. वो बता देता है फौरन आप NMR डालें, स्ट्रक्चर सामने आ जाएंगे कि इसका 90% फिट है, इसका 85% फिट है, इसका 70% फिट है. सो इट विल सेव यू अ लॉट ऑफ टाइम इन इंटरप्रिटेशन एज लॉन्ग एज यू हैव प्योर सैंपल जिसका आप NMR कर रहे हैं. फॉर इंस्टेंस ये आपने ये वाला जो NMR है आपने उस पर डाला सॉफ्टवेयर में, और सॉफ्टवेयर में फौरन इंटरप्रिटेशन बताई कि साहब ये लग रहा है कि कंपाउंड है, और मुझे लगता है कि इसका स्ट्रक्चर प्रॉब्ली ये होगा, और इसको प्रिडिक्ट कर दिया कि इस तरीके से इसका स्ट्रक्चर आएगा. तो यू जस्ट पुट इन द NMR and you get out the structure through a software, again in a matter of seconds. And similarly, if you have a mass spectrum, आपने mass spectrum डाला सॉफ्टवेयर में, और सॉफ्टवेयर जो है उसने फिर आपको बताया अच्छा ये mass spectrum है, तो इसका जो है इसकी जो likely chances of structure हो सकते हैं, ये वाला जो है स्ट्रक्चर है इसका 95% फिट है, ये वाला जो है इसका 82% फिट है, इसका 70. फिर आपने दूसरे NMR वगैरह से देखकर और कर दिया. अगेन दिस टेक्स सेकंड्स ओनली, एंड देयर आर वेरियस NMR टूल्स मास स्पेक्ट्रल टूल्स व्हिच आई हैव जस्ट आइडेंटिफाइड. तो डॉक्टर मुशर्रफ से कहूंगा कि ये एक आप ट्रेनिंग वर्कशॉप्स करें फोकस्ड ऑन यूज ऑफ मास सॉफ्टवेयर्स फॉर आइडेंटिफिकेशन. और ये जो है इस पे मैंने रिव्यू भी लिखा है जो एल्सेवियर की किताब में आ रहा है, DNA फर्स्ट अप्रोच, उसको भी मैं दे दूंगा जिसमें डिटेल्स है सॉफ्टवेयर वगैरह की जो कि आजकल इस्तेमाल हो रहे हैं. और ये हर महीने, हर 2 महीने, हर 4 महीने में नए सॉफ्टवेयर आते जा रहे हैं. तो यू नीड टू बी अप टू डेट विद द लेटेस्ट सॉफ्टवेयर और व्हाट यू आर डूइंग. मास स्पेक्ट्रोमेट्री में सीरियस है, CSI फाइंड है, MS नोवेलिस्ट है. सिंथेटिक केमिस्ट्री में आपको एक सिंथेसिस करनी है, आपने स्ट्रक्चर डाल दिया और सॉफ्टवेयर से आपने सिंथिया इस्तेमाल किया, तो ये बताएगा कि किस सिंथेटिक रूट से इसको आसानी से बनाया जा सकता है. पाया AI डाला, उससे रेट्रोसिंथेटिक प्लानिंग हो सकती है आसानी से. रूट डिजाइनर है ये, मल्टी स्टेप रूट ऑप्टिमाइजेशन यूजिंग रूट डिजाइनर AI. इसमें नोबेल प्राइजेस मिली हैं अभी, एक्सक्यूज मी माफ करिएगा, इसमें नोबेल प्राइजेस मिली हैं AlphaFold 2 के ऊपर अभी 2 साल पहले कि जो प्रोटीन का 3 डायमेंशनल स्ट्रक्चर. इसी तरह रोजेटा फोल्ड है, ESM3 है फॉर प्रोटीन जनरेशन एंड प्रोटीन इंजीनियरिंग. मॉलिक्यूलर डिजाइन के सॉफ्टवेयर्स हैं. आई वोंट गो इनटू द डिटेल्स. रिएक्शन प्रिडिक्शन के लिए रिएक्शन करेंगे, क्या क्या प्रोडक्ट्स मिल सकते हैं वो प्रिडिक्ट करता है बेस्ड ऑन दुनिया का सारा लिटरेचर चंद सेकंड में देखता है, और उसके बुनियाद के ऊपर आपको बता देता है आधे मिनट के अंदर कि आपके जो सिंथेटिक प्रोडक्ट्स हैं इससे लाइकली ये मेजर प्रोडक्ट होगा और ये बाय प्रोडक्ट्स होंगे. एंड दीज आर वेरियस सॉफ्टवेयर अवेलेबल. फिर साथ के साथ और भी चीजें हो गई, जैसे इंटेलिजेंट ट्यूटरिंग सिस्टम्स आ गए हैं. अब पर्सनलाइज ट्यूटर, एक मुल्क में 2.5 करोड़ आउट ऑफ स्कूल चिल्ड्रन हैं पाकिस्तान में. अब इनको किस तरह एजुकेट किया जाए, हर बच्चे के लिए अलग अलग AI ट्यूटर मौजूद है. ये इन्होंने इंट्रोड्यूस किया है एक्टिव लर्निंग प्लेटफॉर्म्स जो कि ड्रीम बॉक्स है, इसी तरीके से और एक्टिव लर्निंग प्लेटफॉर्म वेयर यू कैन गेट खान एकेडमी ने आप सुना होगा खान एकेडमी का नाम, तो खान एकेडमी ने खान मीगो के नाम से इंट्रोड्यूस किया है, जिसमें हर बच्चे के लिए स्कूल लेवल पर अलग अलग एआई ट्यूटर जो जांचता है उसकी कमजोरियां कहाँ हैं और उसी कमजोरियों को जांच कर उसको स्टेप बाय स्टेप आगे लेके जाता है। तो बहुत सारी चीजें हो रही हैं जिसका मैं जिक्र कर रहा था, मैं ज्यादा टाइम इस पर नहीं लेना चाहता, बट दिस इज अ होल न्यू फील्ड वेयर यू हैव पर्सनलाइज्ड पर्सनलाइज्ड ट्यूटर्स अवेलेबल, और रिसर्च के अंदर अब एआई हैज कम इन, लीगल सिस्टम रिप्लेस हो रहा है बाय एआई, लॉयर्स, मेडिकल सिस्टम रिप्लेस हो रहा है एआई डॉक्टर्स। कल माइक्रोसॉफ्ट ने अनाउंस किया है कि 4,800 लोगों को उन्होंने फायर किया है माइक्रोसॉफ्ट में, बिकॉज दे डोंट नीड दोस पीपल, एआई इज डूइंग दैट जॉब। छह महीने पहले अमेजॉन ने अनाउंस किया था कि 30,000 लोगों को उन्होंने फायर कर दिया है अपने साथ, क्योंकि नाउ दे आर यूजिंग एआई पीपल। सो देयर इज अ कंप्लीट चेंज गोइंग ऑन इन द एंटायर landscape of research and then of employment. So we need to train our students to this new world of tomorrow, where genomics and artificial intelligence and nanotechnology and other such new areas that are coming up. So this is the challenge for iccbs. Are you training our students for today or for this amazing world of tomorrow, where AI is integrated into almost every sphere of human activity? And that is the challenge I pose to all the faculty members here. Sorry, I can't talk to you in great detail here because I was only given about 15 or 20 minutes, so I have to stop here. But this was just a simple reflection कि नए horizons अब आ गए हैं साइंस में, नेचुरल प्रोडक्ट केमिस्ट्री में नए horizons आ गए हैं, और this institute has been a world leader in this field. Let us continue to lead in the manner in which I have just shown you. Thank you. Assalamu alaikum.
We are already starting.
Thank you so much, Dr. Ataur Rahman, for your valuable presentation. Now, our next speaker is Professor Kausar A. Malik, who will be presenting the topic on knowledge-based next-generation biofertilizers, and he will be joining us via online. Now, I request Dr. Shahid Mansoor to provide a brief introduction about him.
Thank you. I think it is my great honor to introduce my mentor, Professor Kausar Abdullah Malik. I mean, he has many distinguished and started his career in Atomic Energy Commission and then became chairman of Pakistan Agriculture Research Council, member Planning Commission, and then again, he was minister in cabinet, federal minister for food security and research. And I think that we are very proud of him that he has continued his journey and research. So without wasting time, I request Professor Kausar Abdullah Malik. At the moment, he is also our president of our Pakistan Academy of Sciences. So he has all the, wealth and champion of science in Pakistan. So thank you, sir, for joining us. So please start. So you can share your screen.
Bismillahirrahmanirrahim. Thank you so much, Shahid, for introducing me, and it's an honor to follow Professor Ataur Rahman Sahab. I enjoyed his talk, and I'm happy that we are following his footsteps as well. See, we have been working for a long time on biofertilizers, and I think the people at the NIPG are still working at it. Our product biopower is still in the market, and it's heartening to see that all those things that we try to develop are still in the market. You see, the other interesting thing that I would like to share, and I'm sure that most of you are already aware, that the government of Pakistan has approved the National Agricultural Biotechnology Policy, and that has been a big achievement. And because that also includes all the use of biofertilizers, and then based on that, a lot of the activity is already going on in different areas. We have been part of a project sponsored by the USDA and ICARDA to test all these biofertilizers on the farmers' fields, you see. So we have been doing that for the last 2 years, and then our results have been excellent. And I think that will be a good development. But as Dr. Tauraman Saab also mentioned in his talk, you see that we have to look at the other avenues also, you see, because isolation of all these organisms has already been there, and it has been fully utilized, you see. So let me, if I could have my slides. Do you have, Shahid, do you have those slides I had already sent you? No, that's not the one.
डाउनलोड कर ली ये लो चल जी सर your presentation is there.
Yes, that's the one. Okay. You see, our main work had been looking at the rhizosphere of the plant, and we started our work on associative nitrogen fixation with roots of non-legumes. You see, that was the one which initiated the all the work that was done in Brazil by Professor Tobrainer and her colleagues, and that was all based on isolations from the rhizosphere of plants. And at that time, the acetylene reduction technique had been developed, and that was the basis of selecting all the nitrogen-fixing microorganisms. See, so all our biofertilizers are based on these preliminary isolations, you see. So, I mean, then there was a development of microbiome, that is looking at the metagenome of a plant or of any environmental sample, that we could isolate the whole genome, whole DNA from that sample, and then we could find out which kind of microorganisms are there and which kind of genes are there, and that was the basis of whole new era of metagenomics, and we have been involved in that for a number of years now, and based on that we have developed certain approaches. One approach that we recently, over the last 3-4 years, we followed is regarding cotton, you see, because we thought that we must know the microbiome of cotton, having all these CLCV viral infections, you see. And based on that, we had been working on different areas, and we have developed. I will show you in a while the different areas that we have been doing. You see, for example, all these we. You see, important thing has been that we must look at the plant itself also. You see, not only the rhizosphere or that, but the whole plant physiology through different developmental stages. Root exudates are an important composition, and then all the environmental influences and the plant immune system. You see, biofertilizers at the moment are being used only for that we call them PGPR, which is plant growth promoting bacteria. You see, so these are for nitrogen fixation, for phosphorus utilization, and for all hormonal effects. You see, so we wanted to enlarge this scope and use these for the suppression of any diseases, you see. And for that, we had to look at those microorganisms which are involved with the disease itself, you see. So therefore, we started working on microbiome of cotton having CLCV infection, you see. So these are we were able to then look at some of the microbes which are involved with these studies, you see. Could I have the next slide, please? Yeah, as I said, that this was based on plant physiology, the metabolomics, microbiome, the dynamics, and environment. All these interactions have to be there. And as the Dr. Ataur Rahman also mentioned, that we have to take into account all these, you see. I mean, this NGS that we call next generation sequencing, that is such a strong tool, you see, that if we have the whole genome of an organism, we can find so many new functions, you see, because there are so many different softwares available which can do the job, you see. You can know that which are the functional genes over there, which are the ones which are responsible for different functions. And that's the way we try to approach this whole phenomenon, you see. And then understanding, obviously, based on that, understanding plant microbe interactions, and then precision biofertilizer design. We want to use all this information to design our biofertilizers, you see. They have the next one, please. Yes, you see, my colleague Aisha Badar, who is our postdoctoral fellow, she's also, I think, online, and I would like her also to talk about it. And this all depends on the plant release signals, you see, because as I think our colleagues who are in rhizobium business also, they all know the signaling compounds which are released by the host plant. And in this particular case, we found that this paracomeric acid is the one which is released in the environment, you see, and that triggers the interaction of different microorganisms, and this formulates the whole phenomena of plant microbial interaction, you see, and that is all based on different compounds. And I would like to hand over this part of the talk to Aisha Badar, Dr. Aisha Badar. Are you here? Can you log in? Aisha, are you there? Anyway, so these are the microbial selections. We make the microbial selections, and then we have a mechanism of home metabolism, you see, and that eventually we, I mean, all these strains that we isolate are developed into the sin, this. I mean, all these compounds, all these strains are then isolated, and then these are, I mean, tested on the plant and to see whether there is any disease suppression or not, you see. On the other hand, with all these, we have the whole genome information, and based on that genome information, we can determine that which kind of strains are available and which kinds of genes are activated. You see, we do the transcriptome analysis of the whole thing and see that which are the genes which are functional. You see, could I have the next one, please? Yeah, this is, we call it a precision biofertilizers for sustainable agriculture. That is the rationally designed synthetic microbial communities, which we call SynCom. See, I was forgetting the name. You see, the SynComs are synthetic community because all these organisms that we use are in a consortium. You see, and therefore we have to know that which organism is doing what. You see, and then we can do the genome editing and synthetic biology to enhance beneficial microbial traits and improve plant responsiveness, and then precision selection of microbes, etc. You see, so but our future vision is that we have to have a natural plant microbiome, and this multi-omics characterization and then identification of beneficial uh, 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 plant growth promoting functions, and then synthetic biology and genome editing and precision biofertilizers for climate resilient agriculture. These are all areas which are doable, and this is only possible with the advent of NGS, with this next generation sequencing, and also the availability of different softwares that we can use to identify different genes with specific functions that we require. You see, so. In other words, the future of biofertilizer is not simply discovering better microbes, it is understanding how plants naturally assemble beneficial microbial communities and using that knowledge to design precision crop specific and environmentally adaptive microbial solution. So I hope that we are at a stage that we have done some field trials on our synthetic communities to see whether there is any suppression of CLCV in cotton or not. You see, so we are in process of doing field trials in Multan, and we hope that we can find some exciting results as a consequence. So I thank you all, and I wish you all the best. Thank you.
Well, thank you very much, Dr. Attar here. It's a special honor to have Dr. Qasim Malik participating in our function. He's the president of our academy, and he's been a trailblazer in the field of biotechnology. And it's a real honor, sir, that to have you join us. And I hope next time we'll be able to persuade you to come here physically to Karachi and participate in these conferences. But thank you very much for your very nice presentation. And we hope that we can work together and you can guide us in the future development of biotechnology in our institute. We have Shahid Mansoor here, and he and his other colleagues can be guided by you, especially the area of microbiome in plants and how they are affecting the development of plants themselves and the development of secondary metabolites, because within the microbiome you have good microbes and you have bad microbes. And so a detailed study of the microbial world which exists within a plant provides deep insights into the plant species. So very, very grateful for your very interesting talk, sir.
Thank you, sir. Thank you, sir. It's an honor to be here and to be talking in presence of yourself. And we keep on learning a lot from your knowledge. You see, thank you so much.
Thank you so much, Dr. Qasim, for your valuable presentation. Now it is my pleasure to introduce Professor Dr. Beena Shaheen Siddiqui, Distinguished National Professor at iccbs and a world-renowned natural product chemist. She is a fellow of TBAS and Pakistan Academy of Sciences and has received numerous prestigious awards, including the Sitara Imtiaz, Tamga Imtiaz, and the Khwarizmi International Award. Now I would like to request Dr. Beena Shaheen Siddiqui.
12 minutes.
Thank you very much. It's an honor for me. Mansoor told me that I have only 12 minutes and I already lost 2 minutes perhaps. Thank you very much. I'm so glad and I'm also very nervous because it is very difficult to speak in front of my teacher and very senior colleague, Dr. Atta Rahman. So I learned a lot from him, no doubt. So NEEM has been a passion to me for a long time. Soon after completing my PhD, my mentor and the founding director of the institute, Professor Dr. Salim Ahmad Siddiqui, he wanted me to continue and he assigned this plant as one of the plants on which I should work. We work a lot, produce a number of PhDs, produce a large number of papers. But one thing always bothered me, that what we have given to the masses. So almost 2 decades now that I started thinking in this direction. So alhamdulillah, we got some very good colleagues from zoology department. Professor Dr. Naeem Nafi Sahab, late. He was a very sincere and serious worker. So in collaboration with him, we tried to develop some biopesticides from neem because neem is known to have marvelous activities in this direction. So we got in different directions, but because we don't have much time, I will not go into the details of these things. We have studied almost every part of this fantastic tree. Major compounds, classes of compounds are listed over here, which we isolated and determined their structures and activities. We also worked out and discovered what are the distribution of triterpenoids in various parts, where they are present in larger quantities. We also developed structure and activity relationship. This is chemistry, and obviously I will not go into the details of these things. So I would like to come towards the products which we have developed. These include a biopesticide which we name Biosal for use in agriculture. Other is Nemosal, a little bit different from Biosal. This is for horticulture. BioAman is another bioinsecticide for household use. And biofertilizers, which is we call Neem Plus, from pesticide and herbicidal product Nemectin, which is a cream or spray for livestock and mosquito repellent. These are the moleys. Everyone would be knowing that. So this is the bottle of Biosal. It is a emulsified fire-bit concentrate prepared by solvent extraction of neem seeds and formulating water in a matter to give water-miscible pesticide. It has been found effective in protecting crops like cotton, very effective. Rice, vegetables, including several vegetables like bean, tomato, etc., as well as of fruit trees like mango, coconut, palm, guava, zappers. These are the fields where we work. After conducting studies in the lab scale, we contacted various agencies and we were able to go to their fields to try our products. So this is in Bhavatpur. Again, these cotton seed fields after treatment with Biosal. Nemosal is another product. This is for horticulture. This is a milder one and this can be used in household plants. This is the results of Nemosal. So the observations were best results are obtained on prophylactic use, that is cure before infestation. Cure before infestation. If the plants, like for example of cotton, they are treated before an infestation is observed, and regular use of these pesticides is done. Treatment by 1% concentration is effective. 1 liter Biosal is enough for 1 acre area, and it has pronounced effect after 72 hours of treatment. Advantages is that resistance does not develop against these pesticides. Otherwise, the other pesticides, which are synthetic pesticides, they have resistance after some time. No toxic residues are left as they are biodegradable. Only negligible quantities of solvent is left in the final space. I cannot read it hence. These have pollution-free products and environment friendly. IGR, in fact, growth regulator, they are the IGR, antioxidant and repellent effects, and show repellent effects. Bioman is another product. This is for household pest control, for mosquitoes, for example, and cockroaches. Neem Plus is a pesticide. It's not moving. Neem Plus is a processed neem seeds product, enhances the growth of plants and fruit yields, and protects the plants from pests and nematodes. Like other neem products, it is also environment friendly. It is spread in the beds. They subject. These are some pictures of the field trials. Limectin, this was also very interesting to note that limectin is very good germicide which is used for animals, equine haggis. So several years before we found a goat in our institute. You can see the picture on the left hand side here. So sorry, I'm sorry. How can we put that? That's a pointer. Pointer. Yes. Yeah. So this is, you can see the skin of a goat, very much affected by some insects. This is also the skin of the goat. So I, along with my colleague, Dr. Asana, Dr. Saab, you know very well. So this was just like a joke, and we started working on that. And we took the services of a sweeper and asked him to apply our product on the skin. So now you can see and the effect after 1 month. You see, nobody would believe that is in the same effect. So there are many problems in promoting this product because the greatest difficulty is encountered in this application for pest control because the farmers are not aware. However, we conducted some farmer field schools training in Bhagalpur, and farmers were trained with these pesticides to know what pesticides are, how do they act, how much safer they are, and what benefits they may provide. So conclusion was that in spite of the difficulties encountered in development on large scale use of pesticide, the scientists all over the world are enthusiastic and optimistic about the utilization of biopesticides as a source of sustainability and safe pesticides. These efforts have culminated in the development of many products which are successfully used in several countries. A backup support and participation of government agencies, industry people, and also NGOs is required to make use of these materials viable in the real sense. There is a need to create awareness in the masses, to take steps for the protection of already existing plants and trees, plantation of new trees, proper harvesting and processing of the seeds, and so on. I will skip this. So actually, I missed the real actual presentation because there I have included the use of this pesticide against locust sometimes back. I think it was in 2021. There was a very severe attack of locust on the trees, on different vegetable trees. So some people tried and my product Biosal on their field and found it very effective. So I have included those pictures in another actually presentation which is not included here. So this was run on this information was run on the TVs and also published in newspapers, in Urdu newspapers and English newspapers, but no government agency reacted on that. So this is the dilemma. So now the present status is that we developed a small private plant unit in this institute, and there we can produce, say, about 1,000 liters per day on a small scale. It is small scale, but can be translated to a larger one. And we have registered this product with the government plant protection department of the government, which is what it is process. So it is now product. It is now registered and also patented. And now Dr. Mansoor is also helping me a lot, and we are now proceeding towards technology transfer. Inshallah, I hope it will be soon. Okay, thank you very much for your time.
Yeah, are the active principle in basic lately, or are there some other also based?
Yes, yeah, Doctor, very good question, Rosa. Actually, it is considered that as a directive is the only active ingredient, and it has been for long realized, believed that this is the only compound, and the other what I should say that the people who deterred from using neem as a biopesticide was that as the directive is not very stable. It is deteriorated very soon. But our studies are that because we isolated hundreds of new compounds, and we dozens of students were awarded PhD on this one. So azathioprine is not the only active responder. There are many other triterpenoids which are active. So this is active. Therefore, this product is quite active. We gave some product to somebody with the reference to Professor Dr. Shahid Mansoor to work to see its effect in Balochistan cotton. It was found very effective, and the product was 2 or 3 years old at room temperature. So it is quite stable.
Thank you.
Thank you. I just like one thing. I'm very thankful to Madam because she is my teacher, and I'm learning a lot from her. I mean, because all the extraction that was done from the plant that I am using in my TDF project, I mean, we are using all facilities, and also Madam has extended her full support for pilot production, scale production, and commercialization. I hope that this product will be commercialized very soon from and under the leadership of our director. I hope that we will have some agreements very soon. We are actually finalizing the agreement for transfer of technology because large scale production is not possible at the institute level. So I'm sorry, Madam, कि आपको मैंने जरा ज़्यादा कह दिया था. So, but she is helping a lot, and actually we are helping each other.
Yes.
So there were some issues with the renewal of license from DPP, so we went there and then it was done. So thank you, sir.
Thank you. Thank you. Dr. Mansoor has been very cooperative and very helpful, and is helping a lot in taking this product towards development and market. Thank you, Dr. Mansoor.
Thank you so much, Dr. Veena, for your valuable presentation. Ladies and gentlemen, it is now my pleasure to introduce Professor Dr. Mohammad Kashif Riaz Khan, Head of the Plant Breeding and Genetics Division at NIBE and PAC. He is a distinguished crop geneticist and an award-winning scientist who has made significant contributions to Pakistan cotton breeding research. May I now invite him to deliver his talk.
14 minutes.
Respected Chair, distinguished Chief Guests, my esteemed colleagues, ladies and gentlemen, Assalamu alaikum. It really gives me immense pleasure to be here, and I'm very thankful to the organizers, Director iccbs, Dr. Shahid Mansoor, and especially the Dr. Razaul Rahman. They give me a chance to speak here. So, the title of my talk is Breeding Cotton for Resilience and Quality. Okay, as we know that the cotton is the pivotal point of our economy, especially in Pakistan, and it contributes a lot in the GDP. And for the last decade, if we see that the production is declining. Under this scenario, we have to identify the new areas where we can grow the cotton. Because the older areas of cotton grown has not been suitable for cotton growing. Okay, especially the current challenges in cotton production is mainly the abiotic stresses that we talk about, the drought, heat, and also the salinity is now becoming a big challenge, especially in lower Punjab and also the parts of Balochistan as well. So heavy metals and waterlogging are the major cause. And because of these challenges, the germination effects and photosynthesis ultimately leads to the lower production of cotton. Why breeding must evolve? If we look at the previous approaches in breeding, that was very simple. High yielding, superior fiber quality, and the disease resistance. Now the future breeding objectives has been modified as climate resilience, especially the heat, drought, and salinity, if we talk about the Pakistan scenario, and the fiber quality, or for the industrial perspective, especially in the early maturing. Early maturing varieties are very good enough because we can escape the heat stresses and also the effect of later generations insect attack like pink boll. And resistance to the biotic and especially the biotic stresses CLCV, cotton leaf curl disease, and by their adaptability and adaptation to the sustainable production system. This is very important. So breeding for stress resistance, for resilience and quality. If we look at that, there's conventional breeding deals with the introduction, selection, or we can do some mutations and hybridizations. Ultimately, it is a time-consuming, laborious, and rely on visual observations. Normally, if we are going to breed a new variety, it took about 12 to 15 years. So we have to jump into a new era of genetic analysis and the modern breeding approaches. The molecular breeding, genomic selections, and speed breeding. Now we have going to establish a speed breeding setup in PAEC at NIAB, and the chambers will be available for speed breeding. And we got a PSTP project on it, and we are focusing the three major crops in this speed breeding setup, that is cotton, rice, and wheat, and particularly for hybrid cotton, hybrid rice, and hybrid wheat as well. So the advantages of these is they are timely, efficient, precise, and based on market trait association. So ultimately, the genome editing is also helping us a lot in introducing the new allelism for the resistance, whether it's a abiotic or it's a biotic stresses. So sustainable production can be achieved through practically, if we go with modern breeding, genetics, genomics, and omics. They should be interlinked, or we can move ahead together with all these techniques in the resilience and high yielding breeding for sustainable production. This is resource efficient and environment friendly. This symposium especially will talk about the organic cotton, regenerative agriculture, and climate resilience practices. So in my next slides, I will discuss this in detail. So ABI's PAEC contributions to the climate resilience cotton breeding. Stress tolerance studies, we identified a lot of germ flowers which showed better osmolyte accumulation and antioxidant activities because of these. They behave like a salt tolerant. In the market rate association, we identified the 125 significant MTAs for the salt tolerant traits, and also the key chromosomes. They are involved, chromosome number 9, 12, and 22 are linked with the salt tolerance. And we also identified the 874 genes identified which are associated in the genomic region. So we identified the 6-7 major genes which can be targeted through genome editing or other advanced techniques, and they are available in our population. We have a lot of germ flowers from China as well under our last MOU, and we got so many genetic resources from other countries as well. Under ACARDA, we got good germ flowers from USDA and others. So heat stress studies, likewise the salt tolerance, we identify a lot of germ flowers which have potential for the heat tolerance. And you know that all over the world, if we look at the world scenario, Pakistan has the most heat tolerant germ flowers. China is also looking for us for heat tolerance. And also the US also looking for heat tolerance is really good in Pakistan germ flower, especially available with NAIA, NIPG, and NIA. You see, we are dealing with the mutations, and we create series of allelism through mutations, and we found number of alleles for heat tolerance. Drought is very important. Drought is a very big term, but I would like to say it's a limited water, we can say that. Because if we look at in the natural conditions, drought is very difficult to evaluate. So limited water is especially for Balochistan, we have to breed such varieties which can withstand limited water conditions. And luckily, we found a number of genes which are associated with the this drought tolerant trait, and we chromosome 19 is the basis of genome which are most of the genes are available on the chromosome number 9, that is D6. NIA also developed, NIA Nuri is also cultivating largely in Balochistan as well. And with the collaboration, I am very thankful to the Dr. Shahid Mansoor. He helped us to introduce our cotton varieties in Balochistan, especially for organic cotton. This is a climate smart cotton variety recently released by NAIA. It has a high yield potential, about 61 mands per acre. If we follow if we provide the optimal conditions, and also the tolerant to salt, drought, and heat all together. And it is very difficult to trickle down all these three traits into a single genotype because some are negatively correlated, and it is very difficult to break that linkage. But luckily, we are able to break that linkages through mutations. So perform well under stress conditions and fine fiber quality. And also, this is another our breeding line, upcoming line, and in the next Punjab Seed Council meeting, it will be released as a variety. And I hope and I think that this variety will have a very good potential, and maybe we will have an era of NIOP 78 again because of its resistance to the CLCUD. It's moderately resistant to the cotton leaf curl disease and fine fiber quality and wider adaptability. You can grow it from the mid of the Punjab, South Punjab, Sindh, Balochistan, and even some parts of the Dera Ismail Khan. This variety throughout the Pakistan played a very good yield potential. Oh, this is we did an experiment in the South Punjab, the new Chapu area, Bahawalpur, is going to be developed under development stage with the Green Pakistan initiative. And you see the performance of NIOB 878, 512 is much higher than that of the even triple gene and with the double gene and SS32. And SS32 is famous in most of the South Punjab and even some parts of the Balochistan as well. So it's even high yielded than that. So NIPG also produce a lot of germ flower with double genes and triple genes and under evaluations, and there are certain events are available, and we have an infrastructure of omics platform at NIOB, and we have a lot of genetic resources. Organic cotton is a chemical free and others, everything, everybody knows that. I come to the NIOB Kiran. This variety is about covering 70% organic cotton area in Balochistan, and it has a high yield potential. And we went there with Dr. Shahid Mansoor at Hosdar and Barkhan, and this genetic factory is dedicated for organic cotton only. And also the seed purity is also important. Seed is not only a seed parameter, but it's also a market promise. So breeding target should be devised. We should develop the varieties which can withstand with the promises of farmers, ginners, and textile industry. So we have to shift our breeding objectives according to all these scenarios, and the quality target is maintain genetic identity from the breeder seed to the certified seed. This is a major problem of failure of one of the major problems of crop failure, cotton crop failure, because of adulterated seed is available in all around the Pakistan. So that, and also if we want to ensure the seed quality, we must follow these steps: the field selections, isolation, history, androgen. And the last years, we went with the Dr. Shahid Mansoor, and we physically drove out the plants in Balochistan, and we purified the seed genetically, and the iccbs, they ensure the purity, germination, vigor, and GMO traits validation. And traceability is very important. Nowadays, Europe and everywhere, they always demand if it is an organic cotton product, there should be a traceability. So there should be a barcodes and others, and even you can just scan a barcode and they will tell you from which field it's coming out and from which farmer they are producing. The traceability is very important, and the seed treatment, that must be a biological source and approved treatments. So industry perspective on the success factors and barriers that always drive with the market pull, farmer economics. Why farmer grow cotton? If we give the premium, low input cost, and risk sharing drive adoption, then the adoption of the farmers in enhance. And also the seed integrity and regularity, clarity must be there, and extension capacity of our farmers and others and the local growers. We have to train them well, so to maintain the integrity of the seed. We have also developed the brown cotton, and that was presented to the DG International Atomic Energy Agency. It's a naturally mutant of brown cotton, and staple length is improved, and strength has been improved, and soon it will be released as a variety. I will simplify this. If we cross the Barbadans is a quality, 36 millimeter. 36 millimeter. In the Hirsutum is up to 30 to 30 millimeter. If we even cross them, and in F2 generations, when the segregation starts, the Barbadans chromosomes and the Hirsutum chromosomes, they divert from each other, and we will not get the introgression of the Barbadans into the Hirsutum. There are series of alleles which are responsible for non-homologous pairing. They inhibit homologous pairings. So it can be done by two techniques, either through mutations or through gene editing, and there are a series of these alleles which can be gene edited, and we can better performing Hirsutum with the high introgression part of the Barbadan. And we did it. We have made a cross, and this is a Barbadans cross with Hirsutum, and it's in advancing, and the fiber length is near 35. But it's under trial. Future climate smart breeding, multi-omics integration, the speed breeding and genome editing, and the fiber quality improvement, and the sustainable cultivars and advanced seed system must be there. And if we अगर वो दूसरे में बंद करने की करें तो इस एक स्लाइड से कि हमें रेजिस्टेंस वाइट सोर्स से भी लेनी पड़ेगी, हमें मल्टीओमिक्स को ऑप्ट करना पड़ेगा, हमें नेचुरल ब्राउन कॉटन्स या ब्लू या दूसरे कलर्स पे जाना पड़ेगा, एंड एआई स्पीड ब्रीडिंग जो है, एआई स्पीड ब्रीडिंग, we have to integrate this, and also the hybrid seed production in the early generations and the mechanisms. And this is the take-home message. The breeding priorities must shift not only the yield, but we have our resilience and quality as well, sustainability, wider adaptability, and modern breeding tools we have to adopt. And ultimately, the sustainable production we can achieve through this integration of genetics and seed quality, and ultimately innovation and collaboration will drive the next generation of cotton improvement in Nayab and beyond. Thank you.
Thank you so much, sir, for the valuable presentation. Ladies and gentlemen, it is my pleasure to introduce Dr. Nizamuddin Gapar, Head of the Soil and Environmental Sciences Division at Tandojam. He is a distinguished scientist with remarkable contributions to sustainable agriculture and soil health management, and a recipient of the PAC Gold Medal and Distinguished Excellence Gold Medal. May I now request him to deliver his talk.
Respected chair of today's event, and it's my privilege to admire Dr. Attar Rahman Sahab, who is the proud of Pakistan scientific community and a winner of the highest award of scientific community. Thank you so much, sir. And ladies and gentlemen, today I am going to introduce about the contribution of Pakistan Atomic Energy Commission in agriculture technology through biocontrol technologies and biofortification for sustainable organic and regenerative agriculture. Sir, the mandate of ABIs is the application of nuclear technology in food and agriculture, research-based development of agriculture sector, introduction of the transfer of technology towards the farming community, and then socio-economic stability and the food security. And Pakistan Atomic Commission is providing analytical advisory service to the farmer, growers, fertilizer community, and other stakeholders, and human resource development. Sir, in PAC, having the 4 agriculture research institutes, first agriculture research institute was established in Tandojam in 1962, naming then Pakistan Atomic Energy Agriculture Research Center. Then in 1998 was renamed as the Nuclear Institute. Renamed a Nuclear Institute of Agriculture. And after interval of 10 years, PAC established our second agriculture research institute in Faisalabad with name Nuclear Institute for Food and for Agriculture and Biology. And after the interval of 10 years, again, Pakistan Atomic Energy Commission established the third agriculture research institute in the name of Nuclear Institute for Food and Agriculture, NIFA, at Peshawar Tanab Road. And after 12 years period, Pakistan Atomic Energy Commission established the NIBG, National Institute for Biotechnology and Genetic Engineering, at Faisalabad. Sir, this is the basic strategies to improve the agriculture sector. First, the development of new crop varieties, which are the high yielding and better quality, abiotic stress and biotic stress tolerant, wide adaptability, and marginal or salt affected lands for agriculture. These soils may be the drought, salinity, and waterlogged, and the aim is to increase the cropping area. And then the third is the improved water and fertilizer management through improved irrigation techniques, development of biofertilizers, and the plant nutrient management strategies in soils. And the fourth strategy of ABIs is the pre and post-harvest losses. These are the strategies for reduced losses to the pathogens and integrated pest management technology, biocontrol programs. And the fifth strategy of ABIs is the animal reproduction and health, and then further disease diagnostic treatment, feed and feed supplement, and the synchronized breeding of animals. And the sixth strategy is the improved export potential, that is the removal of contaminants from the export commodities, analytical service to the exporters, GMO testing, etc. So this is the first institute established in 1962. So this institute is contributing in national agriculture and biotechnology, as this institute has developed 41 crop varieties for general cultivation in the national provinces, particularly in the Sindh province, and now more 5 varieties have been released for general cultivation in the Sindh province. So this is the seed production of this institute contribution in the farming community of the Sindh province. We are every year producing the highest early generation seed, pre-basic seed, near to the 100 tons for the farming community. And we have also signed MOUs with more than 20 private companies and some public institutions. So there is the biocontrol technology, especially in Sindh. We have established trichogramma colonies and for the field release. So trichogramma colonies for the biocontrol of the borers, sugarcane, top, medium, and the root borers. So we have two products. Product first is the fresh parasitized parts, which can be controlled or treated within 4 to 6 days in the field crops. If we are going to treat early as compared to 4 to 6 days, then we have the mature parasitized cars, which can treat on the same day after application. So then in another we have product of NIA, Kerasupella carnia parts. These are the cards which can be released in the field for control of the insect pests, especially sucking complex. So 15 cards per normal infested area, approximately 250 to 30 eggs per card is recommended for field application in the crops. So sir, we have the fruit fly traps to control the fruit fly in fruits, particularly in guava, in mango trees. So we are using the methyl eugenol in traps. So another, so we have the biofertilizers, NIA BioBooster, for the field application of growers. And this product contains 30% organic matter and 80 to 20% phytophile, which is nearly replacement of the DAP in the field. So we have the NIA, the first product of NIA for the field community. This is the foliar fertilizer, sir. Then we have the phosphorus enriched compost for the farming community. Then we are providing analytical services to the farming communities, and this to control the adulteration of fertilizer in the market. This is the Nuclear Institute for Agricultural Biology, Riyadh, at Faisalabad, and they are producing the NAM phosphate fertilizer. That is a very good initiative for the farming communities. And the production capacity of this is 100,000 to 1,500 tons per year for the farming community. This is the NIA compost for farming communities. And this is the NIA HS vaccine, hemorrhagic septicemia, for the animals to control the HS disease. Now, sir, the NIFA. NIFA is developing a rapid test kits for fortification of vitamins. And then NIFA also preparing the rapid testing kits for different. And then some NIFA dengue guard, which is used for to control the dengue in the humans. Dengue is marketed locally. So then we have the NIFA fruit fly traps for the fruit orchards in the KPK to control the fruit fly in fruit trees. And then sir, NIPG is contributing in national agriculture as by providing the biofertilizers, super biopower, and also the phosphorus pool and save urea and DAP 20% to each and increase the soil fertility and sustainable crop productivity. PAC also putting the tricho shields, fungal biopesticides, trichoderma-based product for enhance the yield and protection of the agriculture pests in the field, sir. NIDI is having the technology floating treatment wetlands to control the wastewater and treated, that is the pressure resource for field. This is the countrywide application of floodland technology. This is a foot and mouth FMD vaccine to control the foot and mouth disease in animals. So this is the way forward. Way forward is the development of crop varieties for high yielding, disease resistant, climate resilient cotton, wheat, rice, soybean, brassica using genome editing or genome selection or genetic multiple techniques. And then the seed production of early generation seeds, that is a pre-basic to meet the seed demand of private companies and farming community, and development and scaling up of the biofertilizers, biopesticides to support sustainable organic and regenerative agriculture. And then, sir, biocontrol technology, different biocasts are prepared in different institutes to control the fruit flies, sucking complex insects in the field crops. And also we are promoting application of wastewater treatment technology. There's new industry wastewater, and scaling of production of HS and FMD vaccine for animal production could be a good sign for the animal keeping growth, and analytical services for the soil, water, plant, biofertilizer for farmers and other stakeholders, and the commercialization, that is the linking of ABI's commercial service to the Pakistan, single window, ETOP, connected, SciTech, etc. Thank you so much.
Assalamu alaikum. I'm Dr. Adya from Genome Center, and I am going to announce our next speaker is Dr. Farooq Ahmed Khan, Assistant Professor at iccbs. Dr. Khan earned his master's degree in fine chemistry from France, completed his PhD in Switzerland, and pursued postdoctoral research in Canada. His research focuses on advanced organic synthesis and nanocatalysts. So please welcome Dr. Farooq Ahmed Khan to deliver his presentation on linking synthetic chemistry with pest control.
Thank you very much, Dr. Shad Mansoor, for giving me opportunity to present my work here in this symposium. So before delving deep into our presentation, let me show you a video which is made by my student, Mr. Faizan Kahar. He has spent weeks to summarize the work which we are doing here in our lab at iccbs.
शाम ढलते ही मादा पिंक बॉलवर्म एक खास खुशबूनुमा कीमियाई सिग्नल फेरोमोन हवा में खारिज करना शुरू करती है। चूंकि ये फेरोमोन आम आंख से नजर नहीं आता, इसलिए इस वीडियो में इसे हल्की जर्द धुंध की शक्ल में दिखाया गया है ताकि आप इसे आसानी से समझ सकें। नर पिंक बॉलवर्म अपने निहायत हसास एंटीना की मदद से इस खुशबू को काफी दूर से महसूस कर लेता है। फिर वो इसी खुशबू के रास्ते पर चलते हुए सीधा मादा की तरफ परवाज करता है। जब नर मादा तक पहुंच जाता है तो दोनों के दरमियान मिलाप का अमल शुरू होता है। कामयाब मिलाप के बाद ही इस नस्ल का सिलसिला आगे बढ़ता है। मिलाप के बाद मादा एक मुनासिब कपास की डोरी तलाश करती है जहां वो अपने अंडे महफूज तरीके से रख सके। वो बड़ी एहतियात से डोरी में एक छोटा सा सुराख बनाती है ताकि उसकी आने वाली नस्ल अंदर महफूज तरीके से नशोनुमा पा सके। इसके बाद वो नन्हे नन्हे अंडे कपास के बीज के अंदर रख देती है जहां बाद में सुंडियां निकलती हैं और बाहर के माहौल से महफूज रहती हैं। ये सुंडियां बीज के अंदर ही खुराक हासिल करती हैं, जिसकी वजह से कपास की कुदरती नशोनुमा मुतासिर होती है, पैदावार कम हो जाती है और फाइबर का मयार भी खराब हो जाता है। इस तरह कपास की डोरी के अंदर छुपी हुई पिंक बॉलवर्म की नई नस्ल अपनी जिंदगी का आगाज करती है और यही जिंदगी का चक्कर दुनिया भर में कपास की फसल के लिए मुसलसल एक बड़ा मसला बना रहता है। अब आइए देखते हैं कि डेल्टा ट्रैप किस तरह इस मसले का हल पेश करता है। डेल्टा ट्रैप के अंदर एक खास पॉलीमर डिस्पेंसर लगा होता है जो बिल्कुल वही मसनूई फेरोमोन खारिज करता है जो मादा पिंक बॉलवर्म कुदरती तौर पर खारिज करती है। नर अपनी हसास एंटीना की मदद से इस मसनूई खुशबू को भी महसूस कर लेता है और इसके मंबा की तलाश में निकल पड़ता है। लेकिन जब खेत में एक तरफ असली मादा और दूसरी तरफ डेल्टा ट्रैप दोनों फेरोमोन खारिज कर रहे होते हैं तो नर उलझन का शिकार हो जाता है। वो मस्नूई फेरोमोन को ही असली मादा समझ लेता है और उसी खुशबू का पीछा करते हुए डेल्टा ट्रैप के अंदर दाखिल हो जाता है। ट्रैप के अंदर मौजूद चिपकने वाली सतह उसे फौरन अपनी गिरफ्त में ले लेती है और वो बाहर नहीं निकल पाता। इधर मादा नर को अपनी तरफ बुलाती रहती है, लेकिन कोई नर उस तक नहीं पहुंचता। जब मिलाप ही नहीं होता तो मादा के अंडे बराबर नहीं होते और वो कपास की डोडी के अंदर अंडे नहीं दे पाती। नतीजतन सुंडियां पैदा नहीं होती, कपास के बीज और फाइबर मुकम्मल तौर पर महफूज रहते हैं और डोडी को किसी किस्म का नुकसान नहीं पहुंचता। इस तरह मस्नूई फेरोमोन के जरिए नर और मादा के मिलाप को रोका जाता है, जिससे पिंक बोलवर्म की तादाद में नुमाया कमी आती है। ये एक माहौल दोस्त तरीका है जो कपास की फसल को महफूज रखता है, फाइबर का मियार बेहतर बनाता है और मजमूई पैदावार में इजाफा करता है।
So Mr. Faizan Tahir has effectively summarized the work which we are doing in our lab. Pest, as we know, cause significant damage to crops worldwide, as you can see from these pictures. And Pakistan saw a severe decline in cotton output during 2024, with production falling by approximately 34% compared to the previous year. To make up for the shortfall, Pakistan imported cotton worth approximately $2.5 billion. So, in this backdrop, Dr. Shahid Mansoor came to my lab and suggested to do the synthesis of pink ball pheromone. And you can see how this insect damaged the cotton in Pakistan. Traditional insecticides being used have serious drawbacks like insect resistance, as we know that pesticides are causing a gradual insect resistance. Similarly, destruction of natural predators like pollinators is the result of traditional insecticides. Then high cost and financial burden to farmers because most of the insecticides are being imported in this country, environmental and water contamination due to rain and irrigation runoffs, the pesticides go into the water and make it poisonous. Health risks to laborers and consumers, these are some serious drawbacks, but the most important drawback is export rejection due to chemical residues. Because international markets, particularly European Union, US, and Gulf countries enforce strict regulations for maximum residue limit for pesticides. So what is the solution? Well, sustainable alternatives include pheromone-based technologies which need to be adopted in Pakistan, just like in Europe, in Canada, in Australia, and US. So, like Faizan has effectively summarized mating disruption technology in his video, I will not go in details of this. So, there is another lure and kill strategy that also uses a similar type of strategy to attract the insects with the help of pheromones. So, Dr. Shahid Mansoor kindly provided me a product of Japan, pink ball pheromone, which was made by Shinitsu, very famous chemical producing company, which is producing pheromone and supplying it worldwide. And in Pakistan, it was imported by government of Pakistan in 2019 to analyze how it affects the insects in the cotton fields. But due to high cost, it was never imported again in Pakistan. So I gave this product to my students to analyze it chromatographically and spectroscopically. My two students, Sana Sheikh and Mr. Kamran, they have analyzed and found that this Japanese product contained a mixture of these two isomers. So then there is synthesis. We synthesized this pheromone in our lab. It was done in 13 steps. These students were involved at different stages for the synthesis of this pheromone in our lab. Initial assessment of this pheromone was done by Dr. Shahid Mansoor in our biotechnology wing. Initially, we miserably failed, but we quickly learned that these two pheromones are basically a blend of cis and trans isomers. They need to be mixed in certain ratio. And then our improved pheromone was sent for field trial to Nuclear Institute of Agriculture, Tandojam. This is the certificate I recently received from Nuclear Institute of Tandojam, which clearly shows that our developed pheromone has superior trapping efficiency, and it is recommended for the control of pink bollworm in the cotton fields of Pakistan. And then in a second field trial for this pheromone, which was synthesized in our lab, it was sent to Nuclear Institute of Biotechnology and Genetic Engineering by Dr. Shahid Mansoor. And we received this certificate from NICGI, which clearly shows that the locally synthesized pheromone, which was developed by our lab in iccbs, has a superior efficacy compared to the imported one. So, what is making disruption? As we see, this pheromone basically helps to confuse the signal of female pink bollworm. Therefore, male insects get confused and they forget to reach to the female insect, and then they move around the synthetic pheromone dispenser. And this is how we can alter the population of pink ball insects in the cotton field. Advantages of pheromone-based technologies. There are a lot of advantages, like they are environmental friendly, safe for human, wildlife, and pollinators, no chemical residue are produced, and there's no risk of pest resistance development. Similarly, these pheromones are highly specific. They target only pest moth species. But there are some limitations and challenges. For example, high cost of pheromone. This is the biggest challenge. Separate formulation for each pest is needed. And the biggest challenge, we need advanced synthesis and catalysis scale, which is must to develop pheromone technology in this country to synthesize the pheromones. We see in the developed world, like in Netherlands, we have a company, Pherobank, which is developing pheromones and this type of technologies for the agriculture sector of this country. Similarly, in Spain, we have Econex. In UK, we have Russell IPM, which is developing pheromones for the agriculture department, agriculture of the country. In USA, there are a lot of companies like ProVivi, Teresa, and ISCA. They are developing the pheromones and similar technologies for the agriculture sector of United States. In China, we have Ferrobio. In France, we have Sotera and Bioprox. Whereas in Japan, we have Shinitsu. In Australia, we have Agnova. In New Zealand, we have Grochem. Whereas in Canada, Endermet is developing pheromones for the agriculture sector of that country. But in Pakistan, we don't have anything. Why? Because we have to develop advanced synthesis and catalysis skill. Without this skill, probably we cannot develop pheromones in this country, and we cannot move forward. We cannot move beyond spraying the insecticides and pesticides in our fields. Apart from pink bollworm pheromone, we also developed whitefly lures in our lab. Whiteflies, in fact, inflict severe damage on Pakistan cotton production, causing estimated 20 to 40% yield losses almost every year. And Dr. Shahid Mansoor provided me a product from UK, and this was developed by the company in UK. We have checked it in Pakistan. It works effectively. And what we did the reverse engineering of this product, and we developed our own product. So this product is undergoing field trials at different institutes, at NIA in Tandojam, in NICGI at Faisalabad, and NIAP in Faisalabad. Similarly, we also developed allicin spray in our lab, and Dr. Shahid Mansoor has tested our allicin spray and found that this spray is effective for wheat aphids, for fall armyworm, which gives a serious damage to maize, and then for nematodes. Recently, we had a cry for help from northern areas of Pakistan. Director of Agriculture Department from Skardu region has contacted me and asked to help because there are some pests which are wreaking havoc in those areas. Pesticides are not effective since few years, and there is a serious trouble from codling moth. Codling moth is a major pest for apples and pears in those northern areas, and we are working to develop the pheromones for these for this moth. Dr. Shahid Mansoor and my student, Mr. Shujat Ali, and the Director Agriculture of Northern Areas, Mr. Raza Ali, are helping us to develop these pheromones. Similarly, there's another big trouble in the northern areas of Pakistan, that is oriental fruit moths. This is major pest for peach, apricot, and plums. They are damaging our fruits and resulting in the loss of production. We are also trying to develop pheromones, and we are synthesizing. We are in midway, and we hope that we will be able to synthesize the pheromones for these moths and then start the field trials for our developed pheromones in the northern areas of Pakistan. So there are some conclusions. Pheromone-based lures are vital for monitoring and mass trapping. Mating disruption is highly specific, environment-friendly, and effective tactic that reduces mating success by saturating the environment with synthetic pheromone. Together, they form a powerful core strategy for sustainable management of damaging the pest. Similarly, while challenges exist, for example, we have a serious synthesis gap, skill gap for advanced sciences in this country. The benefit, makes them essential tool for modern integrated pest management. And if you have any questions, sorry for taking a little bit more time.
The major challenge about the cost effectiveness, and the big issue actually arises when you're comparing the parts with the Chinese one. So, have you focused about the industrial production in terms of the cost effectiveness in comparison with China?
Well, initially we have to develop it at a large scale, and these chemicals doesn't require tons of production, because small quantities of these chemicals are highly effective. For example, 1 gram of pheromone will be sufficient for 1 acre of cotton field. So 1 gram, so it means with 1 kg, we can cover around 1,000 acres of cotton. So this is small quantity, high value product. But the biggest problem is that we don't have a skill in this country. I'm sorry to say, we cannot find somebody who has courage and energy to work in the lab for days and nights, and after years can produce some results. This is the problem. So industrial question will come later on, because we need to develop processes. And of course, the product is quite expensive. Like I told you, particular law, the pheromone was imported by government of Pakistan from Japan. Shingitsu prepared it. It was imported in 2019. Afterwards, it wasn't imported. It wasn't imported in the country. It was expensive, and it was quite hard to expensive. But it has promising results. Therefore, Dr. Shahid Mansoor brought me, samples of the imported product. We analyzed it, and then we made it copy of that one.
Thank you so much, sir, for your valuable presentation. Ladies and gentlemen, it is now my great honor to introduce our esteemed chief guest, Dr. Shakil Abbas Rufi. Dr. Rufi is a distinguished nuclear physicist and currently serves as a member at the Pakistan Atomic Energy Commission. He has served in key leadership positions at SPD and GHQ and has represented Pakistan at numerous international scientific forums. He holds a PhD in shock dynamics from the Beijing Institute of Technology and is a recipient of the prestigious President Pride of Performance Award. May I request him to kindly come on the stage and address the audience, please.
औज़ुबिल्लाहिमिनश्शैतानिर्रजीम बिस्मिल्लाहिर्रहमानिर्रहीम काबिल सद एहतराम डॉक्टर प्रोफेसर अताउर रहमान निशान इम्तियाज़ हिलाल इम्तियाज़ सितारे इम्तियाज़ तमगा इम्तियाज़ पैटर्न इन चीफ ऑफ ICCBS मोहतरम प्रोफेसर डॉक्टर एम रज़ा शाह डायरेक्टर ICCBS तमगा इम्तियाज़ डॉक्टर शाहिद मंसूर एक्स डायरेक्टर निबजी प्रोफेसर सैयद गुलाम मुस्तफ़ा मेंबर ऑर्गेनाइजिंग कमेटी साइंटिस्ट और मेरे दोस्तों और ख़वातीन हज़रात अस्सलाम वालेकुम। ये मतलब मेरी किस्मत है कि मैं आपके और चाय के दरमियान हायल हो गया हूँ। मैं सिर्फ 5 मिनट लूंगा आपके और इनफॉर्मल तरीके से कोशिश करूंगा कि आप तक अपना पैगाम पहुंचा सकूं। मेरे लिए एक बहुत बड़ी इफ्तिखार की बात है कि ये 10 महीने पहले मैंने मेंबर साइंस का तौर पे ओहदा संभाला है। एटॉमिक एनर्जी कमीशन में 7 टेक्निकल मेंबर्स हैं और हर एक अपने अपने शोबे को देख रहा होता है। तो लकीली मेंबर साइंस के अंडर एग्री बायो इंस्टीट्यूट्स भी आते हैं, 21 कैंसर हॉस्पिटल्स भी आते हैं जो इस वक्त पूरे अक्रॉस ईस्ट टू वेस्ट और नॉर्थ टू साउथ 21 इंस्टीट्यूट्स हैं और ये तकरीबन 80% कैंसर का बोझ मुल्क का उठा रहे हैं। हर साल 10 लाख पेशेंट्स हम डील कर रहे हैं और पाकिस्तान का 80% कैंसर लोड इस वक्त एटॉमिक एनर्जी कमीशन पे। इसके अलावा लेजर, क्वांटम मैकेनिक्स, क्वांटम कंप्यूटिंग, पाकिस्तान के 3 रिसर्च रिएक्टर्स जो पूरे पाकिस्तान को इस वक्त रेडियो आइसोटोप्स फराहम कर रहे हैं। इसके अलावा इंटरनेशनल लथिया गली समर कॉलेज है, सर्न की एक्टिविटीज है, IAEA की एक्टिविटी सारी मेंबर साइंस के अंडर आती हैं। इसके अलावा पाकिस्तान के जितने भी 6 न्यूक्लियर पावर प्लांट्स हैं मेंबर पावर डील करते हैं और अल्हम्दुलिल्लाह पाकिस्तान का तकरीबन 20% इलेक्ट्रिकल लोड पाकिस्तान एटॉमिक एनर्जी कमीशन प्रोवाइड कर रहा है। और 5वां रिएक्टर C5 के नाम से जो पाकिस्तान का 7th रिएक्टर होगा इंशाल्लाह वो भी लग रहा है और उसके बाद मुजफ्फरगढ़ में भी M1 और M2 लगेंगे। तो ये बहुत बड़ा पाकिस्तान एटॉमिक एनर्जी कमीशन का पोर्टफोलियो है। मैं इसी यूनिवर्सिटी का स्टूडेंट था 1990 से 94 तक और लकीली ये शायद वही पीरियड था जिसमें डॉक्टर अताउर रहमान साहब यहाँ पर तशरीफ लाए और उन्होंने डॉक्टर सलीमुज़्ज़मान से ये टेक ओवर किया। तो मेरे लिए जो यहाँ पे आने का सबसे बड़ा टेक अवे है वो 2 चीजें। मेरी बड़ी ख्वाहिश थी कि मैं सर से हाथ मिलाऊं, तो आज मैं इनके साथ बैठा हुआ हूँ, ये मेरे लिए बहुत बड़ी एजाज की बात है। और वजह बताता हूँ कि मेरी दिल में क्यों कदर इजाफा हुआ कि जब मेरी यूनिवर्सिटी में पोजीशंस थी और यहाँ पे फिजिक्स में मेरी फैकल्टी में पोजीशन थी, जब मैं SNT स्कॉलरशिप क्लेम करने गया तो मुझे मालूम हुआ कि मेरी जगह कोई जा चुका है। दैट वाज द स्टेट ऑफ मैटर एट दैट टाइम। सो आई फेल्ट होपलेस कि मैं करूं क्या। तो जब सर आए और मैं एटॉमिक एनर्जी ज्वाइन कर चुका था आफ्टर डूइंग माय मास्टर्स फ्रॉम किनपो, तो जब 2001 में मेरा कॉमनवेल्थ स्कॉलरशिप हो गया और वो भी मुझे ड्यू टू सम रीजन मुझे वीजा नहीं मिला, तो जब सर ने ये HEC स्टार्ट किया और हमने देखा कि पहली दफा मेरिट पे लोगों को ये चीजें मिलना शुरू हुई और हम जैसे मीडियोकर्स भी कराची से निकल के और अपने मुल्क के लिए आगे गए और डिफेंस के शोबों में हमने PhD किया। तो सर सलूट टू यू सर, आपने जिस कल्चर को डेवलप किया है वो आज तक चल रहा है और पाकिस्तान को पहुंच गया है। तो PhD में तकरीबन 50,000 मैन पावर है और 50,000 मैन पावर हमारी एटॉमिक एनर्जी की जीन में अल्लाह ताला ने कुछ ऐसी चीज डाल दी है कि हम अब चाहे भी तो बेईमानी नहीं कर सकते। हम जिस तरह काम करते हैं हम कोशिश करते हैं कि अपनी पूरी कैपेसिटी पे काम करें। एक मेंबर साइंस के ज़ेर इंतजाम जो एक बहुत बड़ा काम हो रहा है, अभी एक पावर 3 रिएक्टर भी बन रहा है तकरीबन 7 to 8 बिलियन रुपीस की मदद से। और ये रिएक्टर हमें मजीद आइसोटोप्स की फरहमी करेगा। हम कैंसर में नई नई टेक्निक्स लेके आ रहे हैं। और एक बहुत बड़ी बात है कि हमारे पिस्टेक के इदारे ने, जो भी मेंबर साइंस के साथ ही काम करता है, उसने लीनियर एक्सीलरेटर भी बना दिया है। ये बहुत बड़ी अचीवमेंट है हमारी कि हम कुछ साल बाद इंशाल्लाह पाकिस्तान को खुद मेडिकल लीनियर एक्सीलरेटर देंगे, क्योंकि एक लीनियर एक्सीलरेटर की कॉस्ट इस वक्त तकरीबन 600 से लेके 1000 मिलियन रुपीस है। तो हम अपने मुल्क में इन्वेस्टमेंट लाना चाह रहे हैं ताकि हम ये काम भी इस मुल्क में कर सकें। ये जो सिंपोजियम है, ये बहुत बड़ा बहुत बड़ी अचीवमेंट है। जब मैं अक्सर वो मोहम्मद अली शेखी का पुराना गाना सुनता था कि मेरी आंखों से इस दुनिया को देखो, उसमें एक बीच में शेर आता है कि तेज झोंके झूमते पत्तों के पहलू गुदगुदाते हैं, तो पत्ते हंसते हंसते लौट जाते हैं। मैं सोचता था ये कैसे लौटते होंगे। अब जब मैं इन लोगों में बैठा हूँ, रजिया गुंडों में आ गई, तो मुझे अंदाजा हुआ कि ये वाकई में ऐसे ही होता है। क्योंकि जब मेरे लोगों ने बताया कि सर ये प्लांट जिद भी करता है, ये तकलीफ का इजहार भी करता है, ये जब उसको आप मुख्तलिफ माहौल फराम करें तो वो उस अपनी बढ़ोतरी शो करता है। तो मुझे अंदाजा हुआ कि वाकई में ये जो शेर लोग कहते थे, इनका ताल्लुक हकीकत से है। तो मुझे बड़ी खुशी होती है जब मैं क्योंकि मेरी फील्ड है शॉक डायनेमिक्स की। तो लेकिन जब मैं आप लोगों में बैठा हूँ, अब मुझे अंदाजा हुआ कि कैंसर रिसर्च और ये फूड रिसर्च, हेल्थ रिसर्च, एग्रीकल्चर, ये हमारे मुल्क की बका की जिम्मेदारी है। और iccbs ने एक बहुत बड़ी जिम्मेदारी संभाली है, और यकीनन डॉक्टर शाहिद मंसूर इसके लिए मुबारकबाद के मुस्तहिक हैं हमारे। बिल्कुल। और डॉक्टर शाहिद असल पोजीशन तब्दील करते रहते हैं, गोल पोस्ट, तो मुझे याद नहीं रहा कि वो उधर बैठे रहते। तो डॉक्टर शाहिद का एक बड़ी बात है, क्योंकि ये एक्स 10 years, आई थिंक 11 years ही वाज डायरेक्टर निपजी। तो ये अच्छा हमारे दरमियान एक ब्रिज बने हुए। और कोशिश करेंगे कि जो जो ये काम कर रहे हैं उनको हम MOU की फॉर्म में तब्दील करें। रात को मेरी मुलाकात सुबह से डॉक्टर फारूक से भी हुई थी, तो ये अभी बता रहे थे कि ये इस तरह के काम नहीं करने देते। तो वो जो जालिम समाज का कॉन्सेप्ट था वो मेरे जहन में आ गया कि इसको कहते हैं जालिम समाज। डॉक्टर फारूक का जो काम है वो है जालिम समाज का। तो बहरहाल ये कि ये सब काम हम आपको सपोर्ट करेंगे, हम आपके साथ आएंगे। और ये जो चारों इदारे हैं, ये मैं अभी चेयरमैन साहब से, सुबह मेरी चेयरमैन एटॉमिक एनर्जी से बात हो रही थी, मैंने ये सब बातें जो हमने नाश्ते पे की हैं या खाने पे की हैं वो मैंने उनको बताई हैं। एंड ही इज वेरी होपफुल, और ये चाहते हैं कि हम दोनों इदारे शेक हैंड करके आगे चलें और विन विन सिचुएशन में इस मुल्क को आगे लेके चलें। बहरहाल आखिरी में ये कि हमारे जो मुख्तलिफ प्रोडक्ट्स हैं, नैम फॉस्फेट है, ये वगैरह, ये सब चीज़ें हम चाहते हैं कि क्योंकि आपका इदारा कराची से निया करीब है, तो हम चाहते हैं कि बलूचिस्तान को भी आप कवर करें और आप हमारे सिंध को भी देखें। तो निया के साथ हमारा बहुत स्ट्रॉन्ग कोलैबोरेशन हो सकता है, क्योंकि निया आपसे करीब भी है और हमसे इस्लामाबाद से दूर है। तो हम कोशिश करेंगे कि इस इलाके को पहले टारगेट करें। और अगर अब हम कोई MOU करते हैं तो should not be memorandum of understanding. It should be milestone that we can track, outcome we must deliver, and the upshot, the final output that matters. Thank you.
Now I request Dr. Raza Shah, Dr. Dr. Mazhar, Dr. Mansoor to present the token of appreciation to him, to our chief guest. Sir, kindly come on the stage, please. I request you all to kindly give the token of appreciation to our chief guest. Thank you so much, Dr. Shakila Pasruki, for your inspiring address. Kindly come to the stage for the group photo.
Screen off.
I request to all the distinguished speakers to kindly come to the stage for a group photo, please. Assalamu alaikum and welcome back, everyone. Now, I would like to introduce our next speaker, Dr. Jawad Awan from Canada. He is a PhD graduate in biotechnology with a perfect CGPA and current postdoctoral fellow at the University of Alberta. They are a leading expert in functional genomics and genomic editing with an impressive research background in CRISPR-Cas system from top agricultural institutes. Please join me to welcome him. He will be joining us today via online. Sir, I request you to kindly deliver the topic.
Assalamu alaikum. Good afternoon, everyone. I will be presenting on genome editing based hybrid breeding with a particular focus on cotton. The main idea of this presentation is not that genome editing alone can create better hybrids. That would be obviously be an oversimplification. The real argument is that genome editing can remove some of the major bottlenecks that make hybrid breeding difficult, especially in crops like cotton. Hybrid breeding depends on three key things. First, reliable production of hybrid seeds. Second, rapid development of strong parental lines. And third is maintaining hybrid vigor in a practical and affordable way. In the following slides, we will see that how genome editing can contribute to all three through male sterility, haploid induction, double haploid development, and potentially synthetic apomixis. Sorry. Cotton is a high value crop because even small genetic gain can create a large impact at production scale. And globally, cotton production is made in millions of bales. So a few percentage points of yield improvement can translate into millions of additional bales. However, the real issue is not only demand. The major breeding constraint is whether we can create, maintain, and multiply elite hybrid seeds cheaply and reliably. For Pakistan, this point is even more critical because Pakistan's cotton production has been unstable over the years. According to the slides, as you can see, production was around 7 million bales in year 2023, 2024, and in year 2024 and 2026, it was nearly 5 million bales, and it is estimated around 5 million bales in the year 2026. At the same time, Pakistan's 27 target is much higher, around 9.64 million bales. So this gap shows that why conventional breeding alone may not be enough. Pakistan needs faster, more precise, and more scalable breeding approaches. But we should be very clear that genome editing is not a magic solution. For cotton, the credible route is genome editing plus genomics data plus practical seed production systems. Traditionally, hybrid seed production depends on male sterility systems such as cytoplasmic male sterility or CMS and environment sensitive genic male sterility or EGMS. In case of CMS system, Sterility is often controlled by mitochondrial factors, and they are mostly cytotoxic proteins, and they basically disrupt the floral organ development. But this makes the system uh, much complicated because it has to rely on maintainer as well as restorer lines. And the management of these system is additionally very much critical. And seed purity can become a serious problem on the later stages. In case of two-line systems, sorry, that we have environmental sensitive genic male sterile lines. They normally depend on environmental factors such as thermosensitivity and photoperiod sensitivity. Although this system is a bit flexible because restoration can occur through wild type germplasm, but they are vulnerable to environmental instability. If the temperature or photoperiod conditions are not ideal, sterility may become unreliable as well. This is where genome editing becomes useful. Instead of depending only on natural available sterility resources, we can directly target genes involved in pollen or anther development. But there is one important caution. Cotton should not blindly copy targets from rice or maize. Cotton needs proper ortholog identification, expression analysis, and phenotype validation. There are four main genome editing routes into hybrid breeding. The first one is male sterility. By knocking out pollen or anther development genes, we can create either female parents. We can obviously create female parents that do not produce viable pollens. And this can simplify hybrid seed production. The second route is haploid induction and double haploid production, editing genes such as SNS3, MTL, not like that gene, and DMP can help generate haploid plants. These haploids can then be doubled to produce completely homozygous parental lines, much faster as compared to conventional selfing. The third route is hybrid fixation. This uses synthetic apomixis, where meiosis is replaced by mitosis-like division, and embryos are produced without normal fertilization. In theory, this could preserve F1 heterozygosity across generations. And the fourth route is diversity and obviously delivery of the system. This includes reliance on wild diversity, pan-genome morphogenic regulators to enhance tissue culture efficiency and improve delivery systems. For cotton, the strongest near-term route is maize sterility plus genomic target mining. And haploid induction is promising, but still needs a robust cotton system. However, synthetic apomixis is exciting, but it is not yet ready as a production technology in cotton because this system has only been observed in model plants and some monocots like rice. So genome editing, because the following slides will be discussing the genome editing. So let's talk about genome editing. Genome editing is basically alteration of the genetic code of any organism through targeted modifications. So these model changes can include substitution of nucleotides to create specific mutations, deletion of nucleotides to knock out a gene, or addition of DNA or any DNA fragment to knock in a useful fragment or a useful protein. The most common application in breeding is gene knockout, especially when we want to disrupt genes that negatively affect a trait. For hybrid breeding, there is especially this is very relevant for male sterility because knocking out a pollen development gene can create a female parent for hybrid seed production. And genome editing or genome modification can occur at different biological levels, including DNA, RNA, and epigenetic regulation. But in most crop breeding applications, DNA level CRISPR editing remains the most widely used and most practical approach. The first practical route is editing male fertility. The breeding logic is very simple here. First, we chose an elite cotton line. Then we knock out selected pollens or anther-related genes, development-related genes. This creates a male sterile female parent. That female parent can then be crossed with an elite pollinator or to produce a hybrid seed. This strategy reduces dependence on complicated traditional male sterility systems and can speed up development of hybrid parents. However, the edited lines must meet two strict conditions. The first one, it must be truly male sterile. Second, it must retain female fertility, and if female fertility is damaged, the line is useless for hybrid seed production. This is why validation is essential. And for that, a gene edit that produces sterility in lab is not enough. The edited female parent must produce good seeds, good seed setting for when pollinated, maintain agronomic performance, and fit into a practical seed multiplication system. A study identified around 23 candidate genes in upland cotton that may be useful for developing male sterile germplasms. These genes fall into several categories, including transcriptional regulation, RNA splicing and processing, fatty acid transport and processing, sugar transport and processing, and other conserved pollen genes. So this makes biological sense because pollen development requires coordinated gene expression. Obviously, RNA processing, lipid metabolism for pollen ball formation, and carbohydrates to supply energy to the pollens. But here we should not overstate this because these are just the candidate genes. They are not automatically validated male sterility genes in cotton. So the validation pipeline should be, we should first identify the candidate orthologs, then design the multiplex CRISPR targets. So we will be targeting multiple genes at once, especially because cotton is a polyploid. So then we have to test pollen viability and female fertility, and lastly, we will design maintainer or restorer systems if needed. The second route is haploid induction. Hybrid breeding needs strong and stable parental lines. Traditionally, developing homozygous parents take several generations of selfing. Haploid induction can shorten this process. The basic pipeline is shown at the top of the slide. We start with elite cotton germplasm and use an edited haploid induction line targeting genes such as SNS3, MTL, or DMP. This produces haploid seedlings. Then chromosomes are doubled, producing double haploid parents, and that can be used in hybrid process. The advantage is speed here. So double haploids are fully homozygous and can greatly shorten the breeding cycle. But the honest limitation is that cotton does not yet have a routine and a highly efficient haploid induction system comparable to maize or rice. For cotton, this should be seen as a medium-term opportunity, not as an immediate production ready tool. So if I go into a bit detail here, so in panel A, you can see a simple hybrid development, a simple development of a zygote after crossing two gametes. In section B, you can see the elimination of female chromosomes. As you can see, that if you target the SNH3, the proteins, so those proteins will obviously disrupt the binding of kinetochore to the centromere. So because of that, the loss of that binding, those chromosomes will not be available for cell division. And in section C and in section uh, in section D, you can see the elimination of parental chromosomes, and in section C and in section B, you can see the elimination of either of the parent. And again, this is a summary of the cross. You can in the section in the panel E, you can see the elimination of either parent chromosome, and you can cross it with any line any elite line, and you can develop a haploid progeny, and after doubling its chromosome, you can have a double haploid parental line that you can use as for hybridization in a follow-up breeding cycles. So synthetic apomixis is one of the most ambitious idea in modern breeding. So normally F1 hybrids show strong heterosis, but this advantage is lost in later generation because of segregation. Farmers usually cannot replant F2 seeds and expect the same hybrid performance. So synthetic apomixis tries to solve this problem by allowing clonal seed production. In this approach, genes such as REC8, PAIR1, or OST1, basically related to recombination and chromosomal recombination and chromosomal binding during meiosis, are edited to convert meiosis into mitosis-like division. So this produces unreduced diploid gametes. Then parthenogenesis is induced, for example, through the ectopic expression of baby boom gene. So basically, when the zygote is developed, the baby boom gene is basically triggered or delivered by the male component. But in this case, we replace the promoter of the mother baby boom seed, baby boom gene, with the promoter of the male, and then it triggers the expression of baby boom. And this expression, ectopic expression, basically triggered the haploid induction. So it will allow embryo formation without normal fertilization. And in theory, this could allow farmers to replant hybrid seed without losing heterosis. In an other scenario, you basically get a hybrid, and you expose that hybrid to editing or disruption of SNH3 or MTL gene, and these genes will eliminate all the chromosome of one parent, and you will have the chromosome of just one parent, single parent, like it can be the chromosomes of mother. And in this case, you will have a plant just like the mother, and you can also follow the strategy to develop a clone of a father. However, for cotton, this remains a long-term research goal. It has been demonstrated mainly in model plant, as I have mentioned earlier. It has been demonstrated in rice, preliminary, but people are trying to duplicate the system in deployed such as tomato as well. So these genome editing examples establish that the targeted mutagenesis in cotton is moving forward, and cotton editing is no longer just a proof of concept. It is moving toward larger scale, broader genotype coverage and breeding relevance. Random gene lists are not enough. So next generation breeding needs data sets that connect genetic variation with production traits. Cotton pangenome and variome studies can be valuable because they identify loci, presence, absence, variation, and variable allele linked to fiber quality and yield. And the modern cultivar assemblies can help breeders understand structural variation and fiber development genes in current production germplasms, and structural variation maps and resequencing of more than 1,000 accessions can help define superior haplotypes and potential heterotic pools. Finally, male sterility candidate genes provide a direct route for building edited female parents. So far, cotton improvement should be the production of cotton hasn't been very much relying on the pangenomics, but in future, cotton improvement should be data guided. The best CRISPR targets will come from pangenome, structural variations, GWAS, and transcriptomics and field performance integrations. So here's a full breeding stock stock of for cotton. At the first level, we have genomic sources, and at the second level, we will predict genomic variations that and and if we want those variations based on those predictions, if we want them in our breeding program or not. And in the third, we can make edits in the genes that we want to be disrupted, and then edited male sterile female parents can be developed in parallel. Haploid induction or double haploid approaches could help accelerate parent fixation. And finally, everything must go through hybrid field testing. So there has been some couple of bottlenecks, and these are some. There has been some serious scientific bottlenecks, such as cotton has an allo tetraploid genome, so homolog redundancy can make single gene edits weak, and regeneration is still genotype dependent and slow. So synthetic apomixis is not ready for cotton. And there are also serious breeding bottlenecks. Sterile female parent must retain female fertility and agronomy value, and a hybrid seed production must be cheaper or better than conventional system, and heterosis must be stable across all the environments. So here has been some biotechnology advances that can improve cotton. For example, you can increase the tissue culture efficiency by employing some or co-transforming some regeneration enhancing proteins or peptides or some morphogenic regulators that that will have the tendency to increase the genome editing. And in another scenario, we can also rely on viral vectors, and we can obviously harness the potential of grafting to deliver the genome editing components. So to conclude, genome editing can shorten hybrid breeding cycle and help build better cotton hybrids. And thank you. I would be happy to take any questions. And you can also send the queries at this email address.
Thank you so much, Dr. Aman, for your valuable presentation.
So one of the questions which is asked by the farming community or textile is that how to improve the quality of cotton. So I think that one of the possible that Dr. Earlier also discussed that you can make hybrid, but the cost of hybrid production is very high. So what he has discussed, I'm very proud that he is my PhD student. He was at PhD with me at Leipzig. So the basic idea is that we can move to reduce the cost of hybrid seed production by using genome editing technologies. And so he has identified multiple genes. So if there are any quick short question, then otherwise we'll move to next presentation. So this is for the future of breeding in Pakistan, that we need to move to hybrids.
Okay, now we will move towards the next speaker, who is Dr. Arshad Jalal. And he is a postdoctoral fellow at King Abdullah University of Science and Technology in Saudi Arabia. He holds a PhD from Paulo State University in Brazil, where his groundbreaking work on crop biofortification earned multiple prestigious international awards. Currently, his research focuses on cutting-edge biostimulants and innovative fertilizer technologies designed specifically for desert agriculture. With over 80 peer-reviewed publications and global collaborations, he is a true leader in advanced agronomic science. I request you to kindly proceed your lecture, sir.
Bismillahirrahmanirrahim. So today I am very glad and happy, and it is really a great pleasure among a great scientist as a Pakistani. I just like from the morning. The very morning, I'm just listening very grateful presentations. And it is really pleasure for me to know that how advanced we are going in button technology, and still we are getting in more advancing the technology in button. My talk will be a bit different from that one. As from my introduction, you guys are aware that I'm an agronomist, and I am working in biotech lab at King Abdullah University of Science and Technology, Saudi Arabia, where my basic function is I'm working on translation of discovery into practical agriculture. So research, we are actually working a diverse group. I'm working in a diverse group in cows, and we are working on different aspects. So today I will try to focus on the phytohormones and carotenoid metabolism and how we transfer and translated this technology into practical agricultural application. Actually, we discovered some of the phytohormones, sulforaphane and particularly zephyrone. That is our lab discovery. And then later on, I would try to focus first on zephyrone. So zephyrone has been derived from the rice leaf and recognized as a plant hormone. And then it has been tested for different through gene expression and through gene expression, and now how we can know that without this is mutant, that's mutant, and that is wild type. And you can see when we remove this just from knockout this, so it is just a plant repeating dwarf and its growth is drastically affected. So how it works, we are spraying it on the leaves of the rice plants, specifically, particularly we test them these products in the rice because it has been derived from rice. And then later on, when we spray it on the leaf, it goes through into the root system and then pumping this sugar into the upper part of the plant, increasing photosynthesis and crop root architecture, root penetration, these things, and helping plants to grow better, to establish better, and increasing the yield of the crop. First, we were working on the products, the original product, the but then we synthesized these products in our lab because it was too expensive for the large scale application. And nowadays, it is our pleasure that we have the real product for these mimics or these Zubxenon analogs. And soon you guys will come to know that they are commercialized because now a good I would say like a good news that we now established a company at Coast, and Coast will work on these products commercialization. So now I will show you some practical results that are working with different crops, and you guys can see that when we apply these products in a very small amount, so how much it is increasing productivity compared to grain. In different crops, and we have like potato, we have 106% increase in tubers and maize and pepper, and saffron, which is considered as a golden bee for Saudi Arabia, and wheat under the desert condition, and millet, also one of the most important crops in Green Initiative project for 2030 of Saudi Arabia. So here are some practical applications. We have two examples as I showed in my earlier slide. So you can see here we have name, and then we have our two products, Mizas 3 and Mizas 5, similarly in millet. And you can see here in the statistical increase in the form of graphs. So I'm just showing yield here because it is one of the most important factor for farmers. Then we conducted a real field experiment with the farmer, and that was around 1 hectare for each product, and we used around 3 grams of this product. So the beauty of the product is that we are using very less amount, and it is giving us really fantastic results in terms of increasing productivity. So here are the results, and you guys can see that marketable tubers are increasing by 106%, and with another product by 66%. Most of the published data, if we can see, so the people are keeping marketable weight around 120 to 130, but I kept it around a bit high, and to see that how much increase we are getting in this way. And then the product is also amazing in the drought condition, which is in the coming slides I will show some with the cotton as well. And salinity, which was also one of the discussion today in the morning talk. So in the green pepper, capsicum, so we are just getting around 50% increase when we are using this product at very minute amount of 5 micromolar. And here is like working with different technologies. Then we decided that why not we should just apply it once with the seed priming and what we are getting. So these experiments are still in the initial stage, but I was just thinking to share with you guys. And initially we tested in the lab condition, and still we are getting like with the statistics and figures you can see here that we are getting increased by using only once with the seed prime. So when we are thinking that it might be effective against the drought stress. Then again, we use it in combination with fertilizers, which is also one of the big issue. Like is in the recent year, we can see like we were having conflict, Russia and Ukraine and these things. So there was a problem of fertilizers. So we tested these products, and when we use half of the fertilizers, so we are getting much increase in the yield and crop improvement. And here is an example of rice, which has been tested for 2 years in the field condition in Taiwan, and we are getting much increase in the grain weight and the other parameters. This is another experiment in the desert condition of Saudi Arabia, and it is with wheat and still with the 50%, if you can see here, like the grain here, so we are getting much increase as compared to others. Then as everything is getting advanced, so we were thinking that, okay, let's work on the encapsulation of this product, use minimum amount and fertilizers together in one product. So we encapsulated our product in the MOF metal organic framework, and we tested it in the field condition, and we were surprised when it gave us like twofold increase in the twofold increase in zinc content. And again, then we were thinking what it might be like. We should test also in cereal crops, which is one of the most important staple crops for most of the developing countries. Then we test again in the wheat, and it is like a multiple year experiment. And all of the year, 3 years, I conducted this experiment, and for 3 of the seasons, we are getting almost the same increase in the yield and also the zinc content and iron content. Without indication like extra indication, we were having this smoke. Then on a visit of Dr. Ataur Rahman and Dr. Shahid Mansoor to KAUST, so we luckily got this fruitful collaboration, and we were thinking why not to work on, Dr. Shahid Mansoor just suggested an idea to work on cotton crop, which is one of the famous and cake crop of Pakistan. And we are testing, you guys can see here, the these biostimulant has been tested in different locations of our different provinces, and specifically Punjab, Sindh, and Balochistan. And this was one of the initial experiments from where we got started. And we conducted this experiment in the greenhouse and then in the field condition in Balochistan. So we are having this data from Thorbus, going through and other, while this photo presentation is from the other variety. So you guys can see here the product is helping increasing the yield per plant in different regions of Pakistan. Again in Punjab, so we have like this 2 varieties who are there in Punjab, and we are still getting like increase specifically with Vizas 3, which is one of the product, we are getting much increase than the other one. So bringing together all these things, the technology bring a lot of like we are upscaling this now with UPL, United Phosphorus Limited India, and we are also talking in discussion with Dr. Shahid Mansoor. We are thinking to bring the product to Pakistan as the farmers, according to Dr. Shahid Mansoor, the farmers are happy using it in the cotton. So now coming to the second part, strike-electrons. So strike-electrons, we are specifically combating with one of the dangerous parasitic weed, striga. It is one of the biggest problem in specifically in African countries, and we have different analogs, but among different ones we have these two, MP3 and Nemagen. These are specifically working against striga. So these striga electrons have different functions. They are increasing root. They are getting the shootling dark, increasing the leaf area. How our technology works and how this trigger life cycle is working on and how it comes into life. So we can see this most of the time we are saying that this beautiful flower is one of the most threatened to agriculture in African countries. And even though like I have been visited Kenya, Ethiopia, the lands are getting barren and the farmers are getting nothing out when this trial just get emerged. So one of the plans is producing around 200 million of seeds, and they are microscopic seeds. So it is very difficult, and they can be dormant for 50 years. So when they get like a suitable environment, they are getting out, and they are attaching to host, and you can see it is causing around 7 billion losses in African and around the world to the crops, different crops like millet, maize, and the rice. So these are seeds of the striker, and you can see like it has a size of 1.5 millimeter. So very thin seed, and this thing you can see here as I quoted earlier. So and this is now they are affecting here in this region millet. So when they are growing up, the millet is not producing any chemicals and nothing. We are not getting any product. Then we decided, okay, let's move on, and we designed a strategy for it, and we call it suicidal germination. When it is normal growth, so the striker seeds are already available in the soil. They are changing into the roots of the plant, and then they are coming out like this. After attachment, they are growing into a plant. So what we are doing with our societal technology, we are spraying our products under the host plant, and they are smelling that there might be a host plant. These trigers are smelling that there might be a host plant, and when they are coming out, so they are not getting any host plant, they are dying, they are killing. So this is somehow like a deceiving technology that we are working on, and it is really successful. It's working in African countries, and we are working on it and like in different African countries. So these are our technology that I talk about. So we have like clay where we didn't apply, and the other treatments, MP3, as I told you, we have different other products. So MP16 and never. Then later on, we came to know that it has also like a growth establishing power as well. So it is creating some communication with rhizosphere and helping plants in the better root architecture and stimulating yield, increasing photosynthesis, substraction, and helping in the stress resilience. So then we tested it in different, like started from the greenhouse after different under different irrigation regimes. So we have 175% reduced, and 25 and 50%. And this is under the real field condition. Just okay. And then this is one of the most important. These are some of the most important plants that we are working in Saudi Arabia to convert desert into a green valley. So for this, we are working with the wild plants, and it's really helping us who are there by using our product. And I'm really thankful to our team, Professor Salim Babiri, Dr. Shahid Mansour, and his team. Unfortunately, I don't have a very good photo of his team, but I know some of the guys like Waleed, Salman, Hamza. They are working hard with us. So, and looking forward for this collaboration too. Thank you.
Thank you so much, sir.
Thank you so much, sir, for your valuable presentation. Ladies and gentlemen, now I would like to introduce our next speaker, Dr. Atiya Gohar, She holds a PhD in chemical from iccbs and currently working in Genome Center as a research associate. She has an experience as a deputy manager at QC department in Dow. She is an expert in vaccine and antivenom production and has extensive certifications in international safety practices and ISO standards.
Bismillahirrahmanirrahim. Assalamu alaikum, everyone. Assalamu alaikum, everyone. First of all, thank you so much, sir, giving me the chance to present the genomic part of this project. So, let me begin with the highlighting why medicinal plants are so important for the biopesticide development. For centuries, these plants have been used as a traditional medicine, but their value extends far beyond human health. Medicinal plants are the nature chemical factory producing the vast variety of the secondary metabolites and alkaloids, flavonoids, terpenoids, and glycosides. And they also exhibit the potent insecticidal, antifungal, and antibacterial properties. Why these biopesticides, plant-based biopesticides, is so important? Because synthetic pesticides have caused a cascade of problems such as environmental contamination, pest resistance, and health risks. Plant-based biopesticides offer the eco-friendly alternative, and these are the biodegradable, target-specific, and locally sourced, which make them cost-effective. By integrating these natural compounds into pest management, we promote environmental safety and biodiversity conservation. Why we need this change? First, we have extensive environmental contamination affecting the soil and water pollution, and also bioaccumulation in the food chains. Second is the more than 500 pest species have developed resistance, requiring even higher application rate. Third is these chemicals cause health risks to human and also against the beneficial insects, including the pollinators. Finally, economic burden on farmers, especially in developing countries, is unsustainable. So that's why plant-based biopesticides is the solution of sustainable, biodegradable, target-specific, eco-friendly biopesticides. So how genomics can unlock the insecticidal potential of medicinal plants? Genomics allow us to do more than just identify compounds. It is also understand entire manufacturing processes within the plant. The whole genome sequencing and metabolomics will reveal the genes cluster responsible for biosynthesis of secondary metabolites, which help us to identify the metabolic pathways, and functional annotation guide us to selection of candidate gene for biopesticide formulation, ensuring we focus on most promising compounds. In our study, we focus on 3 medicinal plants, well-documented insecticidal activities, in which first is the Citrullus colocynthis, which is the rich source of alkaloids and flavonoids. And these alkaloids and flavonoids, they cause the membrane disruptions in insects. Second is the Calotropis procera, usually known for latex enzyme against the cotton pest. And third is Rhiza stricta, which is rich source of alkaloid that can induce autophagy-driven apoptosis in insect cells. These 3 plants are native to arid region across Africa, Middle East, and South Asia. They have evolved robust defense compounds. So plant-based pest control solution, we have decided to perform the genomics, metabolomics, and nanoformulation of our biopesticides, and then implement into the field against the insects. This is the genomic workflow for these three plants, in which we, first of all, we have collected the sample, then extract the high quality genomic DNA. This is the first and most critical step because which is the source of all downstream processes success. After that, we have performed the library preparation, in which we use the genomic DNA and perform the fragmentation and adapter ligation to prepare the pair and library. And then followed the Illumina sequencing. After the sequencing, we generated the massive amount of raw data, which further process and remove the low quality reads and assemble or join the high quality reads to make the genome assembly. After the genome assembly, we further analyze our data by the functional annotation and comparative genomics. This is statistics of our raw data of pair and library of Citrullus calocynthis and Calotropis procera, in which after the sequencing, we have found total reads 163 million. And in Calotropis procera, we have found the total reads 145 million. After the de novo genome assembly completion of Citrullus calocynthis, we have found the total assembled genome of our Citrullus calocynthis, which has a size of 366 million base pairs. And it consists of total scaffold 85 and 31 million. Largest scaffold in our genome assembly is 36 million, smallest scaffold 12,539 base pairs. After the de novo genome assembly, we have performed the functional annotation. In this figure, please can you help me to here you can see the after the functional annotation, we have found the exome, CDS, 5' UTR, 3' UTR, mRNA, and gene, and these are the counts of our features of functional annotation. In this figure, you have shown that number of genes distributed in all our 11 chromosomes. The richest amount of number of genes in chromosome number 3. The highest amount of the genes present in 11 chromosomes shows that our genome assembly has a good quality and good in gene interpretation. In third figure, you can show that gene ontology category, in which you have seen that the biological function with the highest number of annotation, 8,184 genes, which means our genome majorly consists of the genes which perform the biological function, which include the metabolism, signaling, and development. Whereas cellular function with the 2,965 annotation gene found that confirmed that the presence of protein in organelles and membrane. Whereas molecular function with the 1,846 annotation reflects that binding activities and enzymatic activities of these cellular functions proteins. This figure shows the number of genes distributed in cluster of ortholog group. Here you can see that the transcriptional highest gene present in transcriptions, signal transduction, post-translation modification, and carbohydrate metabolism. Here is the categories we found in our genome assembly with gene count. Transcription has the gene count, highest number of gene count, which shows their biological function, and it shows a very important biological function in the regulation of gene expression, stress response, and development, whereas signal transduction mainly perform the biological function of stress signaling, immune responses, protein folding, and there are a lot of function in these functional categories. These are the pathway analysis in which K pathway consists of a number of highest gene in these identifier. These identifier when we check by the databases. So we found that metabolic pathways in citrulus colosynthesis genome assembly shows the 100% of gene associated with function of the core metabolism in citrulus colosynthesis. And then secondary metabolite biosynthesis, microbial metabolism, biosynthesis of antibiotics, these all are present and perform the biological function in antimicrobial defense system, defense compound, and intermediary metabolism and plant defense system. After the PFAM analysis to find the protein domain in our genome assembly, we have found that P kinase and P kinase tyrosine shows the highest number of genes. These both combinedly perform the protein phosphorylation function, whereas PPR and PPR2 perform the RNA binding and processing, and LRR family perform the protein-protein interaction and pathogen identification or recognition. These are the domain, protein domain, which we have found in our genome assembly, and their associated genes. After that, we perform the metabolic pathways and reactome pathways to find the metabolic gene and signaling and development pathways to find out how much genes are included in these pathways. Here you can see that in first row is the pathway ID, which confirmed by the databases. Here you can see that the pathway phenyl propanoid biosynthesis, flavonoid biosynthesis, polyunsaturated fatty acid biosynthesis, and these all are the pathways such as plant-specific metabolism shows a biological function in which include the defense mechanism, UV protection, lipid metabolism, cell wall, and stress hormones. These are all the biological function which are present in our citrulus colosynthesis genome with the richest amount of the count of proteins and domains. This is also another reactome pathway which include the ubiquitin dependent degradation, autophagy, neutrophil degranulation, protein folding, which perform the function of apoptosis, stress response, immune defense, and protein quality and apoptosis signaling. This is the gene ontology term. Here you can see that associated genes and protein include in our genome photosystem 2 proteins, light harvesting complex, and PPR proteins, transmembrane transporters, aquaporins. These also play an important role in the photosynthesis, light harvesting, metabolite transports, and water transport, drought tolerance, and stress response. These are stress response genes and functional proteins present in our genome. Most importantly, which we want to see in our whole genome sequencing of citrulus colosynthesis is insecticidal protein, which is present in 11,058 numbers and perform the multiple mechanisms such as gut epithelium disruption, membrane pore formation, digestive enzyme inhibition. This is the insecticidal protein which is found in highest number is lectin. lectin, which have the disruption of gut epithelial cell mechanism, and toxin, defensin, protease inhibitor. These all are we identified from our Citrullus colocynthis genome assembly. This is the mechanism, which mechanism prominently work in our Citrullus colocynthis genome is the gut epithelium disruption, which work against the insects, and other are the membrane pore formation, digestive enzyme inhibition, and exoskeleton. These all are the proteins which function against the insects mortality, and this is the cucurbitine, which also perform the main important role in insecticidal. This is have the insecticidal property, and for the biosynthesis of this cucurbitine, we mainly identified the number of genes and categories such as terpenes, so squalene, terpene cyclase, and cytochrome P450. These are enzymes which also present in high score confidence in our genome assembly. These all perform important role to formation of the cucurbitine E, which is start from acetyl-CoA2 by using these all enzymes. So sustainable agriculture application, plant-based pest control solution, eco-friendly pest control, integration with modern farming, and climate resilience. In conclusion, we have found in our genome assembly 21,540 genes, and in which 11,058 genes have the insecticidal function, whereas 6 distinct mechanisms we have identified. So in conclusion, we said that nature has already solved the pest control problem, and genomics is helping us to understand and harness this solution. I must pay the acknowledgement of the distinguished scientists and our team member. Without the hard work of these persons, we could not achieve or complete our research project. Thank you so much.
Thank you, Dr. Adya, for your valuable presentation. Ladies and gentlemen, now I'd like to introduce our next speaker, who is Professor Dr. Sayyid Ghulam Musharraf, Distinguished Professor at iccbs and founder of internationally recognized mass spectroscopy laboratory. He is a renowned scientist, recipient of Tekanga Imtiaz, and a fellow of several prestigious scientific societies. I would like to request him to come on the stage to deliver his presentation.
Assalamu alaikum. Good afternoon. First of all, thanks to Doc Saab, because Doc Saab, is so kind to involve me in his project. The idea, the basic concept of biopesticide is actually driven by him. And he is the one who actually integrated every one of us in his project. Me, Dr. Tina, Dr. Disha, and so many people. So he is actually the mastermind behind all these interesting projects. So, my expertise is very much relevant to the mass spectrometry. This technique, the mass spectrometric protocols, is not restricted with any particular class of compounds or particular class of materials. So it has a very broader aspect to explore. Starting from the human health, the cancer progression, the disease monitoring, herbal medicine, drug discovery, you can just name it. So this technique is indeed has a strong application in all biological and chemical sciences. What I'm going to talk about is about its application related to the plants, right? So the common terminology that we use is a metabolomics. So metabolomics, a kind of data set which actually tells you the current status of either human being or the plant as well. So that's what we are going to talk about in my next few slides. Okay, so I think no one can deny about the importance of plant material in our daily life, right? So, like for example, the most common one is the use of medicinal plant for our daily routine treatment. You know, we all are used to have a kind of decoctions, chai, ये सब चीजें आपके जो है आपके इस्तेमाल में daily basis में. So the plants are actually you know in our use. बैक कैसे हो गए तो बैक जा रहा है so उसको बैक कर so इसमें चलो किया ना Okay, so you know that the first important thing about this medicinal importance, right? The second one is nutritional benefits because nowadays the people are talking about the functional foods. The people are now would like to be treated with the help of foods, with the help of herbs. So this area is nowadays called in, and this part is actually in upcoming days. Hopefully that you know the medicines would be replaced with the functional foods. So this area is equally important to explore. Hydro formulation, I think we all know that the very strong, you know, the indigenous system available across the world, like Ayurvedic system, the Chinese herbal medicine. So they are using those formulations for the treatment of very acute and chronic diseases. So herbal formulations are there. Drug discovery, you know, several important biological molecules are actually derived from the plant source, right? Several anti-cancer agents, anti-malarial agents. So they are actually derived and isolated from the plant. So plants is one of the important source to isolate and identify the potential need for the different disease treatment. And the fifth part is actually very obvious. It's about the agriculture goods. So since morning we are listening about the importance of the enhancement of crops. You know that crop could be food one, that could be the cash crop or the non-food one. So these are the different areas where the plants are actually licensed application within human being. So now come to the metabolomics. As I have mentioned, these values are very important because remember Professor Ratta was talking about the top to down approach. It is actually starting from genes. But don't forget that ultimate outcome, the phenotypic compound is actually the small one. आप सब लोगों को पता है कि आपको अगर ग्लूकोज हो गई है डायबिटिक हो गई फिर छोटे सा मॉलिक्यूल उसकी कंसंट्रेशन चेक करेंगे दैट इज ग्लूकोज. So that is the importance of metabolite. That is actually metabolome, which reflects the current phenotypic condition of an individual or in plants as well. किसी plant में जहाँ grow करो तो कुछ और metabolite आता है, कहीं और grow करो तो कुछ और metabolite set आना शुरू हो जाता है. So it actually is linked with the overall condition of the atmosphere. So that's why the metabolomics part is important to study, right? So if you just look the protocol, especially related to the plant-based or the agriculture one, so you can see the first step, obviously the collection of sample. You have to have a very strong statistics and your collection ethics to collect the sample. The second one is sample extraction protocol. This is again a big challenge, especially for the chemist. I don't want to go into detail, but the chemist knows, that it's always a challenge in terms to cover the broader range of metabolites. The one set of metabolites deals the different extraction protocol, while the another one deals the different set of protocols followed by different techniques. So this is another important challenge. The third one is obviously to record a letter analytical protocol, right? So we all know the very two important techniques that is NMR and the mass, right? NMR actually gives you the 3D structural concept of a compound, while mass provides the 1D kind of identification. But mass, it is actually a highly sensitive technique. The technique is actually available addition with the LC chromatogram. You can straight away separate followed by analysis, right? So that's why the mass is very much in for such type of work. And the fourth is actually very important, and Professor Tawraman has mentioned lots of software which can be used for as an AI and mass spectral interpretation. We are using that tools as well. So the statistical analysis, the outcome, the extraction of the data from the complicated spectra is equally important. And here you require some kind of smart AI tools or maybe some statistical good software from which you can extract out the data. So this is the protocol which you can use for if you are interested to work on the metabolomic side. So what I'm going to tell you that the application of mass spec metabolomics in different aspects of herbal medicinal plants, right? The first one where you can see that we were interested to see the comparative metabolomics distribution of a same genera into various species, right? So this is one of the application where you can study the distribution of important metabolites across the different species of same genera. And you can see that we have investigated the salvia species. So we have investigated around 5 medicinally important salvia species in terms of their metabolome and find out their similarity and differences. And we identified their kind of similarity as far as the few metabolites concerned, but there were few metabolites which were exclusively specific for particular metabolite. So this is one of the application, right? So let's come to the another application of mass spectrometry metabolomics. This is the application where you can actually compare the Cucurbitaceae family member. Cucurbitaceae, आपको पता है that this family is famous as far as bitterness concerned. मशहूर है दरिया कड़वा और जो भी है उसी साथ at the same side you have the watermelon, the other part really sweet. So there is a need to study the metabolite distribution. And one of our studies actually we are also focusing on Cucurbitaceae one. So this is the work that we have done, and we have studied the 11 different species of Cucurbitaceae and find out very interesting metabolite distribution across different species. Another important, that there are two discuss jojoba and jujubas malaria. So this is another very important medicinal plant. The one is medicinally important, the other is not. But the problem is that they are very close in terms of their morphological resemblance. एक बहुत बड़ा मसला ये होता है कि जो आप रॉ मटेरियल ले जाते हैं तो वो बाज़ औक़ात धोखे में कुछ गलत भी कलेक्ट करके ले आते हैं, and because it is not their fault, because of the very close similarity of the medicinal plants. So in that case you require to study either the genetic barcode, right, through which you can differentiate, or the metabolomic pattern. So that this is the metabolomic pattern where you can study and differentiate the two different species. So that is already published. So this is one of the applications using the mass spectrometry metabolomics. This is a very interesting example. There are, as I have mentioned, that there are some plant species which are very close in terms of morphology. So this is one of the examples which actually we developed to differentiate the one of the cestrum species from the toxic species. So the toxic plant material is really similar with the another medicinal plant, and that was the issue actually highlighted by one of the Hakim to us. एक दफा वो हमारे पास आया और बताया कि भाई ये हमें बड़े मसले हैं इसके अंदर और इसी तरह का एक टॉक्सिक. So can you resolve these issues? So being a mass spec person, we developed a kind of metabolomic spectrum, and you can see that if you just look at their, the PC diagram. So based on the metabolomics, all three species were quite differentiated, right? And this is a kind of quantitative chromatogram through which you can differentiate the cross contamination of that species into the original medicinal one, right? So this is one of the applications of mass spectrometry based metabolomics. Okay, now come to the another one where we are talking about the targeted metabolomics. It's so obvious that sometimes you are interested to quantify some biological important compounds in the plant. But very obvious, it requires a strong work in terms of like database development and MRM fingerprinting development. So this is one of the examples where you can see that we have actually developed a kind of a protocol through which you can quantify and qualitatively analyze a large number of alkaloids in plant samples, because alkaloids, they are biologically active. So it's really important to see whether those biologically active compounds are present in the plants or not. So that is the work that we have developed, and we developed a kind of profile of 200 alkaloids. And this is how that we actually identify based on their different fragmentation patterns. This is the ESIMS pattern. These are the different collision energy that we have recorded and make a database. And now you can see that this is the database that we have developed, and this is the analyte that we have analyzed with the plant sample. So rightly matched exactly with our database. This is how that you can not only identify but quantify those biological molecules in the plant extract. So this is another application of. So these are the some plant material where we have identified those alkaloids that we have actually developed. Another important thing, the I don't know how many of you are familiar with the mass-based work with the plant. There is a very important, a big challenge to identify the compound. अताव रहमान साहब जो बात कर रहे थे सुबह की, ये बड़ा बुरा लगता है कि आपने 5 साल काम के मतलब सारे लोगों का पाउंड मिल गए और उसमें तकरीबन हमारे हिसाब के हिसाब से $50,000 खर्च होते हैं. If a person is going to work for 4 years and at the end of this, you identify a known data set. So please don't do this. The techniques are available. Just move towards the mass spec, but at the same time, there's a big challenge of their database development, right? So one of my activities, is to develop a database. So we are the one who are constantly putting and adding the ESIMS database with the LCMS. So the LCMS would be equally useful for the quick read application of compound. So this is the example. Another important example of mass spectrometry-based metabolomics is the network pharmacology. What does it mean? Which means that there are few plants that all those plants are equally good for your headache control, for your stomach, for your another pain, but they have a different set of metabolites, right? So how you can correlate those metabolites with the gene? So this is known as network pharmacology, where we are actually working. The lady just before me explained the biopesticide potential of 3 important medicinal plants. So that's what we are going to work out with the help of network pharmacology, that how we can link those different metabolites with the gene and then make a kind of synergy and understanding to make a pathway which can be, mimic to develop their commercial scale production. So this is the 4th plant that we have actually investigated with the help of network pharmacology, obviously through metabolomics. If you just see this diagram based on the metabolomics and PC, you can see that those plants somehow they are very much differentiated in terms of their metabolomics profile, right? But as far as the biological activity concerned, they are most likely act together. So this is the network pharmacological picture, which is really difficult to explain in a short span of time. But ultimately, as I have mentioned, that it links with the activity and the relevant gene, right? So this is another application of area where the mass spectrometry metabolomics can work together very effectively. Okay, so next 2-3 minutes. So now I'm just going to wrap up my presentation. So in one of the application, we work with the food sample as well. That there are several varieties which the people actually claim they are drought resistant, they are this resistant. But what about their molecular understanding? So this what we have done with the NIAM and the NUTRI. So they just give me a chickpea variety, and that variety actually they are observing as a drought resistant. So we identified with the help of metabolomics, there are so many osmoprotectant molecules formed in that variety, which actually protects the moisture. So these are the studies which actually again, addressing the application of mass spectrometry metabolomics. Okay, so these are the work that we have done. Another important application, just in 1 minute, I would like to tell you the standardization of a polyhydro formulation with the help of mass spectrometry metabolomics, right? So there are tools and the ways, because the big challenge of these products, commercialization of these products is about the standardization. These are the products of Albion. So We just work with the Helpy on the because these products are very valuable for their cough effect and all these things, but they want to send these to the other countries as well. The big challenge was standardization. So we developed the standardization protocol with the help of metabolomics as a whole story. But at the last, what I would like to show that this is the protocol that we have actually, sorry. So this is the protocol which we have developed with the help of mass spectrometry metabolomics for the standardization of polymer formation, right? So I hope that you have learned so many applications of metabolomics as far as the plant research concerned, right? At the last, maybe useful for you because based on my expertise on mass spectrometry, I think just 2 years before we interviewed with the federal minister, and they realized that the HJ could be considered for the establishment of a national reference laboratory for pesticides. So here we are. So I think 2 years before we signed an agreement with the federal minister regarding the first of its kind reference lab laboratory establishment. So that lab is established under the umbrella, and you can see that this lab is fully equipped with the high-tech Orbitrap mass pack, and we are actually heading this laboratory. This laboratory soon it is fully functional, and the notification has been done, but the federal notification is still in process. So soon it will be, functional. So you can see that different the media coverage. So that lab in 1 year of time is actually accredited through a PNAC as well. So we are very much accredited for the commercial sample analysis. So these are the team members, different team members who are working, and this is the quick view of the lab, working. And our lab is not only the running the testing show, but you can see this is the first of its kind paper of the spectrum development based on EI with the help of pesticides. So this is the if you just look you can find out the national reference laboratory. So our laboratory is also research involved in research as well. So I think with this I'm very thankful to all of you for your patience.
Thank you.
Thank you so much, sir, for your valuable presentation. Now, I would like to invite our next speaker, Dr. Shahid Mansoor. He will deliver the topic from seeds to biopesticides and biofertilizers. May I request, sir, to please come on stage to deliver his topic.
I think it's a very difficult task कि आपके और लंच के दरमियान में हूँ. I will try to be as brief as possible because my task has been made easy because Dr. Atia has given some of the work, and then Dr. Arshad Jalal has also given the presentation. So what I will do is that I will just give a synthesis that how we have led this route and what we have done in the last 3.5 years. So I think that the question was very obvious that industry wants to have good quality seed, non-GMO for organic agriculture and biopesticide. So what we need is we need all the input which are required for doing organic agriculture. So what are the requirements? So we need high yielding crop varieties, pure non-GMO seed because cotton is often cross-pollinated, so we get contamination. So it has to be pure. Then biofertilizer, biopesticide, and traceability. I think that this is one of the very important areas. This is going to be. So Dr. Mubarak is here. He understands that how important it is to have traceability there from farm to fashion. Now, this was the first humble success that we had. What we did is that, realizing the weather condition in Karachi, we thought that, we can have multiple generations of cotton in 1 year. And that was successful. And we used this technology, simple technology, to do something very interesting, is to find pure seed. And the other thing was that, to also have selections under the field condition, Kuzdar, and Uthal. We were doing selection there because those are the real cotton growing area. We identified some of the plant variants, and also nature inspired BT cotton. Now, this BT cotton is compatible with regenerative agriculture. What was very, very interesting was that we were getting very high mold rate, and Farooq is here, so we were actually trying to get some of you to Uthal tomorrow, but because of some reason that we postponed it till August. So what we got is that we got very good data, and now Farooq from Tyson and SM Industries, they are going multiplying the seed on around 6 to 8 acres. Now this is something which was then once I was at here, it was infectious to work with all people. So we thought that we can develop a biopesticide. I don't have to go into the detail because Dr. Atia has already discussed some of those activities. So the nutshell is that we use the facilities available with Professor Bina Siddiqui, and we use them to develop this extract. Now the only difference is that she was concentrating on neem. We concentrated on 3 other species that I just mentioned. Other thing that besides insecticidal activity, they also have nematicidal activity and antifungal activity. Now the real challenge is that when you come to the question of transfer of technology and how to commercialize the product. So we were very lucky to have some of the friends. So to be fair, and so what we did is that we got have expression of interest for the transfer of technology. For the idea was to do transfer of technology for large scale trials, technology transfer, and commercialization. And one thing more is that when we are at iccbs, which is a very competitive environment, you need to publish good journals and good journals. So I hope that we are going to touch some of the best journals because of the innovation that we have. That we are not only developing this biopesticide, but also having a backup of genomics and metabolomics. Dr. Atia just mentioned this. We also I will take 1 or 2 minutes to explain that we met synthetic chemists like Dr. Farooq has already discussed about this. So what we did is that as Dr. Arshad has already explained that we developed a collaboration because we need to have real-time solutions for real problems. So the real problem was that farmer need good yield and good quality at less cost. So we did all this experiment in multiple places. There are some areas where I will think twice before traveling to those areas, but by networking with the relevant people, we were able to do some trials in Panjgul, Turba, Uthal, Punjab, and Sindh was very easy. What we found was very interesting. And these are for use that we found a major increase in yield of cotton, and that was both in the quantity of cotton produced and the quality. This is one thing very interesting. I was not sure that whether Dr. Arshad will talk about will talk about this. This is the increase in grain. We are finding the increase in zinc and iron. This was amazing. It's a long-term dream to have iron and zinc, more iron and zinc in our grains. And by just applying biostimulant, what it does is that it helps to mobilize these ions into the plant, and we are getting. And one thing more that we identified was that varieties differ in their accumulation of, and this is something which is again a very good question for people like Dr. Atia to do the genomics and find out, what's going on, why some varieties respond more than the others. So these are some of the conclusion and the future direction collaboration with key players. Very important to have. So I'm very glad that a lot of industries are present now, and then in the future, afternoon as well. Dr. Farooq has just joined us. Welcome. And also expansion of cotton and soybean dye. So, I mean, we are focusing mostly on cotton, but also we need to have legumes in our system. Variety is, we need to change the whole paradigm of varietal development. We need varieties that are more responsive to bio input and technology-based traceability from farm to fashion, and apart paradigm-shift in breeding varieties. So we have to now not only study the genome of variety, but also the microbiome, which are the microbes associated with the root, and also their response to biofertilizers. So because Dr. Malik, pointed out that the different varieties release different levels of metabolites which attract microbes. So obviously, every microbe, every plant species, or every variety is different. So what we need to do is to combine the forces and then come up with a real solution for farming community. With this, thank you very much. Okay, time to be.
Thank you so much, sir, for your valuable presentation. Now I request ladies and gentlemen to kindly proceed towards the multi-purpose hall for the lunch break, and after that we will start the session again. Sorry? At 2:15 we will start the session again. Let's move forward. Our next speaker is Dr. Sir Saqib Suhail, and he will be presenting the topic Industry Perspective for Sustainable Textiles. And may I request Dr. Shahid Mansoor to provide a brief introduction about him.
It's a great honor to introduce Saqib Bhai. Saqib, we met in Egypt. We didn't know each other, but we came to know that he was representing a very big textile industry in Pakistan. They were actually sponsoring the World Cotton Congress. So I was very impressed. So, but he showed his generosity when I came to Karachi. So the first MOU that we signed for seed purity was funded by Artistic Millionaires, and that founded the whole. So a lot of credit goes to Saqib Bhai. Saqib has a lot of experience of working with sustainability. He has a very good insight, and he has his own company, and he's everywhere where there's a discussion on cotton. So Saqib, please.
अस्सलाम जी, ऑडिबल में सबके लिए चलो बहुत शुक्रिया। बहुत शुक्रिया डॉक्टर साहब आपको इनवाइट करने का, और मुझे आपने याद दिला दिया जब ऑलमोस्ट हाल में 4-5 साल पुरानी बात है जब हम मिले थे कायरो में। और मतलब छोटी सी स्टोरी ये कि वहां जब मैं पहुंचा तो मुझे पता चला कि पाकिस्तान में रिसर्च हो कितनी रही है और कौन कौन कर रहा है। तो पाकिस्तान में मेरी मुलाकात इन सबसे नहीं हुई थी, ये सब मुझे वहां मिले, और उसके बाद से माशाल्लाह जो हमारी पार्टनरशिप चल रही है बहुत अच्छी है। तो मेरा नाम साकिब सुहेल है। आई रिप्रेजेंट अपफ्रंट टेक्सटाइल्स। इट्स अ टेक्सटाइल एंड सप्लाई चेन कंसल्टिंग कंपनी, और सोर्सिंग ऑफिस भी है। और मेरा काफ़ी टाइम हो गया टेक्सटाइल के अंदर, मास्टर मेरी पढ़ाई भी टेक्सटाइल में ही है। आज डॉक्टर साहब ने मुझे बताया था टॉपिक बड़ा इंटरेस्टिंग सा है कि व्हाट्स हैपनिंग इन द इंडस्ट्री सस्टेनेबिलिटी के लिहाज से। और अगर ये बात अगर मुझसे कोई 10-15 साल पहले पूछता तो एक अलग जवाब होता, पिछले 5 साल पहले पूछता अलग जवाब होता। जब हम लोग खुद पढ़ रहे थे टेक्सटाइल, तो उस वक्त सस्टेनेबिलिटी का जो मफहूम होता था वो होता था कंप्लाइंस करा लें, ऑडिट करा लें, ISO 9001, बस ये होगी सस्टेनेबिलिटी। इसके अलावा किसी को कुछ पता नहीं होता था। इस तरह 5-10 साल पीछे आप चले जाएं तो सस्टेनेबिलिटी का जवाब क्या होगा कि थोड़ा आपने पानी बचा लिया, थोड़ा सा क्लाइमेट पे काम कर लिया। लेकिन अब जो सस्टेनेबिलिटी है, इट्स अ होल मॉडल, जिसको हम कहते हैं ESG, इन्वायरमेंट सोशल गवर्नेंस। इसको साइलो में यू कैन नॉट वर्क। अगर आप सिर्फ कहते हैं कि मैंने क्लाइमेट पे काम करना है और लोगों पे काम नहीं करना, अगर मैंने इन दोनों पे काम करना है लेकिन स्ट्रक्चर्ड काम नहीं करना, पॉलिसी बेस्ड काम नहीं करना, तो यू विल फेल डेफिनेटली। और ये होता आ रहा है और आज भी हो रहा है। टेक्सटाइल इंडस्ट्री के अंदर जो डिफरेंट फोर्सेस एक्ट कर रही होती हैं, उसमें 3 मेजर फैक्टर्स होते हैं जो हम लेते हैं। पहला फैक्टर होता है एक इंटरनल पुश, कि इंडस्ट्री है उसका अपना इंटरनल विजन क्या है, उसका अपना इंटरनल रोड मैप क्या है, उनका पाथवे क्या है, वो कहीं उनको इंटरेस्ट भी है इस काम में कि नहीं। सेकेंड जो ड्राइवर होता है, इट्स रिटेलर्स, कस्टमर। जो टेक्सटाइल इंडस्ट्री के कस्टमर है, दे नीड टू नो एंड दे कि आप किस तरह का अपना काम कर रहे हो पूरी सप्लाई चेन में। और थर्ड प्रेशर जो ड्राइवर्स को कहते हैं, दैट्स द रेगुलेटरी फोर्सेज हो गई, या आपके थर्ड पार्टीज हो गई। तो आपके ख्याल में इन तीनों में से सबसे ज्यादा प्रेशर किसका होता होगा? अपना इंटरनल पुश है, बायर का पुश है, या रेगुलेटरी पुश है? सबसे पहले कस्टमर का पुश होगा ना। जो कस्टमर कहेगा वो तो मैंने करना है। अगर कस्टमर नहीं कहेगा, क्यों एजाज भाई? अगर कस्टमर नहीं कहेगा तो हम तो नहीं करेंगे कभी भी, चाहे आज से भाई जितना मर्जी हमें कह देते, हमने नहीं मानना था। लेकिन जब कस्टमर आपको कहता है कि आपने ये काम करना है, तो हम उसको रिपोर्टें भी दे देते हैं, उसको डेटा भी दे देते हैं, सारा काम कर देते हैं। तो उस वक्त क्या होता है कि आप अपनी मर्जी से काम नहीं कर रहे होते। उसका नतीजा ये निकलता है कि एवरीथिंग दैट यू आर डूइंग वो बिल्कुल लास्ट मिनट पे हो रहा होता है, पहली चीज, और उसकी कॉस्ट बहुत ज्यादा है कंपेयर टू अगर आप अपनी मर्जी से वो काम कर रहे होते हैं। इसकी एग्जाम्पल है, अगर कॉटन रिसर्च की भी काफी बात हो रही है, बायो फर्टिलाइजर और बायो पेस्टिसाइड सबकी बात होती रही यार। अगर आज इंडस्ट्री या फार्मर डिसाइड कर ले कि मैंने खुद से अपने फार्म को सस्टेनेबल बनाना है, तो वो अपने टाइम से अहेड काम कर रहा होगा। तो उसकी सोच की वजह से उसको जो फर्स्ट मूवर एडवांटेज है वो ज्यादा मिलेगा। इसकी एग्जांपल ये है कि आज से 5 साल पहले जब ऑर्गेनिक का पाकिस्तान में काम शुरू हुआ, कॉटन का जो प्रीमियम मिल रहा था वो अब नहीं मिल रहा। तो जिसने 5 साल पहले स्टार्ट किया, जिस फार्मर ने, जिस इंडस्ट्री वाले ने, जो भी बीच में स्टेक होल्डर थे, उन्होंने उस टाइम से ना सिर्फ अच्छा नाम कमाया बल्कि अच्छा मुनाफा भी कमाया। आज जो ये बंदा एंटर करेगा इस फील्ड के अंदर, वो एज अ पाइनियर नहीं एंटर हो रहा। वो कैसे ऐड हो रहा है? क्योंकि अब उसकी कंप्लायंस है, उसकी ऑब्लिगेटरी रिक्वायरमेंट्स हैं कि मुझे अपने आप को सस्टेनेबल करना ही करना है। तो ये बहुत जरूरी चीज है कि आपके जब तक अपनी पुश ना हो, अपनी ड्राइव ना हो, अपना पाथवे नहीं है, तो आप कुछ भी नहीं कर सकते। और उसको पाथवे को डिफाइन करने के लिए करना क्या है? सबसे पहले अपना बेस लाइन डिफाइन करनी है कि आप खड़े कहाँ पर हैं और आपने जाना किधर है। यहाँ माशाल्लाह काफी स्टूडेंट्स हैं। कितने स्टूडेंट्स हैं? हाउ मेनी ऑफ यू हैव रेड एलिस इन वंडरलैंड? सही, वो 5-6 हाथ खड़े हुए। एलिस इन वंडरलैंड का स्टोरी क्या है? ये एक बड़े अच्छे ऑथर थे, लुइस कैरल साहब, उन्होंने लिखी थी। ही वाज बेसिकली अ मैथमेटिशियन एंड ही रोट दिस बुक बिकॉज इट वाज सपोज टू, क्या कहते हैं, इंस्टिगेट लॉजिक बच्चों के अंदर, कि लॉजिक बेस्ड डिसीजन आप कैसे ले। तो इस बुक के अंदर एक बड़ा अच्छा फेज आता है जिसके अंदर एलिस जो है वो जा रही होती है और एक दोराहे पे आती है, इट्स अ फोर्क इन द रोड, एंड वहां पे बैठ के वो कहती है मैंने जाना किधर है। और वहां पे दरख्त लगा होता है और दरख्त के ऊपर एक बिल्ली बैठी होती है। वो बिल्ली से पूछती है कि मैं राइट जाऊं या मैं लेफ्ट जाऊं। तो बिल्ली से पूछती है कि तुम्हारी डेस्टिनेशन क्या है? उसने कहा मुझे नहीं पता। तो बड़ा अच्छा जवाब बिल्ली का क्या है कि अब फर्क नहीं पड़ता कि आप राइट जाओ या लेफ्ट जाओ, क्योंकि आपको अगर अपनी डेस्टिनेशन ही नहीं पता तो इट डजेंट मैटर आप पाथ पे कौन सा ले। तो इंपॉर्टेंट चीज क्या है समझने वाली, सस्टेनेबिलिटी के एंगल से, कि आपने सस्टेनेबिलिटी प्रैक्टिसेस, हमने कहा जी सुन रहे हैं बायो पेस्टिसाइड यूज कर लो, अभी सब यूज करना शुरू कर दो, बायो फर्टिलाइजर आ गया, सबने यूज कर दिया, ऑर्गेनिक का नाम आ गया, रीजनरेटिव फार्मिंग आ गई। लेकिन मैं ये क्यों करूं? मेरा वाई क्या है? वाई एम आई डूइंग दिस? मेरी डेस्टिनेशन क्या है? मेरा गोल है सेट कि नहीं है? क्या मेरा गोल रेगुलेटरी प्रेशर्स के साथ अलाइन करता है कि नहीं करता? ये ऑपोजिट तो नहीं जा रहे दोनों एक दूसरे के। तो जब तक आपको अपना वाई नहीं पता होगा, चाहे वो इंडस्ट्री हो, चाहे वो एकेडमिक्स हो, चाहे वो रिटेलर हो या फार्मर हो या मिडिल मैन कोई भी हो, आपको अपना वाई नहीं पता होगा तो आपकी हमेशा डायरेक्शन फोर्स्ड होगी। आप हमेशा रिएक्ट कर रहे होंगे, प्रोएक्टिव नहीं होंगे। और रिएक्टिव अप्रोच हमेशा मोर कॉस्टली होती है और आपको पीछे धकेलती रहती है। जब डॉक्टर साहब ने भी नाम लिया कि मैं आर्टिस्टिक मिरर्स के साथ था, और ये बड़ी कंपनियां थी, और बहुत बड़ी कंपनीज जो होती हैं वो बड़ी क्यों बनती हैं? सिर्फ रेवेन्यू से बड़ी नहीं बनती, वो अपने थॉट प्रोसेस से बनी। कि आप अगर टाइम से आगे जाते रहेंगे तो जो रिस्क है, इसमें कभी आप फेल भी होते हैं और कभी नहीं भी होते, लेकिन जो रिस्क है उस रिस्क को आपने अगर पहले अपने साथ जोड़ लिया अपनी स्ट्रेटेजी के साथ, तो उसके रिवॉर्ड बहुत ज्यादा है। तो इंपॉर्टेंट चीज जो मैंने बता दी वो वाई था। अब कुछ एग्जाम्पल्स के साथ बता देता हूँ कि इंडस्ट्री किन चीजों को, प्रेशर्स को फेस कर रही है आजकल, जिसको हमने मिटिगेट करना आगे जाके। तो सिर्फ एक प्रेशर नहीं है कि आपने अपनी सप्लाई को सस्टेनेबल करना है। एक बड़ा इंटरेस्टिंग, बल्कि दो बड़े अच्छे रेगुलेशंस हैं जो यूरोपियन कमीशन की तरफ से आ रही है। एक कहता है कि जो आपने अपने पूरे प्रोडक्ट का लाइफ साइकिल असेसमेंट करके उसका डिजिटल प्रोडक्ट पासपोर्ट बना के देना है। वो डिजिटल प्रोडक्ट पासपोर्ट क्या है कि आपके कपड़ों का के साथ एक लेबल है, वो क्यूआर कोड भी हो सकता है, वो फिजिकल लेबल भी हो सकता है, जो बताएगा कि ये प्रोडक्ट की पूरी जर्नी क्या थी, ये कहाँ से स्टार्ट हुआ और कहाँ पे खत्म हुआ। नॉट ओनली इट्स फिजिकल जर्नी बट ऑल्सो इट्स एनवायरमेंटल जर्नी, बट एंड ऑल्सो इट्स सोशल जर्नी। तो इंपॉर्टेंट चीज सिर्फ ये नहीं है कि आपने, मैंने पहले कहा था कि सिर्फ क्लाइमेट को ऑन इट्स ओन नहीं देखना, सोशल को ऑन इट्स ओन नहीं देखना, आपने अपने फुटप्रिंट को ऑन इट्स ओन नहीं देखेंगे, तीनों को मिला के देखेंगे। जब तीनों मिला के देखेंगे तभी जाके वो आपका पासपोर्ट माना जाएगा। और ये क्यों माना जाएगा? क्योंकि जो कंज्यूमर है यूरोप में, उसको पता होना चाहिए कि जो प्रोडक्ट वो खरीद रहे हैं उसका इन्वायरमेंट का इम्पैक्ट क्या है, कि जो कंज्यूमर है वो अवेयर है वहां पर। अब यहाँ पे कितने हम लोग, सारे लोग शॉपिंग करते जाके कभी लेबल चेक करते हैं कि इस लेबल के ऊपर क्या लिखा हुआ है और क्या वो लेबल सही भी है कि नहीं है? ठीक है ना, खवातीन लॉन के कपड़े लेती हैं, समझ रही है 100% कॉटन है, उसमें ज्यादातर डिस्कस होता है, तो आपको अंदाजा नहीं कि उसका इम्पैक्ट क्या है। तो जो कार्बन इम्पैक्ट की सोच, अवेयरनेस है, वो आपके अंदर भी आनी चाहिए। जब वो सोच आपके अंदर आएगी, जो डिजाइन आप यहाँ पर कर रहे हो, तो उसके हिसाब से डिजाइन करेंगे। तो डीपीपी का बेसिक प्रिंसिपल ये नहीं कि आपने सिर्फ इंफॉर्मेशन दे दी, उसका प्रिंसिपल ये कि इट्स सपोज टू बी एन इंस्टिगेटर, अ कैटलिस्ट, टू मेक श्योर कि उसके बाद जो डिजाइन हो प्रोडक्ट वो इन्वायरमेंटली आपका फ्रेंडली हो। सेकेंड जो बड़ा इंपॉर्टेंट लॉ है जो कि वॉलंटरी फेज में भी चल रहा है, उसको कहते हैं CBAM, कार्बन बॉर्डर एडजस्टमेंट मैकेनिज्म। ये हमारे लिए बहुत इंपैक्टफुल होगा। वो इसलिए होगा क्योंकि इस लॉ की बेस के ऊपर अभी फिलहाल जो इंडस्ट्रीज हैं वो आपकी स्टील, सीमेंट, ये तो लॉ के अंदर आ गई है, मैंडेटरी रिक्वायरमेंट उनकी रिपोर्टिंग की, कि कोई प्रोडक्ट भी यूरोप के अंदर जो इंपोर्ट होगा, अगर वो उनके बेसलाइन, जो बेसलाइन क्या होगी कि अगर ये प्रोडक्ट यूरोप में बनता तो उसका एनवायरमेंटल फुटप्रिंट क्या होगा। अगर वो उसका फुटप्रिंट से ज्यादा है तो जो इम्पोर्टर है, दे हैव टू बाय EU सर्टिफिकेट्स टू कॉम्पनसेट फॉर एक्स्ट्रा कार्बन दैट हैज बीन एमिटेड एट द सोर्स। तो इसका साफ तौर से मतलब क्या होगा, अगर कोई चीज पाकिस्तान से जा रही है यूरोप में और उसका कार्बन फुटप्रिंट ज्यादा है यूरोपियन प्रोडक्ट के बने हुए, तो जो डिफरेंस होगा उसकी एक्स्ट्रा कॉस्ट हमें पे करनी पड़ेगी। लॉ कहता है कि इंपोर्टर पे करेगा, लेकिन हमें सबको पता है कि आखिर में जाके वो कॉस्ट सप्लायर के ऊपर ही आती है, तो वो आपकी कॉस्टिंग भी बढ़ जाएगी और आप कॉस्ट नेगेटिव होना शुरू हो जाएंगे। तो ये हमारे लिए इसलिए इंपॉर्टेंट है कि अब जो भी एक्टिविटीज आप कर रहे हो, बहुत सी एक्टिविटीज होती हैं, आप फार्म के ऊपर चले जाओ, आप एकेडमिक्स में चले जाओ, फैक्ट्री में चले जाओ, सबने वो एक्टिविटीज कर रही है, कोई सोलर लगा रहा है, कोई विंड लगा रहा है, कोई बायो फर्टिलाइजर पे काम कर रहा है, कोई ऑर्गेनिक पे, लेकिन उसका इम्पैक्ट और वेरीफाइड इम्पैक्ट कैलकुलेट करना बहुत जरूरी है। सिर्फ इतना कह देना काफी नहीं कि हमने सारा काम कर लिया। जो ESG रिपोर्टिंग का एक फ्रेमवर्क है, GRI बेस्ड रिपोर्टिंग होती है, आपकी CDP पे रिपोर्ट होती है, तो वो रिपोर्ट आपकी वैलिडेटेड और वेरीफाइड होगी तभी जाके आपका रेगुलेटर उसको एक्सेप्ट करेगा, वरना ये सारा क्या कहलाएगा, ग्रीन वॉशिंग, कि आपने कहा कुछ है लेकिन आपके पास उसको प्रूव करने के लिए कोई सर्टिफिकेट नहीं है, और उसकी भी एक अलग कॉस्ट। तो ये सब कॉस्ट बढ़ती जाती है अनफॉर्चुनेटली। ये हमारे ग्लोबल साउथ जहाँ पर हम लोग हैं, जो मैन्युफैक्चरिंग कंट्रीज हैं, वहाँ पे ये कॉस्ट ज़्यादा है। वजह क्यों? क्योंकि हम लोग सप्लायर, हमारे पास अपना कोई ब्रांड नहीं है बेचने के लिए। ठीक है ना, हमारा जो प्रोडक्ट है, अब हम माशाल्लाह सारे यहाँ पे कॉटन की फील्ड को थोड़ा बहुत जानते होंगे, क्या पाकिस्तानी कॉटन ब्रांडेड है? कोई जानता है दुनिया में ब्रांडेड कॉटन का नाम? दुनिया में कोई ब्रांडेड कॉटन है वैसे? आप सुनते हैं नाम अमेरिकन कॉटन का, कोई ब्रांड होगा, ठीक है ना, पीमा का नाम सुना आपने, सुपीमा का आपने नाम सुना हुआ है, ठीक है। इजिप्शियन कॉटन सुनते के साथ ही कहते हैं बहुत ही फाइन किस्म का उससे कपड़ा बनेगा। तो आपकी कॉटन का क्या ब्रांड है? कुछ भी नहीं। तो आपका कमोडिटी प्रोडक्ट है, और उसके अंदर आप इतनी मेहनत करते जा रहे हैं लेकिन उसको ब्रांड ही नहीं कर पा रहे, तो उस प्रोडक्ट का तो आपको कभी भी एक्स्ट्रा प्रीमियम कोई भी नहीं देगा। तो कोशिश ये करनी है कि अपने प्रोडक्ट को, अपनी इंडस्ट्री को, अपनी वैल्यू को बढ़ाना है आपने खुद। एक कमोडिटी प्रोडक्ट बना के नहीं बेचें इसको, इसको बना के बेचें एक ब्रांडेड प्रोडक्ट जिससे आप वैल्यू करें। और ये मैं बिल्कुल खत्म कर, डॉक्टर साहब आपने मुझे 10 मिनट कहा था, बस 2-4 मिनट और लूंगा आपके। अपने बोर्ड को हमने एक प्रेजेंटेशन दी कि जी हमने ये CSR के काम करने और उसकी ये कॉस्ट है। तो बोर्ड मेंबर्स ने मुझे सवाल पूछा कि इसका ROI क्या होगा। ROI पता है सबको? रिटर्न ऑन इन्वेस्टमेंट क्या होगी। अगर मैं CSR का काम करूँ, कॉर्पोरेट सोशल रिस्पॉन्सिबिलिटी है, किसी को हमने स्कूल की फीस दे दी या कहीं पे हेल्प कर दी, उसका ROI क्या होगा। तो आमतौर पर बंदा सोचता है कि आप ROI क्या होगा इसका, आपकी अच्छी मार्केटिंग हो जाएगी, कोई अखबार में खबर आ जाएगी, आप स्टोरी लगा देंगे। लेकिन ये सब बातें अच्छी लगती हैं, जैसे मैंने आपको कहा कॉन्टेक्स्ट हर चीज का। 5-6 साल पहले बोर्ड आपका सेटिस्फाई हो जाएगा इस बात से, लेकिन अब नहीं, क्योंकि आपकी कॉस्ट ऑफ मैन्युफैक्चरिंग बढ़ती जा रही है। ये सब चीजें जो आप कर रहे हैं वो आपकी कॉस्ट को बढ़ाती जा रही है। तो अब आपको सोचना है, स्टूडेंट्स के तौर पर भी सोचते हैं कि आपने इंडस्ट्री में जाना, किसी भी कोई काम भी जो हम कर रहे हैं जो कि प्रोडक्ट बनाने के अलावा है, वो क्या वैल्यू ऐड कर रहा है मेरे प्रोडक्ट के अंदर। वरना तो वो जो हम पढ़ते हैं सप्लाई चेन की डिपार्टमेंट में, वो जो वेस्ट है, वन ऑफ द वेस्ट है कि अगर कोई चीज वैल्यू ऐड नहीं कर रही है और सिर्फ कॉस्ट ऐड कर रही है तो वेस्ट हो गया हमारे लिए। तो मैंने कैसे CSR के प्रोजेक्ट की ROI निकाल ली है, मैंने किस तरीके से सस्टेनेबिलिटी की प्रोजेक्ट की ROI निकाल ली है। एंड ये कहा जाता है, वापस जाके पॉइंट मिलेगा कि आप आइसोलेशन में काम नहीं करेंगे। जब आप सस्टेनेबिलिटी पे काम करते हैं देन यू हैव टू वर्क टुगेदर विद द फैक्ट्री फ्लोर। चाहे फैक्ट्री फ्लोर हो, चाहे फार्म का एरिया हो, आपने ऑन द ग्राउंड आके काम करना होता है। मेरी कोई भी सस्टेनेबल प्रैक्टिस मेरे प्रोडक्ट की वैल्यू के अगेंस्ट नहीं जाती, वो उसके साथ साथ चलती है। सिंपल सी एग्जांपल, अगर मैंने अपना फैक्ट्री का कार्बन फुटप्रिंट कम करना है, कि मैंने जाना फॉर एग्जांपल कोई कोल पावर कंपनी है, मैंने कोल से कन्वर्ट करना उसको रिन्यूएबल्स के ऊपर, तो मुझे उसका कॉस्ट बेनिफिट एनालिसिस निकालना होगा कि अगर मैं कोल से सोलर पे जाता हूँ या विंड पे जाता हूँ, मेरी अपफ्रंट कॉस्ट ये है और मेरी ऑपरेटिंग कॉस्ट ये होगी, और अगर मैं कोल से मूव करता हूँ तो मुझे बेनिफिट क्या होगा। ये एक एग्जांपल होगी। दूसरी एग्जांपल है कि आपके फैक्ट्री ऑप्टिमाइजेशन पे आप जाते हैं, जहां जहां अपना पानी का यूज रिड्यूस करते हैं, जहां जहां हम अपने बिजली का यूज रिड्यूस करते हैं, तो वो एक एंड से आपको कॉस्ट रिड्यूस करके दे रही होती है, और दूसरी एंड से जब आप रिन्यूएबल पे जा रहे हैं तो वो आपको स्टोरी भी दे रही होती है। लेकिन उसके साथ साथ क्या मिल रहा है आपको, आप रिन्यूएबल की रिपोर्टिंग भी अच्छी कर लेते हैं, और जो अक्लमंद लोग हैं वो उसके अगेंस्ट आयरेक्स या कार्बन क्रेडिट ले लेते हैं। अब उसकी मार्केट भी ऐसे तो लो गई, लेकिन ये एक एग्जाम्पल है कि आप जो भी सस्टेनेबिलिटी का प्रोजेक्ट करते हैं उसका कोई ना कोई आपको ROI निकालना जरूरी है, तब जाके वो सस्टेनेबल होगा। वरना वो आप 1 साल पायलट करेंगे उसके बाद वो बंद हो जाएगा। एक बड़ी इंटरेस्टिंग चीज जो आ रही है, और आखिरी बात डॉक्टर साहब फिर मैं फिनिश कर देता हूँ, जो इंडस्ट्री के अंदर आ रही है वो आ रही है आपकी रिसाइक्लेबिलिटी के ऊपर और डिफरेंट फाइबर्स के ऊपर। बहुत दफा बहुत पेपर्स में लिखा जाता है, और उसी कॉन्फ्रेंस में डॉक्टर साहब जो हम थे तो वहां पे भी यही हो रहा था कि नंबर्स कोट हो रहे थे कि कॉटन जो है वो इतना पानी खींचती है कि बस इससे अच्छा है कि वो ना ही पानी खींचे। लेकिन वो नंबर्स बार बार गलत कोट हो रहे होते हैं कि 10,000 लीटर पानी एक क्या कहते हैं KG ऑफ कॉटन के लिए, कोई कहता है 14,000 लीटर पानी। लेकिन इस सब के पीछे इसको रिफ्यूट करने के लिए आपको क्या करना पड़ेगा, साइंस एंड टेक्नोलॉजी यूज करनी पड़ेगी। मेजरिंग डिवाइसेस आपके पास होनी चाहिए, सिर्फ सोच नहीं, सिर्फ साथ साथ उसके ऊपर काम भी करना पड़ेगा। पीटर ड्रकर जो बहुत बड़े इकोनॉमिस्ट है वो ये कहते थे कि इफ यू कैन नॉट मेजर इट यू कैन नॉट इम्प्रूव इट। तो आप जब कोई चीज़ मेजर नहीं कर पाएंगे तो आप इसको इम्प्रूव नहीं कर सकते। तो अगर आपने सस्टेनेबल कोई चीज़ उगानी है तो सबसे पहले उसको मॉनिटरिंग और इसकी वैल्यूएशन कैसे करें। आप वो पेपर बेस्ड काम तो नहीं हो सकता ना सिर्फ। तो एक चीज़ ये इंपॉर्टेंट है। दूसरी चीज़ क्या है, जब आप रिसाइकल के ऊपर जाते हैं आपका कंपैरिजन जो है वो किससे हो रहा होता है, वो आपका कंपैरिजन होता है वजन बेस्ड फाइबर के ऊपर और पॉलिएस्टर से भी होता है। तीसरा जो इसके अंदर इशू है वो अगेन जाता है LCA पे, क्या आप रिसाइकल के ऊपर या दूसरे मटेरियल पे जा क्यों रहे हैं, क्योंकि आपका जो लाइफ साइकिल असेसमेंट है इस प्रोडक्ट का, उसका एनवायरमेंटल फुटप्रिंट आपने रिड्यूस करके दिखा जितना हो सके। और अब यूरोपियन रेगुलेशन भी आ चुकी है कि आपके प्रोडक्ट के अंदर मिनिमम 15 to 20% रिसाइकल कंटेंट होना चाहिए। तो इसलिए ये प्रेशर इंडस्ट्री के ऊपर काफ़ी है, और इस प्रेशर की बेस पे अगर आप किसी भी इंटरनेशनल कॉन्फ्रेंस में जाएंगे यू विल फाइंड लॉट ऑफ मैकेनिकल एंड केमिकल बेस्ड रिसाइकल्स जो कि आपको कहते हैं कि ये हमारा वाला फाइबर यूज करें, वो कहते हैं ये हमारा फाइबर यूज करें। तो R&D प्रोडक्ट डेवलपमेंट बहुत इस फील्ड में हो रही है एंड आई थिंक इस यूनिवर्सिटी में होते हुए इट्स अ ग्रेट अपॉर्चुनिटी स्टूडेंट्स के लिए कि वो सिर्फ बेसिक्स पे ना देखें बल्कि इस पे भी देखें कि किस तरीके से यहाँ से इस इदारे से नए फाइबर कैसे जनरेट हो सकते हैं, वो फाइबर जो कि कॉम्प्लीमेंट करें हमारी इंडस्ट्री को, वो फाइबर नहीं जो कि हमारी इंडस्ट्री से बिल्कुल ही टेंजेंट पे जा रहे हो, लेकिन साथ साथ एक नई नीश भी प्रोड्यूस करें जिसकी बेस के ऊपर आप जितने भी सस्टेनेबिलिटी स्टैंडर्ड्स हैं उनको भी कवर कर रहे हैं, लेकिन जो प्रेशर आपके पास बाहर से आ रहा है उसको आप इंटरनली रिफ्यूट कर सकें बजाय इसके कि हर चीज़ बाहर से इम्पोर्ट करें। तो इन कंक्लूजन सबसे बेहतर क्या है कि आप अपना इंटरनल आपका पुश हो, अपने गोल्स हो, अपने टारगेट सेट करें और उस टारगेट्स की बेस के ऊपर आप चलना शुरू करेंगे तो जितने भी एक्सटर्नल प्रेशर्स हैं उसको भी कवर कर रहे होंगे। तो बहुत शुक्रिया आपके सुनने का, थैंक यू सो मच।
Thank you so much, sir, for your valuable presentation. Now, I would like to invite our next speaker, Dr. Syed Azaz Mehdi, who will be presenting the topic of OCA project on pure non-GMO cotton seed. May I request Dr. Shahid Mansoor to provide an introduction about him?
It's a pleasure to introduce Syed Azad Mehdi Saab. He is a very good friend, and we have been working together for last couple of years. Basically, he's representing a very important international project, which is Organic Cotton Accelerator. So they are providing funding for to 4 companies to produce pure non-GMO seed at relatively what we call as large scale. And I happen to be helping some of these scientists in purifying and getting pure non-GMO seed. And I think so we have one presentation by Shahrukh. We will tell that how we have been able to produce seed at one of the companies. So with this, over to you.
Thank you so much, Doctor, for the invitation as well as for giving the opportunity to talk this huge amount of relevant audience. I think my colleague Rubab is also in the call. If you can check in, yeah. My colleague Rubab is here with me. We will be co-presenting what is the role of OCA for in general, and also we have project for the seed purity as well. So meanwhile, after her, I will be communicating what we are doing in that seed project, how when it was started, and at what stage we are, and what we have done till now. Rubab, over to you.
Hello, everyone. Good afternoon, and thank you for having Organic Cotton Accelerator in this one of the very important event by iccbs University of Karachi. We are very grateful for this invitation. My name is Rabab Zara. I'm leading Organic Cotton Accelerator in Pakistan, and I hope most of you are already familiar with Organic Cotton Accelerator's role. We are a multi-stakeholder organization fully dedicated to advancing organic cotton, not just in Pakistan, but worldwide as well. In Pakistan, we are very closely working with suppliers, manufacturers, farmers, spinners, seed companies, and with brands to strengthen the organic cotton ecosystem. We work on a linkage part, we work on the overall supply chain integrity and security as well. Can we have next slide, please? And in this slide, you can see a wonderful map and it is showing our current presence. We are in Turkey, Pakistan, in India. We have also recently opened our program in Tanzania, while in Peru and Benin, we are doing assessment to again start some of the programs over there as well. So this is our geographical spread so far, and our headquarters is in the Netherlands. And we are operating from there in all those countries, and we have country mission offices in all three of and the four countries for now. And as today we are here in this call to share that how OCA is supporting the development of pure non-GMO cotton seed systems in Pakistan, and why we believe that seed purity is fundamental to the future of sustainable and organic cotton production. And if we think, we just think about that, why seed purity matters. So we all know that seed is the basic foundation, especially when it comes to cotton and to organic cotton. So the source of the cotton the seed is very important. I'll quickly set a context so that Azaz can explain how we are working under this project with 4 seed companies and 1 technical partner, and how we step into this program 2 years back from now, because we have felt a need for the genetic seed purity, because as you know, in the organic cotton projects, it's very important. And when we look into the seed selection, so it's very critical stage for us in our projects, because even for setting up organic projects, and every year when we do organic projects in Pakistan, so we are very much concerned about the seed source, and we are always very reluctant to trust on the market available organic seed, because and then at the later stage it causes some issues, especially at the germination stage. So we have developed a separate supply chain for our projects, which we are doing in Pakistan, because we are linking that produce with brands as well, and it has a lot of testing and validation being part of those produces, and we make sure that it is corely organic. So this is how we get to know that there is still a room and need in Pakistan to invest in this. And then we started this project 2 years back. It has 2 components. One is seed multiplication and then the commercialization phase. So in the first 2 years, we have worked on the seed multiplication. And now moving forward, because we're entering in the 3rd year, so we'll be focusing on the seed commercialization part. And I think most of the stakeholders would also be receiving an invitation from our side, who are already in this room, to take part in that event, which we're going to host in August, probably, to push forward the seed commercialization agenda. So I'll stop here because considering the time limitations, and I'll hand over to Azaz to run through all of you to one of the very exciting projects, and to let you know what are the learnings and what is the overall progress till now. So over to you, Azaz, and thank you everyone for having me in this call.
Thank you so much, Rubab, for setting up the context. I can start it from here. When I was drafting this presentation for you guys to get it understanding, or is tarah se, when you guys are up to somewhere, so you have in mind these sort of discussions would be there. So most of them, you guys were here from the morning, and you have heard that most of the seed research, development, multiplication techniques, different machineries, and also the softwares. Yeah, most of the things that have been communicated to you guys. But can you believe that in सबके पीछे what's the reason why everyone is doing it? Why the yield is important? Why the textile? We have heard like 3 aspects of it. Like with one, first is the benefit of the farmer, and then dinner, and then is the finished product or user end के लिए. तो क्या इनके back end के ऊपर कोई तो reason है that everybody is taking interest into it and spending their own time into that? So what's that interest? I think that this is more where we have to align on before starting of any of the project या कोई भी initiative कि उसकी जरूरत क्यों है? I think this is the question written on the top of my slide as well. Why this is important? If you guys are aware about the organic or the regenerative projects in Pakistan or overall, I think seed is the main component. कोई भी क्रॉप अगर उसका सीड जेनेटिकली मॉडिफाइड है तो वो क्रॉप कुछ भी हो सकती है. आई थिंक वो ऑर्गेनिक नहीं हो सकती. So I think this is where it all started. The need of the non-GM pure seed in Pakistan या overall भी जब हम बात करते हैं. So this was the reason for thinking on this project, and we drafted an idea, and we are now in the successful completion of its 3rd year. So I will be getting you through कि हमारा project कब start हुआ है and कहाँ पे है. But these are few of the reasons because of the contamination and non-availability. I think when you deep dive into the organic projects, तो आपको idea होगा market में शायद उसकी उतना availability नहीं है, but जितनी उसकी demand है. And also the purity is also a question. So that's why we think of drafting a project ताकि हम at least source को जरूर किसी तरह से purify कर सकें. So yeah, for the project, as Rubab has communicated, कि हमने 2024 में एक project draft किया, which was actually to purify the source of the seed, ताकि if anybody wants to multiply it, तो उसके पास एक pure source जरूर मौजूद होना चाहिए. So the project was started in 2024, and now we are in the 3rd year, and in the end of the 2027 we will be having like 3 variety का हमारे पास source जरूर pure होगा इंशाल्लाह, which we can be providing to different stakeholders as well. So we are planning for it. And the purpose of the project still remain with the farmer and जो हमारे end users हैं, farmers and the complete supply chain के साथ हमारा ये project हमने design किया to assist the farmers as well and also the ultimate users of it. We have 4 partners who are working with us. These are mostly basically an experienced certified seed companies as well, and also some of the groups who has expertise in in-house expertise in multiplication or the breeding concept of the seed. So we have with us Hyacinth is one of our partner who is implementing this into Balochistan, and then we have AG360 Search who are working with us in the province of Sindh. And RBDC is working in the province of Punjab, and then so Surti is also doing this in the 2 districts in Sindh and Balochistan. So these are our partners who facilitated us for the multiplication of the seed as well, and also the implementation of that. We have a technical partner because Rabab has communicated we are a multi-stakeholder platform, so we usually done the work through the third parties or our implementing partners. So Savi is with us as a technical partner. So they are supporting us during the implementation of this project. Methodology, I think I have tried to briefly mention the methodology, but we are actually using it within our project because most of you guys are very relevant to the seed as well as also for the agricultural productivity as well. So you guys can understand between the like किस तरह से जो seed था that was purified through different processes as well. So I think I would say thanks to Dr. Shahid, who was a very big support for us as well, and also he supported like going beyond the limit for identifying the gaps and through where it can be started and what's the target, what's the end. Even Saqib bhai was also communicating that if you don't know the destination, so you cannot do anything about it. So I think thank you for Dr. Shahid for providing us that instinct, and also we are working on it. So first we have done the single plant selection on a very small level with all of the 4 partners. It was a very small number of plant in the year 1, but we have gradually increased the percentage of the seed produced as well through kanamycin test. This is the test which is done initially like 45 to 50 days of the initial crop, and the symptoms occur on it. If that is if that occurs, and then we will do the process accordingly. BT strip test is a test which is used in organic as well, and most of you guys are aware about it. ये जो test हैं, kanamycin and BT strip test, these are used to detect the GM presence into a seed या फिर उसके leaf के अंदर GM presence के लिए. तो हम GM presence की ही बात कर रहे हैं. And the third test is qPCR. we opted the qualitative PCR instead of a quantitative, so that it would be a yes or no. If there is an impurity or the genetic presence into it, so we will be definitely discarding it, and then we will only be using the seed जिनका qualitative qPCR negative है. Learnings की I think it's important to share the learnings as well, क्योंकि हम अभी third year में हैं. तो the most important learning is when you start from the scratch, जिस तरह हमने start किया from the single plant and then we shifted it to the multiplication process. So I think resource efficient भी वही होता है कि जब आप base से start कर रहे हैं, when you know the आप अपना base single plant को भी अगर purify कर लें तो in the multiple years you can go beyond it. And also the site selection, because in organic it is important कि आप organic certified land के ऊपर ही अपने प्रोड्यूस को मल्टीप्लाई कर अपने सीड को मल्टीप्लाई कर रहे हो. The most important thing which I have learned during being in the organic cotton sector and working with the supply chain actors, most of the time, not all of the time, जब आप genetic percentage अपनी crop के level पे देखते हैं, it is comparatively lower than जब आप जिनिंग के लेवल पे या जिनर के पास जहाँ पे सेपरेशन होती है लेंट और सीड की, because there are a lot of processes involved like transportation में, उसकी storage में, mixing में, and then the process of the ginning. तो जो जिनिंग का प्रोसेस है, दिस इज ऑल्सो वेरी ग्रे एरिया वेयर वी हैव टू वर्क ऑन इट. बट ये हमारा तो स्मॉल प्रोजेक्ट था, तो वी हैव अ डेडिकेटेड जिनिंग मशीन जिसके अंदर से वो प्रोसेस हो गया, एंड देन वी विल हैव अ प्योरिफाइड सीड. So I think this is also important. Whoever is working or interested in working in organic cotton, so they have a separate ginning facility. अगर आप उसको conventional भी वहाँ पे process कर रहे हैं, तो this can be one reason. And also one other important learning, there are a lot of things that are mentioned into here. You can read it as well. But one of the learning is that you have to build a team of it. Nobody alone can do it. अगर आप सोचते हैं कि फार्मर का काम है ये प्योरीफाई करना, बिकॉज वंस वी हैव गिवन द सीड टू द फार्मर तो हमें प्योर ही वापस करेगा, दिस इज नॉट पॉसिबल. उसने लगाना है, देयर वुड बी एन आइसोलेशन डिस्टेंस, ही इज लाइक ही कैन नॉट ऑब्जर्व द आइसोलेशन डिस्टेंस या इस तरह की चीजें वो ना कर रहा हूँ. But this is important. We have to develop the team of like research institutes, educational universities, seed companies who are involved in the multiplication of it. ताकि eventually what we actually want कि हमारे पास pure source मौजूद हो. So these are some learnings from our project. At OCA, what I can communicate is that basically we work for the organic integrity. So whatever comes in the organic integrity word, because this is a very holistic word, integrity के अंदर जो भी चीजें आएंगी, so OCA will be working on it, and also for the guaranteed procurement. So these are basically two terms, but we are fully committed to the seat. If any one of you, we are very committed, and also we are running our project as well. So in the 2023, by this year even, we are planning some commercialization events as well, in which we involve the new stakeholders so that they can also get to know what we are doing, at what stage we are, how they can play the role into it. And also we encourage the applications from you guys as well. If any one of you want to share their knowledge, or they want to submit their inputs somehow for the purity of the genetic seed, so we are very welcome. But I think seed is very important, because इसके बगैर हम organic तो नहीं कर पाएंगे. So day by day the demand in the market is also increasing, but the challenges also still exist. So we need to work on it, and we need to do a sincere efforts to do that. And I would definitely appreciate ICCBS के जो colleagues सुबह काफी बहुत important बातें भी हुई, and also the processes भी define किए गए. I think some of them are new for me as well. So I think I'm glad that we joined this workshop, and also we are learning we have a lot of like learnings for ourselves as well. But we will still be open for any sort of your feedback or input, because this is some project that will be with you guys, will be with the farmers eventually. तो जब हमने वो implement ही किसी isolation में नहीं करना, farmers के साथ ही करना है. So why don't we should be striving alone? So you guys should also be coming into being the part of it. then that's all from my end. So if you have any questions, so please go ahead. Rubab is also in the call, so we will try to accommodate your questions. Thank you, everyone.
Thank you, sir. Now we will move forward towards the next speaker. Our next speaker is Sir Minhaj Khoker from USA, and he will be presenting the topic on food, water, and energy security under climate change and running of data centers in the most sustainable way. Sir, I request you to kindly deliver the topic. Thank you.
विराज साहब आप ऑनलाइन है ना?
जी जी बिल्कुल. Okay. Can you hear me?
जी जी, we can hear you. And Minhaj Saab is a good inspiration. He is from Silicon Valley and entrepreneur and technology expert, sustainability. इनकी कुछ यहाँ पे activities पाकिस्तान में भी हैं, and he has established some very interesting models in near Islamabad. And I would like to welcome Minhaj Saab. Thank you very much, sir, for joining us.
बहुत शुक्रिया. बिस्मिल्लाह रहमान रहीम. Assalamu alaikum wa rahmatullah. My subject is a little bit different. I mean, I've heard some very interesting presentations today. My focus is how we can build new industries. If we can build climate change as an industry, using agriculture and new technologies in agriculture, then I think Pakistan can move forward very well. On the other hand, if we keep doing the things the way we have been doing, we will keep struggling. I was just curious and looking at the GDP growth of Pakistan, and it was above 6% in 1960s, and since that time it has not gone up there. Anyway, so let me quickly go to what is the subject here. If we can use agriculture and combine it with energy security and water security, and if we can run data centers and AI for climate change as well as agriculture, I believe that Pakistan can earn about $1 billion in credits within 6 to 7 years. I'm going to show you just a quick slides here. This is what I do. I'm growing, this is the size of my lemons. I'm using solar energy to grow my food and at the same time conserve water. I use only about 5 to 10% in my vertical farms and about 25% in outdoor agriculture. Just going back a little bit, here's a picture at the very bottom with Dr. Ataur Rahman Saab in the year 2000. This is in New York with the Foreign Minister of Malaysia. I used to be the advisor to the Prime Minister of Malaysia, and started with 5 people at the Advanced Microchip Design Training Center. And now there are more than 12,000 engineers in Malaysia. And the work that I did, alhamdulillah, Malaysia earned more than $32 billion. So I'm used to building multi-billion-dollar projects. Dr. Ataur Rahman, he visited our microchip design training center. I had about 128 engineers working with me. And but the good thing was I was advising Dr. Mahathir. And the diagram here, this is worth $50 million. This diagram, you see, it's Four Seasons Hotel Chicago. Dr. Mahathir, he came to Chicago, and then he went to the United Nations conference. And we had this slide, but unfortunately, fortunately, my assistant, he forgot it in Malaysia. And so I just took a piece of paper from the Four Seasons Hotel and I drew this diagram. And we had about 200 people come from all over United States, but mainly from Silicon Valley. Most of them scientists and engineers, and what we could do for Malaysia. And so I drew this diagram, the magnitude of action and whether you're late or early. If the magnitude is small, if it is little and you are late, you are going to have failure. On the other hand, if the magnitude is large and you are early or the effort is huge, then you will succeed. And this is a very important diagram. Dr. Mahathir, he kept looking at this diagram, a small piece of paper in his hand, and then he said, how much money do you need to get the microchip design project started in Malaysia? I said, we need about $50 million. And he said, okay, you'll get it. He went back, got an approval from the cabinet, and this is how we started this project. And you see here, most of the people came from United States to help. Nowadays, I am building climate change industry, as an industry, and I'm doing this vertical farms. Dr. Gerald Raymond of Kassel University is the head of the Organic Federation of Germany. He called me and he said, I heard you have a vertical farm. And by chance, I was in Pakistan. So I said, yes. He said, I would like to visit. But then he said, I have only about 30 minutes. I said, no problem, come. He came. He stayed 3 hours and 35 minutes. He said, Pakistan is not a technology country. I'm shocked to see this thing in Pakistan. And right now, he's in negotiations with me that he wants to have 300 of these in Germany. Anyway, then this is Dr. Masood Al-Mari. He is the head of food security in Doha. He visited this, and we're in discussion right now with the Qatar Education City for a pilot project of about $30 million. So, if the technology is there, you can build something. If you take a look at these pictures, you see in the background, there is a solar panel. This solar panel is producing energy. My 1 acre vertical farm produces about 550 kilowatt hour or half a megawatt of electricity. That is enough for me to run a small data center. Now, I'm not going to take much time in going through some of these slides, but Netherlands, they built an agriculture, as an industry. How did they do that? During Second World War, they used to have famine, but they built it as an industry by going indoor. This is a very important step. Dr. Rudy Rabinj was the person, and he said, our environment is not suitable for outdoor agriculture. We need to go indoor. And they started to build greenhouses. And that's what made Netherlands an agriculture powerhouse. Today, last year, their exports were $131 billion. What they did is, again, they built massive miles and miles of greenhouses. Spain copied it in Almeria, Al-Andalus province. Spain, during the 2015-16 financial crisis in Europe, Almeria saved Spain. One of the biggest problems with these farms is they use these grow lights, and electricity cost is going higher. And this is where I had to come up with the idea how I can have zero electricity cost and at the same time produce enough electricity that I can even sell electricity and have another stream of income. This is our vertical farm in Islamabad. And it looks beautiful, works even beautifully. China built one in Beijing. They spent $45 million. I didn't spend $45 million. I'm not going to tell you how much I spent, but anyway, I'm able to grow rice, not 3 to 5 tillers, but 16 to 20 tillers from a single seed. You're talking about multiplication of seeds, speed breeding, yes. We did, even for Dr. Shahid Mansoor Saab, I mean, we did wheat. And I'm going to quickly go through. This is basically, if you take a look at it, it's about 14.5 feet tall corn plant. Then we can do even, basically fodder. Lately, we have been doing cotton. This is the former CEO of FONGRO, the military division of the corporate culture, Major General Aslam Chaudhry. He is the former CEO. And then 4 brothers in Lahore. They came to me and they said they've been looking at different countries and to see how they could do speed breeding. And so this is where we are doing the testing of cotton for them. This is basically cotton in aquaponics system. But then we tried it with different grow lights and red solar. Then also again in soil beds. And if you take a look at it, our best result was that we could have pure seed, good quality, grow it in soil with red light and red solar, not just red light, 2 different lights, FS1 and FS5. And this is where we are able to grow cotton seed in about 60 to 70 days, not 90 plus days. Now, all of this basically is to get to this project of whereby we can do cluster of vertical farms and have solar. We have a special technique of solar in the field. And this is where we produce enough electricity and at the same time grow food, and we can run data centers. And data centers, AI, this is the future. So anyway, I think I'm just going to, I have a few more slides, but I'm going to stop here. I've been working in Nigeria as well. But basically, it's a multi-billion-dollar project that we are doing in few countries. But in Pakistan, I'm hoping that we can utilize the scientists and the students there. We could train them and build this as an industry. So I think I'm going to stop here. There is a financial part of it, but looks like my time is up. So that's it. Thank you.
Thank you so much, sir, for your valuable presentation. Now, I would like to invite our next speaker, Dr. Rafi Lassi and Shahrukh, Mr. Shahrukh, and he will be presenting the topic on role of housing in maintaining and producing pure seed and biofertilizers. May I request sir to kindly deliver the topic.
Assalamu alaikum. I am Shahrukh Ali Kirio, and I am member of SM Group. Today, I've given a topic, role of hyacinth in maintaining and producing pure cotton seed. But before telling the approaches of hyacinth and maintaining the seed, I'll introduce the SM Group. SM Group is established in 2012. It is comprises of SM Cotton Industries, SM Agricore, SM Constructions, Hyacinth Agricultural Services. It is a seed unit which is working on the purity of seed. SM Trading Co. and SM Enterprises Bela. It's also a ginning industry which is located in Bela, around 50 kilometers away from Las Bela. So let's start the activities of Hyacinth. Hyacinth is working on the research and development of GMO and non-GMO seeds and maintaining the pure, genetically pure seed, multiplication of pure seed, and working on the development of sustainable agricultural technologies. And Hyacinth is working on the biofertilizer plant, which is manufactured in Uthal Las Bela, which is ready for production now, and it is signed a MOU with NIPG for technology transfer, which is scientifically oversighted by Dr. Fatiha Mubin and Dr. Hazrat Ali. So now discuss the approaches for pure cotton seed. So for the purification of seed, firstly we need pure varieties. We need non-GMO varieties. So we have two non-GMO varieties. One is Nia Kiran from Nia Faisalabad, and other one is Nia Nuri, which is from Nuclear Institute of Agriculture, Tandojam. And we are technically supported by my mentor, Dr. Shahid Mansoor, and Mr. Haider Naqvi Saab, who is the Deputy Director of Agriculture Extension, Balochistan. And we are also working with OCA, एजाज भाई पहले ही एक्सप्लेन किया है, मैं थोड़ा उसमें ऐड करूंगा कि we have MOU with OCA and working for since 3 years. In first year we selected 350 plants, then go through the 25 acres, and now in the next year our plan is for 500 acres. These are the field management, field establishment. ये भी एजाज भाई बता के गए हैं, मैं थोड़ा ऐड कर देता हूँ कि before purity, when we start the purification of seed, we need some steps. First step is kanamycin screening test. Basically we use leaf paint and of kanamycin on the cotton leaf before flowering to avoid the contamination. Then it's go through the BT strip test, then it's go through the qPCR, and then in the fourth step we multiply the seed in a single line. And when we preserve the seed, the labeling and storage and ginning facilities are very important. For labeling the seed, you should focus on these labels, these components. Which first one is farmer name, location, plant number, date of picking, date of harvesting, the taste status, and the unique IT code which is given by breeder. And we have a ginning facility, small rolling machine for ginning single plants. And we have also a big machine with I think 80 RRs for large amount of cotton seed. And we have plant collaboration with iccbs for genetically pure seed production, biostimulation production. एंड हमने जो है बायोस्टिमुलेंट जो है बलूचिस्तान में यूज किए हैं, जिनके बहुत अच्छे रिजल्ट्स आए हैं, जिसमें जब हमने कॉटन पे वो यूज किए तो नंबर ऑफ फ्लावर्स जो है कॉटन के बढ़े और अल्टीमेटली द यील्ड विल आल्सो इंक्रीज. और बायोपेस्टिसाइड भी हम इस साल यूज कर रहे हैं और उनके रिजल्ट्स बहुत इफेक्टिव है माशाल्लाह. द की अचीवमेंट्स आर, वी हैव established a cotton processing facility in 2012. Organic cotton ginning was started in 2018, and we have signed an organic cotton and wheat project on 15,344 acres under NOP USDA certification, a strong partnership with WA Pakistan and AED Balochistan, seed multiplication program, and we have a biofertilizer plant which is fully developed and ready for the production. Thank you, everyone.
Thank you so much, sir. Now we will move towards our next speaker, who is Dr. Farooq Hassan, and he will be presenting the topic on commercialization of biofertilizer. May I request him to kindly come on stage and deliver his talk.
We are very proud that one of our students from NIPD, Dr. Farooq Hassan, has established first biotechnology company in Pakistan. And being a lady and then working in area was a big commitment. So and she has done very well, established a company. तो इनके जो लिविंग स्टाइल से पता चलता है पैसे भी ऐसे कमा रही है. So this is a very good opportunity for students to think that this, don't only think about jobs after PhD, but also establishing a business. So here we have a role model. We are very proud of her. Thank you, madam, for coming all the way from Lahore.
बिस्मिल्लाहिर्रहमानिर्रहीम, अस्सलाम वालेकुम. और मैं इससे पहले थैंक यू करूंगी डॉक्टर शाहिद मंसूर का. मैं इनकी स्टूडेंट भी हूँ और निफ्टी की स्टूडेंट हूँ. इन्होंने आज मौका दिया कि मैं आपके साथ अपना काम और जो कुछ हमने अचीव किया और जो कुछ हमने डिलीवर किया उस पर बात कर सकूं. जब मैंने अपनी प्रेजेंटेशन रात फाइनल की तो जो मेरा इंट्रोडक्शन स्टार्ट करने का था वो थोड़ा डिफरेंट था. बट जब मैंने अभी प्रेजेंटेशन सुनी है साकिब साहब की और आजाद साहब की, तो मैं स्टार्ट थोड़ी सी चेंज के साथ करूंगी. और वो चेंज ये है कि दोनों जो हमारे गेस्ट स्पीकर्स हैं और जिन्होंने हमें इतना अच्छा हमें बताया, और इनका टॉपिक भी सस्टेनेबिलिटी के बारे में था. दोनों ने जो बात की वो ये कि आपको अपनी डेस्टिनेशन का पहले पता होना चाहिए, फिर ही आप आगे बढ़ सकते. और क्या बीइंग पाकिस्तानी हमें अपनी डेस्टिनेशन का पता है? हमने बात की यहाँ पे ऑर्गेनिक कॉटन की, ओका काम कर रहा है, डिफरेंट इंस्टीट्यूट्स काम कर रहे हैं, डिफरेंट कंपनीज काम कर रही हैं, लेकिन वो क्या पाकिस्तान के लिए काम करें? हम यूरोप के लिए काम कर रहे हैं. पिछले 1 साल में 1.5 साल में मुझे कुछ 7-8 यूरोपियन कंट्रीज में जाने का मौका मिला, जिसमें बेल्जियम, फ्रांस, स्पेन, नॉर्वे, स्वीडन एंड नीदरलैंड, डिफरेंट और भी 2 डेनमार्क. उन सारे मुल्कों की जो पॉपुलेशन है वो हमारी उन सब की अगर आप नेट पे देखें तो हमारी पॉपुलेशन के बराबर उन 6-7 मुल्कों की मिलाकर भी नहीं है. 6 से 7 मुल्कों की पॉपुलेशन हमारे मुल्क एक अकेले की पॉपुलेशन से कम है. एंड दे हैव वेरी गुड क्वालिटी लैंड, they have very good quality fertilizer, they have very good quality of seed, they have all the infrastructure, government support बहुत है, ज़्यादा है, पैसा बहुत है. Then why they are not growing crops, organic crops in their own country, and they are asking ourselves to bring organic cotton and organic crops from our country? सोचने की बात है ना कि नहीं? स्टूडेंट से मेरी बात करी, मैं साइंटिस्ट से बात करी. हम अपने मुल्क में ग्रो करके उनको दें, बहुत अच्छी बात है. एज अ बिजनेस पॉइंट ऑफ व्यू पाकिस्तान को बिजनेस मिलेगा, रेवेन्यू होगा. आम कॉटन जो है, जो कमोडिटी कॉटन है, वो हमें कम पैसे देती है. अगर आप उसको ऑर्गेनिक करके और उसकी ट्रेसेबिलिटी करके उसका सारा कुछ करके आप उनको करते हैं तो आपको ज्यादा रेवेन्यू जनरेट कर. लेकिन क्या डॉक्टर सर, आपसे मेरा सवाल है कि क्या ये सस्टेनेबल है? हम अपने मुल्क में माइनिंग करके अपनी जमीनों को बंजर बनाकर उनके लिए ऑर्गेनिक कॉटन बना रहे हैं बिकॉज दे कैन अफोर्ड। आप अफोर्ड कर सकते हैं ऑर्गेनिक कॉटन की शर्ट? ये जो स्टूडेंट्स जितने बैठे हैं यहाँ पे, इवन आई कैन नॉट अफोर्ड। एक्सपेंसिव इतनी है ना। आप जब बाहर जाते हैं वहाँ ऑर्गेनिक कॉटन की चीजें पड़ी होती हैं सारे, वेरी वेरी एक्सपेंसिव। यहाँ से भी एक्सपेंसिव जाती है, जैसे मैंने बताया कॉस्ट इनकर होती है, ट्रेसेबिलिटी पे कॉस्ट है, उसकी प्रोडक्शन पे कॉस्ट है, उसकी सीड मैन्युफैक्चरिंग पे कॉस्ट है। तो फिर हमें क्या करना है? हम पैसा तो नहीं कर सकते, हमें पैसा भी कमाना है। सो वी हैव टू ग्रो दैट। हम उनको अगर छोड़ भी दें, उनकी बात छोड़ भी दें, तो हमें अपने लिए भी तो अर्निंग करनी है ना। तो हमें कमोडिटी से निकल के स्पेशलाइज्ड प्रोडक्ट्स में जाना है और ऑर्गेनिक कॉटन या ऑर्गेनिक फूड प्रोड्यूस करना है, बट एट व्हाट कॉस्ट? क्या हम अपनी माइनिंग करनी शुरू कर दें? अगर हम उसमें फर्टिलाइजर जमीन में डालेंगे नहीं, तो वहां से हम माइन करते रहेंगे तो 15 से 20 साल में बंजर हो जाएंगे। इज इट नॉट ट्रू? ऐसे है ना। तो फिर आज इस फोरम पे जब बैठे हैं, जहाँ पे हमारे फ्यूचर साइंटिस्ट्स हैं, हमारे कॉलीग्स हैं, हमारे टीचर्स हैं, तो हमें आज वो सोचना है कि वो एंड तो हमने करना ही करना है फॉर आवर सस्टेनेबिलिटी, फॉर आवर प्रोग्रेस, बट दूसरे एंड को भी सस्टेन करना है, उसको भी बचाना है। हम उसे खुला एंड नहीं छोड़ सकते। आज से 15 साल बाद जब मेरा बेटा यहाँ पे खड़ा होगा तो वो क्या बताएगा कि बंजर बलूचिस्तान है, बंजर सिंध है, वो फलां कंपनी आकर इतना ऑर्गेनिक उगा के चलेगी, नाउ यू हैव नथिंग टू डू। ठीक है ना। आप खुराक अपनी आवाम नीदरलैंड से खरीदें, कैन वी अफोर्ड दैट और शुड वी अफोर्ड दैट? तो उसके लिए जब सस्टेनेबिलिटी की बात करनी है, अपनी जमीन के लिए काम करना है, तो हमें करना है, उन्होंने नहीं करना। उन्होंने तो आपको टाइट करते जाना है, अब ये डॉक्यूमेंट ला दें, अब ये करते हैं, फलां करते हैं। करें, जरूर करें, बट अपने लिए भी रास्ता बचा के रखें। ओके। और उसका रास्ता क्या है? उसका रास्ता ये है कि फॉर ऑर्गेनिक प्रोडक्शन, फॉर एनीथिंग लाइक कॉटन, फूड एंड एनीथिंग, आपके पास ऑर्गेनिक फर्टिलाइजर्स होनी चाहिए। सस्टेनेबल क्लाइमेट उसके जो कंपटीशन में जो क्लाइमेट चेंज आ रहा है, उसके कंपेटेबिलिटी में आपके पास अच्छी फर्टिलाइजर्स होनी चाहिए। यू शुड यूज integrated use of chemical and organic fertilizer or biofertilizer to sustain your own fertility. लकीली पाकिस्तान में मुझे इसी चीज के ऊपर काम करने का मौका मिला। जैसे हम अभी सुबह से आप पढ़ रहे होंगे, तो मैं बहुत ज्यादा डिटेल में नहीं जाती कि केमिकल फर्टिलाइजर क्या करते हैं और फलाना क्या करता है फलाना। वो हमारी जरूरत है, हमने उसको मिटिगेट करना है, उसको कन्वर्ट करना है, ताकि हमारी यूज, हमारा जो एंड डेस्टिनेशन है, जो हमारा एंड टारगेट है, कि हम ऑर्गेनिक और ब्रांडेड चीजें प्रोड्यूस कर सकें और साथ साथ अपनी जमीन को भी बचा सकें। हमने केमिकल फर्टिलाइजर्स को इंटीग्रेट करना है विद सम ऑर्गेनिक फर्टिलाइजर्स एंड बायो फर्टिलाइजर्स, ताकि हमारी जमीनें जो है वो सस्टेन कर सकें और हमारी प्रोडक्शन भी बेहतर आ सके। सारी चीज लोग बुक्स में पढ़ चुके हैं, क्लाइमेट चेंज में फला फला, मैं इसको स्किप करती हूँ। हम ये कहते हैं कि हम बायो फर्टिलाइजर्स के ऊपर काम करते हैं। तो बायो फर्टिलाइजर्स क्या है? एनीबॉडी हैव आइडिया? सुबह की कोई प्रेजेंटेशन सुनी बायो फर्टिलाइजर पे? स्टूडेंट्स हो चुके हैं? मैं सिर्फ पीछे वालों से बात कर रही हूँ, आगे वाले तो पढ़े लिखे लोग हैं, उनको सब पता। तो पीछे वालों से थोड़ा सा जाके 2 मिनट के लिए मेरा साथ दे, तो आपको मजा आएगा आगे। देंगे साथ? थैंक यू। बायो फर्टिलाइजर्स क्या है? एनी आइडिया? बायो फर्टिलाइजर्स जो हैं, दे आर प्रोडक्ट्स कंटेनिंग लिविंग सेल्स ऑफ डिफरेंट टाइप्स ऑफ माइक्रोऑर्गेनिजम्स। वो एल्गल भी हो सकते हैं, वो फंगल भी हो सकते हैं, वो बैक्टीरियल भी हो सकते हैं, that convert the nutritionally important elements into from unavailable form to अवेलेबल फॉर्म। और जब आप इनको इंटीग्रेटेडली यूज करते हैं विद द हेल्प ऑफ केमिकल फर्टिलाइजर एंड ऑर्गेनिक फर्टिलाइजर, तो ये आपको केमिकल फर्टिलाइजर से बेहतर रिजल्ट्स देते हैं और आपकी जमीन की जो फर्टिलिटी है उसको बढ़ाते हैं। फर्टिलिटी क्या होती है, कुछ कोई पता है किसी बंदे को? हम ये कहते हैं ये जमीन फर्टाइल है, ये जमीन फर्टाइल नहीं है। व्हाट इज द डिफरेंस? जी जी सॉरी, मिनरल रिच सॉइल तो आप कहेंगे वो फर्टाइल सॉइल है जिसमें माइक्रो ऑर्गेनिजम्स की एक्टिविटी हाई होगी। यस, अकेले मिनरल्स कुछ नहीं कर सकते। हमारे पास बंजर पहाड़ हैं पूरे पूरे पाकिस्तान में, मिनरल्स से लदे पड़े हैं, बलूचिस्तान में ही देखें। अनटिल और अनलेस उनके अंदर माइक्रोबियल एक्टिविटी नहीं होगी, आप उसको फर्टाइल कंसिडर नहीं कर सकते। तो हमें उनमें फर्टिलिटी उसको बढ़ाना है, माइक्रोबियल एक्टिविटी को बढ़ाना है उन एरियाज में जहाँ पे हम ऑर्गेनिक प्रोड्यूस करना चाहते हैं, ताकि हमारी जमीन की फर्टिलिटी भी कायम रहे, बल्कि वो एनहांस हो और हमें प्रोडक्शन भी अच्छी मिले और हमें ट्रेसेबिलिटी भी अच्छी मिले। अब उस सिलसिले में मैंने क्या किया, मैं अपनी सिर्फ बात करूंगी कि मैंने क्या किया। मैं ये बात नहीं करूंगी कि फलां जगह क्या हो रहा है, फलां जगह क्या हो रहा है, मैंने क्या किया या हमने क्या किया, हम सिर्फ उसकी बात करें। मेरी बात सुनिए, कहानी मेरी सुननी है कि नहीं? सर आपने इनको बिरयानी खिला दी है, सो रहे बिरयानी वैसे वाकई। बायो फर्टिलाइजर की इस वक्त जो ग्लोबल मार्केट है वो तकरीबन 2 से 5% है, लेकिन जो अब फिगर्स आ रही हैं उसमें तकरीबन 8 से 13% बायो फर्टिलाइजर्स पर ईयर जो है दुनिया में इंक्रीज होना शुरू हो गई हैं, उनका यूजेज जो है वो बढ़ गया। अभी आप देखें कि पाकिस्तान में कितनी लैंड जो है एरेबल लैंड कम हो गई है। हम हाउसिंग सोसाइटीज बना रहे हैं, हम पुल बना रहे हैं, हम सड़कें बना रहे हैं, हम नए शहर डेवलप कर रहे हैं, तो हमारी जो एरेबल लैंड है वो बहुत कम होती जा रही है। अभी मुझसे पहले जो प्रेजेंटेशन आपने सुनी उसमें था कि जी आपको वर्टिकल जाना है, आप जो अपने फॉर्म्स हैं वो वर्टिकल ग्रो करने शुरू करते हैं। शायद हम पे भी वो वक्त कभी आ जाए, लेकिन अगर हम उस वर्टिकल फॉर्म बनाने की बजाय अगर वर्टिकल हाउस बनाना शुरू करें तो शायद हम ज्यादा कामयाब होना शुरू हो जाए। तो इस तरह ब्राजील में, जो कि इस तरह सोयाबीन प्रोडक्शन में ग्लोबल लीडर है, वो अपनी फर्टिलाइजर का 85% बायो फर्टिलाइजर यूज कर रहे हैं। इंडिया, चाइना, यूरोपियन यूनियन, यूनाइटेड स्टेट्स में भी ये बढ़ती जा रही है। हम क्या कर रहे हैं? हमने पाकिस्तान में 3 कंपनीज इंट्रोड्यूस की, माइक्रोबियल बायोटेक्नोलॉजीज, बायोटेक फर्टिलाइजर्स एंड फर्स्ट बायोटेक्नोलॉजी कंपनीज। ये प्राइवेट सेक्टर में बायोटेक्नोलॉजी की बनने वाली सबसे पहली कंपनी। मैं अपना थोड़ा सा डॉक्टर साहब ने करवाया, मैं थोड़ा सा कहूँ कि आई एम पीएचडी इन बायोटेक्नोलॉजी जेनेटिक इंजीनियरिंग, मुझे 20 साल हो गए हैं पाकिस्तान में बायो फर्टिलाइजर की कमर्शलाइजेशन, डेवलपमेंट एंड मार्केटिंग के ऊपर। आई हैव रिसीव्ड मल्टीपल अवार्ड्स एंड डिस्टिंक्शंस। लेकिन जो मेरी सबसे बड़ा अवार्ड ये है कि मैंने पाकिस्तान में निजी से पीएचडी करने के बाद मुझे जॉब मिल गई पंजाब यूनिवर्सिटी में। पहले निजी में मिली, फिर मैंने पंजाब यूनिवर्सिटी ज्वाइन की। 3 साल मैंने वहां पे एज असिस्टेंट प्रोफेसर काम किया एंड आई वाज 32 ईयर ओल्ड एट दैट टाइम। और फिर एक दिन मैं लिप्ची गई और लिप्ची में एग्जीबिशन लगी हुई थी। उन्होंने कहा कि जी हम ये बायो पावर, जो बायो फर्टिलाइजर की टेक्नोलॉजी है, उसको कमर्शलाइज करना चाहते हैं, तो कोई है जो उसको लेना चाहे? तो मैंने हाथ खड़ा कर दिया। आई सेड यस, आई वांट टू डू। मैंने अपनी पंजाब यूनिवर्सिटी की 3 साल की जो मेरी सर्विस थी, आई रिजाइंड। मैंने उसी दिन अपनी जॉब से रिजाइन किया और मैंने अपने घर नहीं बताया कि मैंने जॉब से रिजाइन किया। 2 साल मेरी माँ को पता था कि यूनिवर्सिटी ही जाती है। ड्राइवर को भी कहा हुआ था कि हमें फोन करें तो बताना बाजी के साथ यूनिवर्सिटी। मैंने अपना किचन खाली किया और अपना ड्राइंग रूम खाली किया एंड आई स्टार्टेड आइसोलेशन ऑफ बैक्टीरिया। 2 साल मैंने बैक्टीरिया आइसोलेशन पे काम किया। मेरा ऑटोक्लेव जो था, मेरा प्रेशर कुकर था, मेरे जो लैमिनार फ्लो थी, वो 2 मोमबत्तियां थी जो मैंने गत्ते के कार्टन में रखी हुई थी और उनको मैं चलाती थी। और दैट वाज इट। केमिकल्स खरीदने के थोड़े से पैसे थे, तब सस्ता जमाना था। ग्लासवेयर ले लिया, प्लेटें ले ली, क्योंकि काम नीचे से सीखा हुआ था, काम आता था करना कैसे है। और जो हमारे प्रोफेसर थे, डॉक्टर शाहिद मंसूर वाज वन ऑफ देम, और बाकी हमारे डॉक्टर फौजी डॉक्टर सुहेल हमीद वगैरह, इन्होंने इतना अच्छा सिखाया था और हमने इतना अच्छा सीखा था कि हमें फर्क ही नहीं पड़ता था कि इंस्ट्रूमेंट है या नहीं है, हमें ये जुनून था कि काम करना है। तो उस 2 साल में मैंने तकरीबन कोई 50 स्ट्रेंस जो हैं पाकिस्तान के मुख्तलिफ इलाकों से आइसोलेट किए, जॉब रिजाइन कर दी और घर में काम, किचन और ड्राइंग रूम, दैट्स इट। सारी सारी रात में काम करती थी, दिन में जाहिर है फैमिली को भी देखना, बच्चे भी देखने, उसके बाद मैं अपना काम करती। आफ्टर 2 years मैं इस काबिल हो गई कि मैंने अपना पहला प्रोडक्ट लॉन्च किया। और जिस दिन मैंने अनाउंस किया कि मैंने आज अपना प्रोडक्ट लॉन्च करना है, सेम डे एक बहुत बड़ी कंपनी ने मुझसे कांटेक्ट किया कि हमें आप पे बिलीव है, हम आपका प्रोडक्ट लेना चाहते हैं, हम आपके साथ मार्केटिंग करना चाहते हैं। मैं उसका नाम बाद में बता। तो मैंने स्टार्ट किया उस कंपनी के साथ प्रोडक्शन करने में। 200 बैग्स का ऑर्डर मिला और हम इतने खुश, हमने ड्राइंग रूम में फर्श साफ किया, स्पिरिट से उसको सारा कंटेमिनेशन फ्री किया, स्टरलाइज किया, एंड वी मेड 200 बैग्स। और वो बैग्स बने, ट्रायल में गए, उसका रिजल्ट आया, इतना अच्छा उनको रिजल्ट आया कि उन्होंने मेरे साथ अगले 10 साल का एग्रीमेंट साइन किया। और उन्होंने मुझे 2 ऑफर्स की। ये मैं स्टूडेंट्स के लिए बता रही हूँ स्टोरी, ये मैं स्टोरी पहले बतानी नहीं थी, इनकी बातें मैं सिर्फ टाइम बताया। वो जो उन्होंने एग्रीमेंट साइन किया तो मुझे 2 ऑफर्स की। एक ऑफर ये थी कि हमारे पास जॉब कर लो, और ये बात है 2009 और 10। और उन्होंने कहा या तो आप हमारे पास जॉब कर लें, 10 लाख सैलरी, 1800 सीसी गाड़ी और इस्लामाबाद में घर, और हम आपको पूरी फैक्ट्री स्टैब्लिश करके दे देंगे, आप प्रोड्यूस करें, आप यू वुड बी द इंचार्ज, और आप प्रोड्यूस करें और आप करें। दूसरी ऑप्शन है कि खुद ही करो सारा कुछ, प्रोडक्ट बनाकर हमें दो, हम इसको सेल करेंगे, तुम्हें पैसे देंगे, तुम अपने आप को डेवलप करो। तो मैंने कौन सा रास्ता चूज किया? सेकंड वाला। मैंने कहा नहीं, हम खुद बादशाह बनेंगे, हम खुद सीईओ बनेंगे, ठीक है ना। अब वो कंपनी कौन सी थी? आईसीआई पाकिस्तान, जो कि उस वक्त एक्सो नोबेल थी, और वो मल्टीनेशनल, जब तो लोकल होगी ना, लकी सीमेंट ने ले ली, उस वक्त वो एक्सो नोबेल के साथ थी, एंड दैट वाज मल्टीनेशनल। और मेरा काला सा कमरा था 10 बाय 12 फुट का, जिसपे एक बेड लगा हुआ था और एक छोटा सा टेबल, उस पे बैठ के वाइस प्रेसिडेंट ऑफ आईसीआई ने एग्रीमेंट साइन किया। उन्होंने कहा कि हमें सिर्फ आपके काम पे यकीन है कि आप जो कह रही हैं वो होगा। तो दैट वाज माय ग्रेटेस्ट सक्सेस। और वो एग्रीमेंट साइन हुआ, 10 साल हमने वो प्रोडक्ट चलाया, अभी भी हम वो चला रहे हैं। और पाकिस्तान का कोई कोना, बलूचिस्तान हो, सिंध हो, केपीके हो, आजाद कश्मीर हो, पंजाब हो, जहां मैं खुद नहीं गई फार्मर को ये इसकी प्रोडक्ट की जानकारी देने के लिए, हम डेरों पे, ट्यूबवेलों पे, खेतों पे, मतलब बदीन हो, ठट्ठा हो, वो कबल हो, सवात में मानसेरा हो, बुनेर हो, इधर हमारा बलूचिस्तान के इलाके हो, हर जगह खुशदार हो, मैं खुद गई, उमरकोट हो, और वहां पर जाकर हमने लोगों को ट्रायल कराया, लोगों को इंट्रोड्यूस कराया, 15-15, 20-20 फार्मर्स की गैदरिंग्स को इकट्ठा करके उनको प्रोडक्ट बताया। अल्हम्दुलिल्लाह हमारा वही प्रोडक्ट आज भी आईसीआई मार्केट कर रही। अच्छा, उसके बाद क्या हुआ? आज हम क्या कर रहे हैं? आज मेरे 6 पेटेंट्स हैं मेरी कंपनी के, जिसमें से 4 यूएसए में पेटेंट हुए हैं, 6 पाकिस्तान में पेटेंट, वही जो रिपीट भी हुए 4, वो तो 2 नेक्स्ट और पाकिस्तान में 2 पेटेंट्स इस तक हमारे यूके में सबमिट हुए हैं, जो कि यूके में हो जाएंगे। वो जो मैंने 50 स्ट्रेंस किए थे, आज वो 200 स्ट्रेंस हो चुके हैं। और हम हमारे प्राइवेट सेक्टर में हमसे ज्यादा बड़ी कल्चर कलेक्शन किसी के पास नहीं। उन 200 स्ट्रेंस में से तकरीबन 80 स्ट्रेंस जो हैं वो हमारे एक्सेशन नंबर्स के साथ हैं, आइडेंटिफाइड है। मैक्रोजन जो आप लोग जानते हैं उसको, वहां से सारे के सारे उनकी आइडेंटिफिकेशन, उनका जेनेटिक हुई हुई है। उसके बाद हमने सोचा, मेरे साथ कुछ पार्टनर्स मिल गए जो फर्टिलाइजर सेक्टर से थे। सर 2 मिनट बस मुझे देंगे, बस आई नो बस, लेकिन ये छोटी सी स्टोरी उनके लिए। वो पार्टनर्स मिले, ये सर आपके लिए स्पेशली है, और उन्होंने कहा कि देखो, एक फार्मर को तुम ये बैग देती हो बायो फर्टिलाइजर का, तो वो तकरीबन 1400-1500 रुपए खर्च करता है। ठीक है, उसको यील्ड इंप्रूवमेंट आती है, उसको बेनिफिट होता है, और उसको फर्टिलाइजर कॉस्ट में भी कमी होती है, लेकिन उसको लगता है कि वो डबल कर रहा है। हालांकि उसकी फर्टिलाइजर बच रही थी, 15 से 20%, 30% फर्टिलाइजर में बेनिफिट हो रहा था, उसकी केमिकल फर्टिलाइजर कम हो रही थी, लेकिन एक दिल में तो था ना कि वो भी खरीदते हैं, ये भी खरीदते हैं, यूरिया भी खरीदते हैं, वो भी खरीदते हैं, पहले उसका छट्टा लगाओ फिर उसका छट्टा लगाओ। तो क्यों ना हम एक ऐसा प्रोडक्ट डेवलप करें कि फर्टिलाइजर और बायो फर्टिलाइजर और केमिकल फर्टिलाइजर या यूरिया को आपस में मिला दें, कि फार्मर एक ही छट्टा लगाए, एक ही दफा वो अपनी प्रोडक्ट को यूज करे, तो उसको बैक्टीरिया भी पूरा मिल जाए, उसकी कॉस्ट ऑफ फर्टिलाइजर भी कम हो जाए, और उसको साथ साथ उसकी यील्ड भी बढ़ जाए। तो हमने इस पे सोचना शुरू किया, R&D शुरू की, और इस पे हमें 4 साल लगे। तो हमने एक प्रोडक्ट डेवलप किया जिसका नाम रखा जबरदस्त यूरिया। तकरीबन हमें R&D में 4 साल लगे, कॉन्सेप्ट को डेवलप करने में 3 साल लगे, तो तकरीबन ये कोई 7 साल का प्रोसेस था कि हम पैसा अंदर डालते गए, R&D करते रहे, और आखिर में जबरदस्त यूरिया निकला। अब जबरदस्त यूरिया क्या था? जबरदस्त यूरिया एक ऐसा यूरिया है कि जिसके ऊपर आपने जिंक को भी कोट कर दिया, आपने बैक्टीरिया भी कोट कर दिया। अब फार्मर को पहले जिंक डालने के, और इसमें अंदर जो जिंक है वो ऑर्गेनिक है, यूरिया वैसे ही ऑर्गेनिक होता है। फार्मर को पहले जिंक डालने के लिए तकरीबन 6000 रुपए खर्च करने पड़ते हैं पर एकड़, अगर वो 2 बैग डालता तो। यूरिया को की बोरी 5500 या 6000 की है, तो वो 12000 खर्च करता है, और फिर बैक्टीरिया के लिए 2000 अलहदा खर्च करता है, तो 14000 रुपए का पैकेज है फार्मर के लिए फॉर 1 एकड़। जबकि जबरदस्त यूरिया का बैग जो है, वो अगर यूरिया 6000 का है तो ये 6500 है, तो सिर्फ 500 रुपए में फार्मर को जिंक भी पूरा मिल जाता है उसको, और ऑर्गेनिक जिंक मिलता है, 100% ऑर्गेनिक, और बैक्टीरिया भी मिल जाते हैं, और साथ में यूरिया भी आ जाता है। और जहां उसको 2 बैग यूरिया के इस्तेमाल करने थे, वो 1.5 बैग यूरिया का, 1 बैग यूरिया का इस्तेमाल कर, जहां 1.5 करना था वहां 1 इस्तेमाल करता है, उसकी कॉस्ट भी कम हो जाती है। तो आपने एक सस्टेनेबल प्रोडक्ट बना दिया, जो फार्मर की हैबिट को उसने चेंज नहीं किया, उसने सिर्फ बोरी के साथ बोरी बदल ली, और आपने उसने कहा ये यूरिया इस्तेमाल की बजाय ये यूरिया इस्तेमाल कर लो, और कोई जिंक डालने की, 0 जिंक डालने की जरूरत है। और उसकी हमने सर टेस्टिंग की, वो ग्रेन में अपटेक बहुत ज्यादा हो गया यूरिया का, जिंक का, जिसको ये बिल गेट की ऑर्गेनाइजेशन ने भी उसको ओके किया, वो हार्वेस्ट प्लस ने उसको सिंध में रिकमेंडेशंस में डाल दिए। तो ये हमारे प्रोडक्ट्स हैं जो आपको स्क्रीन पे नजर आ रहे हैं, फर्टिवायर और जबरदस्त यूरिया सिंबल। इनके डिटेल्स में नहीं जाऊंगी कि ये क्या काम करते हैं, लेकिन अब तक के जो गवर्नमेंट के रिजल्ट हैं कि जबरदस्त यूरिया ने कहीं भी 10% से कम यील्ड डिफरेंस नहीं दी, हमेशा हायर यील्ड दी। फिर इसने रिडक्शन इन नाइट्रोजन अमोनिया एमिशन भी की, आपके क्लाइमेट चेंज के हवाले से, कि जब ये कोटेड हो गया और इसके ऊपर जिंक और बैक्टीरिया आ गए, तो उसने 22%, यहाँ पे हमने 3 साल इसका प्रोजेक्ट लगाया ऑन 4 क्रॉप्स, तो 22% अमोनिया एमिशन में कमी हुई, और नाइट्रेट लीचिंग में कमी हुई। तो आपकी जो क्लाइमेट में इफेक्ट आ रहा है फर्टिलाइजर का, केमिकल फर्टिलाइजर का, उसमें भी इसका फायदा। इसी तरह हम सल्फर कोटेड यूरिया और डीएपी कोटेड हाइब्रिड डीएपी भी मार्केट में ला चुके हैं, वो भी इस तरह मार्केट हो रही। जब हमने जबरदस्त यूरिया बनाया तो फिर एक और बड़ी कंपनी आई, उसने कहा ये तो जबरदस्त है, हम इसको ट्रायल लगाते हैं, अगर अच्छा निकला तो हम इस पे काम करेंगे। और वो कंपनी थी एग्रो फर्टिलाइजर। पाकिस्तान में एग्रो फर्टिलाइजर की 70 इयर्स हिस्ट्री में था कि उन्होंने पहली दफा किसी लोकल कंपनी के ऊपर ट्रस्ट किया और उसके प्रोडक्ट को मार्केट किया। और जब हम उनसे वो मिले तो हमें था कि ये इतने बड़े हाथी हैं, तो ये हमें कहेंगे टेक्नोलॉजी ट्रांसफर का, एंड वी वर रेडी फॉर दैट कि जी हम बिल्कुल, हम दिलो जान से हाजिर, आप लें टेक्नोलॉजी और काम करें। दे सेड नो, यू आर द साइंटिस्ट, आपने ये प्रोडक्ट बनाया है, आपको जो इसके बारे में जो मुसलसल R&D चाहिए, बायो फर्टिलाइजर का प्रॉब्लम ये है कि उसके पीछे एक मुसलसल R&D चाहिए, वो आप लोग कर सकते हैं। तो R&D और प्रोडक्शन अपने पास रखें, मार्केटिंग हमें दे दें, एंड दिस इज व्हाट दे आर डूइंग नाउ। तो वो मुझे उस दिन, जिस दिन उनके साथ मेरा एग्रीमेंट हुआ 2017 में, तो मुझे ऐसा लगा कि आज मुझे निशाने अदर मिल गए। तो ये वो स्टोरी है जो कि मैं हर स्टूडेंट्स के लिए कहती हूँ कि अपने मुल्क के लिए काम करें। और जब अपने, और उसके बाद हमारे पास ये ऑर्गेनिक कॉटन वाले आए, मैं बताती नहीं कौन है, हमारा प्रोडक्ट बाहर प्रोडक्ट है जो विद इन 2 days कंट्रोल यूनियन से ऑर्गेनिक डिक्लेयर हो जाएगा, इसी वीक में इंशाल्लाह हो जाएगा। अब सारी उसकी हो गई पूरी, मेरा कल या परसों में इसका सर्टिफिकेट मिल जाए। साथ साथ मेरी तो साथ साथ से हुई, 7 तारीख, ये 2 दिन में हमें पता चला कि मुझे अभी मैसेज आया, मैसेज बस लगे। सो ये मेरी फैक्ट्री है जो मैं दिखा रही हूँ। ऐसे यूरिया आता है, फिर वो कन्वर्ट होता है। मैंने इस तरह मशीनों से शुरू किया था एंग्रो के साथ। ये प्लांट था छोटा सा, ये मेरे डॉक्टर नदीम तारिक मेरे पार्टनर, और ये यहाँ तक पहुंच गया। ये इसकी जिस दिन उसकी इनॉग्रेशन थी तो एंग्रो के वाइस प्रेसिडेंट्स वगैरह आए हुए थे सारे। ये हमारे फर्मेंटर्स हैं जो हमने खुद डिजाइन किए। ये जो एक फर्मेंटर शायद आपको 50 लाख का मिलता है, हमने सारे जो है 28 फर्मेंटर्स जो है वो 40 लाख में बना दिए, और ये हमने खुद डिजाइन किए। तो इस तरह हमारा बैक्टीरिया जाता है और वहां पे फिर वो इनोकुलम यूज होता है। सारा प्लांट जो है वो ऑटोमेटिक है। This is our lab, and I must say that it is one of the finest microbiology lab in the private sector. कंपनी में ऐसे के 6 PhDs हैं इंक्लूडिंग मी, और कोई 12-13 MPhil और इंजीनियर्स हैं बाकी। इस वक्त हम, आप कहते हैं बायो फर्टिलाइजर की पाकिस्तान प्रोडक्शन की कितनी है, इस वक्त हम 44,000 बैग्स डेली प्रोड्यूस कर रहे हैं जबरदस्त यूरिया के, और तकरीबन 4 लाख टन पर ईयर हमारी प्रोडक्शन है, जो कि 7% ऑफ द मार्केट शेयर बनता है यूरिया में। टेक होम मैसेज, जब आप जाते हैं रहीमिया खान और इर्द गिर्द के इलाकों में, तो जब फार्मर आपका बैग लेके जा रहा होता है, तो दिस इज माई इम्पैक्ट फैक्टर, एंड दिस इज अ ट्रिब्यूट टू माई टीचर्स, डॉक्टर फौजी यूसुफ, डॉक्टर सहेल हमीद। उनसे मैंने सीखा, इस मुल्क से ही हासिल किया, इस मुल्क को वापस करते।
Thank you so much, ma'am, Dr. Farooq, for your valuable presentation. Now, I would like to invite our next speaker, Mr. Asif Mahmood, and he will be presenting the topic organic cotton production. May I request him to kindly come on stage to deliver his topic.
स्लाइडें तो है, आप तो एजाज में बता दो सर। मैं फिर मैं तो कह रहा है मेरी आवाज आ रही पीछे से? वो आखिरी साथी को अच्छा जी। हाँ इसको अच्छा मैं अपना तारुफ करा दूं 5 मिनट में, ये तो हो सकता है ना। मेरा आसिफ महमूद नाम है, एग्रीकल्चर यूनिवर्सिटी फैसलाबाद से मैं एमफिल किया है मैंने प्लांट ब्रीडिंग एंड जेनेटिक्स में, और प्रोफेशनली मेरा सारा एक्सपीरियंस सस्टेनेबिलिटी में है। मैंने WWF पाकिस्तान को सर्व किया है, BCI में सर्व किया है, ओका में सर्व किया है। अब मैं सुरती का जो एग्रीकल्चर का शोबा है उसको हेड कर रहा हूँ। कोई साथी जिसको ने सुरती एंटरप्राइजेस का नाम ना सुना हो हाथ खड़ा करें, ताकि मेरे ना सुना हो, ताकि मैं सुरती का भी मुझे फिर पता लग जाए, बताना पड़े। सुरती एंटरप्राइज कराची में अभी रिसेंटली मूव हुआ हूँ, 1 साल हुआ है, तो मुझे अल्हम्दुलिल्लाह किसी भी जगह पे कोई एक बंदा ऐसा नहीं मिला, सड़क पे, मार्केट में, मोहल्ले में, कि जिसको मैंने बताया कि मैं सुरती में जॉब करता हूँ तो वो कहे कि ये क्या है। तो कराची में एटलीस्ट मेजॉरिटी पीपल आर फेमिलियर कि ये सुरती एंटरप्राइज डेनिम एक्सपोर्टर्स में लीडिंग एक ग्रुप है जो डेनिम जीन्स बनाते हैं, कपड़ा भी बनाते हैं, दोनों को एक्सपोर्ट करते हैं। सारा मटेरियल इनका एक्सपोर्ट क्वालिटी के लिए बनता है। मैं प्रेजेंटेशन सारी जो है वो एग्रीकल्चर को ध्यान में रख के बनाई थी, इसमें मैंने डेनिम का कंपोनेंट बिल्कुल भी। समरी स्लाइड के ऊपर हम जो प्रोजेक्ट्स फील्ड में चला रहे हैं, खुशदार में, लसबेला में, भावलपुर जो प्रोजेक्ट था, अब हमने इस साल से दादू शिफ्ट किया है, वो सिलेक्टेड स्लाइड दिखाते हैं, ये स्लाइड दिखाते हैं। तो ये लोकेशंस हैं जिन पे हम मौजूद हैं। कराची हमारा हेड ऑफिस है, साथ हम एक R&D स्टेशन चला रहे हैं, वो भी कराची में लांडी के अंदर है। मैं साथ साथ ऑफर्स ऐड कर देता हूँ काम की बात के अलावा। तो जो अगर जिस तरह ये सेशन आज हुआ है, थैंक्स टू डॉक्टर साहब के जिन्होंने ICCBS यहाँ में इनवाइट किया। वी वुड लव टू इनवाइट कि आप हमारे R&D स्टेशन पे यहाँ से स्टूडेंट्स का ग्रुप लेके आए, हम आपको R&D स्टेशन पे वेलकम करेंगे वहाँ। तो फील्ड प्रोजेक्ट में विजिट करना चाहे तो हम आपको दादू का और लसबेला का, तो खासतौर इन 2 जगहों का, तो हम एज अ ग्रुप फार्मर्स के साथ भी आपको फार्म लेवल की इंटरवेंशंस डायरेक्टली भी दिखा सकते हैं क्या कर रहा है। खुशदार लसबेला प्रोजेक्ट ऑर्गेनिक कॉटन के हैं, और दादू बहावलपुर प्रोजेक्ट जो है पहले 25 में बहावलपुर था, इस साल से हम दादू में कर रहे हैं, वो रीजन एग्री सर्टिफिकेशन के ऊपर है। ये एक बात मैं करना चाह रहा था। और तीनों प्रोजेक्टों से 6000 मैट्रिक टन लेंथ जो है वो सस्टेनेबल प्रोडक्ट जो है फार्म से आती है, जो सूरती से होके इंटरनेशनल ब्रांड्स को ही सर्व कर दी जाती है। ओका के साथ हमारी पार्टनरशिप है, सीड कमर्शलाइजेशन की यहाँ बात हुई है, उसमें भी हम द ओनली टेक्सटाइल ग्रुप जो कि एज अ स्टेक होल्डर काम कर रहे हैं सीड प्योरिटी के ऊपर। इसके अलावा इम्पैक्ट जो है, हमने जो चीज़ें इम्प्रूव की हैं, हमने फार्म लेवल पे कैलकुलेट की कि पीएच को हम थोड़ा सा फार्म लेवल पे नीचे लेके आए। इनकम फार्मर की इन प्रोजेक्ट्स के थ्रू अप टू 25 to 30% तक इम्प्रूव हुई है। पोस्ट लंच सेशन वैसे ही थोड़ा सा लेजी थोड़ा सा होता है कंपैरेटिवली, तो टेक्निकल बातें कम हो तो अच्छा है। ये स्लाइड में इसीलिए ओका के साथ हमारी जो कमिटमेंट्स हैं FCA के लिहाज से वो चल रही। ये एक समरी स्लाइड है हमारे तीनों प्रोजेक्ट्स की, कि जिसमें ऑर्गेनिक का प्रोजेक्ट है, रीजन एग्री का प्रोजेक्ट है, प्रोजेक्ट की सब डिवीजन। हम एक लफ्ज अपने प्रोजेक्ट्स में यूज करते हैं प्रोजेक्ट स्ट्रक्चर का, कि फार्मर कितने हैं, एरिया कितना है, प्रोड्यूस कितनी है, और बेल्स कितनी आएंगी। तो ये 3 चीजें रेगुलरली हमारे प्रोजेक्ट की पहला डॉक्यूमेंट हम ये तैयार करते हैं। और आगे चलते हैं। ये मेरा डिपार्टमेंट है, इस वक्त सुरती के अंदर मेरा डिपार्टमेंट कॉटन क्लब कहलाता है। सुरती को आए तो आप पूछें कि कॉटन क्लब के आसिम महमूद साहब से मिलना है, तो लांडी यूनिट 11 में आप आ सकते हैं, मैं वहाँ बेस करता हूँ, ताकि 5 मिनट के बाद की मुलाकात तफसीली वहाँ हो सकती। स्लाइडें मैं गुजारूंगा सारी, लेकिन तेजी से। ये हमारा इम्पैक्ट है। देखें 2 चीजें मैं समरी वाइज, पहले तो मैं आई वांट टू एक्नॉलेज सेशन के दौरान ही पहले साकिब भाई की मैंने बातें सुनी, समझी, उनसे सीखा। फिर डॉक्टर साहब जो वर्चुअल थे, उनकी बातों में बहुत मोटिवेशन थी। मैडम इज हाईली इंस्पायरिंग फॉर अस। मैं आई थिंक वी ऑल नीड टू क्लैप फॉर मैडम वर्क। इंस्पायरिंग स्टोरीज को हमें देखें, पाकिस्तान का पॉजिटिव चेहरा हम ही लोग हैं, जो यहाँ से हमें आगे पास ऑन करना चाहिए। 2 चीजें होती हैं, प्रोडक्ट होती है और स्टोरी होती है, दोनों पे काम करना होता है। सिर्फ प्रोडक्ट भी ठीक नहीं है, सिर्फ स्टोरी भी ठीक नहीं। प्रोडक्ट आपने प्रोडक्ट भी बनानी है, प्रोडक्ट सेल भी करनी है, और उसकी स्टोरी को भी सेल करना। तो स्टोरी, वो कहते हैं ना, साथ ढोल भी उतना ही, सिर्फ ढोल भी नहीं बजाना और सिर्फ आपने
साथ साथ दोनों चीजें लेके चलते रहे। ये हमारी स्टोरीज हैं जो हमने ग्लोबल प्लेटफॉर्म्स पे प्रेजेंट हुई हैं। इन फार्मर ने नेशनल प्लेटफॉर्म पे आके खुद प्रेजेंट भी किया है कि हमने ऑर्गेनिक कॉटन में ट्रेनिंग ली। अच्छा, हमारे पार्टनर, हमारे साथ फार्म लेवल पे हम थ्रू पार्टनर WWF का अभी लोगो आया, तो गुजार प्रोजेक्ट हम WWF पाकिस्तान के थ्रू कर रहे हैं, लसबेला प्रोजेक्ट एग्रीसर इम्पैक्ट के थ्रू कर रहे हैं, और जो दादू प्रोजेक्ट है वो रीड्स पाकिस्तान के साथ कर रहे हैं। डिजिटल जो ट्रेसिबिलिटी है उसमें जो काम है उसमें सावी इकोसिस्टम के साथ काम कर रहे हैं। तो ये फार्मर है जिन्होंने हमारी ट्रेनिंग के बाद इतना पानी और इतने प्रॉफिट्स अर्न किए। मैं एक फार्मर नहीं जा सकता था, तो उससे टेक्सटाइल एक्सचेंज कॉन्फ्रेंस हुई यूके में, तो मौके पे उन्होंने कहा जी फार्मर के पे आप प्रेजेंट करें। तो ये असलम की स्टोरी तो मैंने यूके में बैठ के पाकिस्तान के हवाले से खुद सुनाई जाके। 850 फार्मर से शुरू हुए थे, आज हम 4000 से ज्यादा फार्मर के साथ काम कर रहे हैं। प्रीमियम ऑर्गेनिक बेसिकली सॉइल को बेहतर करता है, साथ में एंड पे फार्मर को, देखिए फार्मर बहुत ही सिंपल और देसी आदमी, उसको ये याद रहता है कि कोई मुझे ट्रेनिंग दे रहा है कि नहीं, और एंड पे मुझे कुछ प्रीमियम मिल रहा है कि नहीं। तो वो प्रीमियम ऑर्गेनिक का फार्मर के में जो पर्सपेक्टिव है, ये ऐसी प्रोडक्ट है जिसका मुझे प्रीमियम जरूर मिलेगा। ये एक चीज है जो मैंने फार्मर से डायरेक्टली मोटिवेशन, अदर स्टैंडर्ड्स के मुकाबले में ऑर्गेनिक का एक प्लस पॉइंट जो है। नॉन जीएम सीड, हम प्योरिटी के ऊपर जो काम किया क्विकली बता देता हूँ कि हमने 2000 वो स्पेसिफिक स्लाइड, ये स्लाइड में दिखाता हूँ कि जो हमने ऑर्गेनिक इंटेग्रिटी पे काम किया कि नॉन जीएम सीड के हवाले से हम स्टार्ट हुए 2023 में। हमने साथ में ऐड कर दूंगा कराची यूनिवर्सिटी के साथ और ICCBS के साथ जो काम किया अब तक। तो 2023 के स्टार्ट में डॉक्टर शाहिद मंसूर साहब की रहनुमाई में हमने ये सिंगल प्लांट सिलेक्शन से स्टार्ट लिया, और हम जो 85% कंटेमिनेशन से हम लेस देन 5% कंटेमिनेशन तक पहुंचे। ओका सीड कमर्शलाइजेशन के प्रोजेक्ट का हम पार्ट है। सीड मल्टीप्लिकेशन प्लॉट हम अपने प्रोजेक्ट्स में चला रहे हैं, जहां हम अपना सीड जो है 100% फार्मर्स को रिसाइकिल करके दे रहे हैं, ताकि एक्सटर्नल डिपेंडेंसी भी ना रहे और प्योरिटी का मसला भी पेश ना आए। ICCBS के साथ कोलैबोरेशन रही रिसर्च ट्रायल्स के ऊपर लास्ट ईयर। बायो इनपुट्स के ऊपर हम मैंने बड़ी तवज्जो से मैडम की सुनी, और आखिरी बात हमारे हमसे रिलेटेड थी, वो और तवज्जो से सुनी कि 2 दिन बाद वो सर्टिफाइड प्रोडक्ट आ जाएगा, तो हम इसके ऊपर ट्रायल्स फौरन करना चाहेंगे, ताकि हमें ऑर्गेनिक इंटेग्रिटी में नॉन जीएम सीड भी यहाँ पे अवेलेबल नहीं था जब ऑर्गेनिक स्टार्ट हुआ उससे पहले, तो ऑर्गेनिक की वसीले से हमें वापस नॉन BT सीड पे दोबारा काम करना पड़ा। ये भी हमारे लिए एजाज की बात है कि हम ICCBS के अंदर खड़े होकर नॉन जीएम सीड की खुल के बात कर रहे हैं। बायो इनपुट्स के हवाले से चीजें हम मुख्तलिफ इदारों को एक्सप्लोर कर रहे हैं कि कोई ऐसी चीज मिले जो हमें फार्मर के लिए अफोर्डेबल भी हो, और एट द एंड ऑफ द डे ऑर्गेनाइजेशन भी क्रेडिबल हो, और हमारे लिए इजी टू अप्रोच भी हो। ICCBS के साथ बायो पेस्टिसाइड की भी हमारी बात चल रही है, जो हम रेगुलर इनके साथ काम कर रहे हैं। बेस्ट प्रैक्टिसेस है। डिजिटल ट्रेसिबिलिटी जो बात हुई, 2 बातें यहाँ पे हुई, एक सस्टेनेबिलिटी की बात हुई, दूसरी चीज है ट्रेसिबिलिटी। हमारा सेक्टर फार्म लेवल सस्टेनेबिलिटी और बैक टू फार्म ट्रेसिबिलिटी पे काम कर कि जी प्रोडक्ट अगर शर्ट है तो उसका खेत तक पहुंचाया जाए, जो इंटरनेशनल मार्केट हमसे एक्सपेक्ट कर। उस वाले से भी हम डिजिटल ट्रेसिबिलिटी के प्रोजेक्ट कर रहे हैं, जिसमें हमारा सावी के साथ कोलैबोरेशन है, और हम कोशिश है कि हमारे तमाम प्रोजेक्ट्स 100% ट्रेसेबल हों। आप फिजिकली भी प्रोजेक्ट देख सकें, फार्मर से वेरीफाई भी कर सकें, और सिस्टम पे भी आपको एक्सेस मिले डैशबोर्ड की फॉर्म के अंदर। जी, हम लास्ट बट नॉट द लीस्ट के 2000 हेक्टेयर से हम शुरू हुए थे फर्स्ट ईयर। आज हम अल्हम्दुलिल्लाह 13,000 हेक्टेयर पर पहुंचे हैं, और हमारी कोशिश है कि हम इसको मजीद अल्टरनेट तमाम सस्टेनेबिलिटी स्टैंडर्ड्स और अल्टरनेट फाइबर्स की तरफ भी ले जाएं। और एट द एंड ऑफ द सर, आखिरी बात सर, कि हमें वी आर ऑल्सो ओपन फॉर द इंटर्नशिप्स एज वेल। तो आफ्टर द सेशन वी विल गेट बैक टू यू सर फॉर एक्सप्लोरिंग द ऑप्शन के हमें 2 से 3 रेगुलर इंटर्नीज यहाँ चाहिए होंगे, तो उसमें हम चाहेंगे कि ICCBS से, कराची यूनिवर्सिटी से हमें रिकमेंडेशन आए, ताकि हम एक रेगुलर इंटर्नशिप प्रोग्राम साथ साथ लेके चल सकें। तो इस पर हम वापसी आपके साथ कॉल करें। थैंक यू।
Thank you so much, sir, for your valuable speech. Now, I would like to invite our next speaker, Mr. Niaz Mahmood Nizamani. Niaz Muhammad Nizamani, and he will be delivering the topic, and I would like to invite him on the stage.
बड़ा क्विक सा इंट्रोडक्शन नियाज साहब के बारे में. नियाज साहब इज अ फार्मर, इज अ एंटरप्रेन्योर, और जो इनकी बातें इतनी साइंटिफिक होती हैं कि ही हैज ग्रिप ऑन ऑल द मॉडर्न टेक्नोलॉजीज, एंड जो भी रिसर्च हो रही है इन्हें क्रिस्पर का भी पता होता है, और बिल्कुल पूछ रहे होते हैं ये होता क्या है. तो आई एम वेरी इम्प्रेस्ड एंड वेरी डिलाइटेड टू इनवाइट हिम फॉर his talk about, so he's producing biofertilizer as well as biopesticides. ऐसा ठीक है.
सबसे पहले तो मैं डॉक्टर साहब ने बता दिया कि मैं एक फार्मर हूँ, साइंटिस्ट नहीं हूँ, और फार्मिंग जो है ना वो स्पेशली बाय डिग्री तो इंजीनियरिंग, लेकिन वो सारी जिंदगी जो है ना वो फार्मिंग ही किया. वैसे डॉक्टर साहब ने जो है ना वो पहले दफ़ा मुझे जो भेजा था वो सिम्पोजियम की इनविटेशन, उसमें तो नाम था और प्रेजेंटेशन का कहा था, लेकिन बाद वाले डॉक्यूमेंट में नहीं था, तो इसलिए तो मैंने न प्रेजेंटेशन बनाई, न मैं इस जहन से आया था कि मुझे बोलना पड़ेगा. लेकिन ये अचानक की कॉल है, अब तो कुछ नहीं पता कि आपसे क्या कहना है मुझे. लेकिन मैंने सारी जिंदगी फार्मिंग की है. तो सबसे अहम बात यह है कि ये एक सिस्टम है, जो एक फ्रेमवर्क है. इसकी मिसाल मैं आपको बताऊं कि वो बताते हैं ना कि अंधों को जब हाथी दिखाया गया, तो जिसने वो उसको उसकी टांग को हाथ लगाया तो उसने कहा ये तो दरख्त जैसा है हाथी. और किसी ने जो है ना वो पेट को हाथ लगाया तो उसने कहा या तो ये दीवार जैसा है या पहाड़ जैसा है. जिसने वो सूंड को हाथ लगाया उसने कहा या तो ये तो पाइप जैसा है या कोई हसदे जैसा है. तो ये एग्रीकल्चर जो है वो एक एक सिस्टम है, और इस सिस्टम के अंदर बहुत सारी चीजें हैं. मैं सबसे पहले आपको बताऊं कि जो सॉइल हाइड्रोलॉजी है, यानी कि जमीन में पानी किस तरह जाता है, किस तरह चलता है, ये बहुत बड़ा मैटर रखता है. अगर जमीन से नीचे जड़ों तक पानी आ गया तो सारी हमारी माइक्रोबायोलॉजी मर जाएगी, कुछ नहीं बचेगा. तो हमारे सिंधी में एक कहावत है, वो बहुत पुरानी कहावत है, कि वो कहते हैं ना कि सिंधी में आपको बताता हूँ कि हारी हो जे अंधो तो वनी जरे पुराण जो कंधो. हमारे यहाँ ऐसी जमीनें हैं जो दरिया के ये समझे कि पुराने जो दरिया थे, जो मेरी अपनी जाती जमीन है मैं उसका बताता हूँ ना, उसके मेरी जाती जमीन के एक नहर के साथ है. तो नहर का सीपेज है, बीच में जो है ना वो पुराना दरिया है. तो वो मुझे जब मैंने वो कल्टीवेशन, जब वो सेंटर में दरिया था वहां शुरू की, तो ऊपर वाली जमीनें मेरी मुतासिर होने लगी. तो फिर जो है वो मैंने जो नहर के साथ भी दरख्त लगाए, बीच में भी दरख्त लगाए, तो मेरी जमीनें जो थी वो ऊपर वाले हिस्से की बहुत ज्यादा जो है ना वो फर्टाइल हो गई. तो कहने का मकसद ये है कि बहुत इम्पोर्टेंट फैक्टर है सॉइल हाइड्रोलॉजी का. दो हमारे बड़े प्रोजेक्ट सिंध में शुरू हुए थे, लेकिन वो नाकाम हो गए. या कभी ना कभी गवर्नमेंट को करने पड़ेंगे LBOD और RBOD, क्योंकि वो सॉइल की जो फर्टिलिटी है और सॉइल की जो हेल्थ है, इस वक्त दुनिया में सबसे ज्यादा जो तवज्जो तलब चीज है वो ये है. जो लोग जो इस फील्ड के अंदर है वो ये बता रहे हैं कि सॉइल की फर्टिलिटी कॉम्प्रोमाइज हो रही है, और आइंदा बता रहे हैं कि 2050 तक हमारी जो सॉइल्स है वो 40% यील्ड कम देंगी, और हमारी पॉपुलेशन 9.2 बिलियन होगी. तो उसके लिए हमें सोचना है. तो ये है कि यूरोप ने भी मुख्तलिफ किस्म के प्रोग्राम शुरू किए हैं, जैसे कि सॉइल लिविंग सॉइल के नाम से भी उन्होंने एक प्रोग्राम शुरू किया है. सो इसी तरह जो है ना वो सॉइल हेल्थ, सॉइल फर्टिलिटी एक बहुत बड़ा टॉपिक है. जिसपे इंस्टीट्यूट को, जैसे ये आपका अहम इंस्टीट्यूट है, कि इसको हम किस तरह, जिसमें माइक्रोबायोलॉजी है, ऑर्गेनिक मैटर है, सॉइल पीएच है, ईसी है, कंडक्टिविटी है, बहुत सारे फैक्टर्स हैं जो सारे मिलके काम करते हैं। और मैं स्पेशली बनाना का फार्मर हूँ, और बनाना इस वक्त जो है वो एक बड़ी क्राइसिस से गुजर रहा है, जिसमें एक बीमारी आई हुई है, फंगल डिजीज है, फ्यूजेरियम ऑक्सीस्पोरम। तो वो एक किस्म का एक ये समझे कि एक कुदरती आफत है बनाना पे। हाँ, मैं उस पे स्पेशली काम कर रहा हूँ कि उस पे से हम जो है ना वो किस तरह जो है वो अपनी क्रॉप को बचाएंगे, जो इस वक्त तबाही के किनारे पे खड़ा हुआ है। उसमें दो चीजें अहम हैं जिन पे मैं काम कर रहा हूँ। एक तो वो वेराइटीज जो रेजिस्टेंट है या टॉलरेंट है, और दूसरा उसकी जो है ना वो माइक्रोबायोलॉजी है। माइक्रोबायोलॉजी का पावर तो आप सब लोगों ने देख लिया होगा कि माइक्रोबायोलॉजी कितनी बड़ी पावरफुल चीज है। जो कोविड है, सबको पता है कि कोविड क्या चीज थी। तो इसी तरीके से ये जो माइक्रोबियल लाइफ है, एक बहुत बड़ा पावर है, जो जब से मैंने इसको देखा है कि मुझे लगता है कि जैसे वो आसमान के तारे हैं, तो जमीन के अंदर भी जो है एक बहुत बड़ी मखलूक है। हैरान हो जाते हैं फार्मर्स जब हम उनको जो है ना वो एक मिट्टी का जो है ना वो मिट्टी उठा के पानी में डाल के माइक्रोस्कोप के नीचे दिखाते हैं कि देखें ये एक बहुत बड़ी कायनात है, जो जो वो जो है हमारे लिए काम कर रही होती है। अच्छा, दूसरी बात यह है कि हम एक मेरी ऑब्जर्वेशन है, मैं बनाना के हवाले से, डॉक्टर साहब भी बैठी है, सब भी सोच सकते हैं कि बनाना में क्या हुआ है कि कुछ ऐसे फील्ड्स मैंने आइडेंटिफाई किए हैं जहाँ पे इन्होंने ना हाई यील्ड के लिए वाटर सॉल्युबल फर्टिलाइजर इस्तेमाल किए थे। मेरा ये ख्याल होता है कि अगर ये इस्तेमाल किए गए हैं, शायद इन्होंने जो है ना वो सॉइल की माइक्रोबायोलॉजी को बहुत ज्यादा डैमेज किया है, और उसके नतीजे में इनके पास डिजीज ज्यादा आई है। तो इंतहाई जो है ना वो खतरनाक सूरत हाल बनाना क्रॉप्स के लिए ये डेवलप हो गई है, और सिंध में सबसे ज्यादा लुक्रेटिव क्रॉप है। तो हमने जो है ना वो उस पे कुछ ऐसे माइक्रोब्स और जो है ना और फंगस इस्तेमाल किए, उसके बहुत बड़े अच्छे रिजल्ट्स हैं। लेकिन क्योंकि इसकी एप्लीकेशन, इसको समझाना फार्मर को जो है ना वो एक बहुत मुश्किल है। डॉक्टर साहब को पता होगा, क्योंकि ये टेक्नोलॉजी बेची नहीं जाती, ये अडॉप्ट करवाई जाती है कि किस तरह इसको फार्मर अडॉप्ट करे, समझे कि भाई किस तरह हम इसको इस्तेमाल करें। उसमें कुछ स्ट्रेन्स बैक्टीरिया के हैं, कुछ फंगस के हैं, जिन्होंने बहुत बड़े अच्छे रिजल्ट दिए हैं। बल्कि अभी मैं रिसेंटली मैंने चाइना का विजिट किया था, तो उनके पास उन्होंने कुछ ऐसे स्ट्रेन्स आइडेंटिफाई किए हैं। तो डॉक्टर साहब की भी मैं इस तरफ तवज्जो दिलवाऊंगा। वो जैसे कि हमारे जो भी स्ट्रेन्स है वो सिस्टमेटिक नहीं है, वो बनाना के अंदर जाके उसको डिफेंड नहीं करते। जो फंगस है ऑक्सीस्पोर, वो सिस्टमेटिक है, वो रूट के अंदर, प्लांट के अंदर चला जाता है। तो उन्होंने एक रबड़ प्लांट से आइसोलेट किया है स्ट्रेन, उसका नाम है सरेशा। तो वो प्लांट के अंदर जाके उसको डिफेंड करता है। तो ये हो सकता है कि डॉक्टर साहब को मैं गुजारिश करूंगा कि वो चाइना से जो है ना वो उनके पास अवेलेबल है उनकी, और वो इंस्टीट्यूट को दे सकते हैं रिसर्च पर्पज के लिए। तो काइंडली उसके लिए आप कोशिश करके जो है ना वो हमें मंगवा के दें। तो मैं ये डॉक्टर साहब को कहूँगा कि हमारा किसी तरीके से निपजी से कोई ऐसे किस्म का कोई एमओयू साइन करवा दें, जिसमें हमारे ऊपर जो है ना वो किसी इंस्टीट्यूट का जो है ना वो हाथ आ जाए। जज़ाकल्लाह, थैंक यू।
Thank you so much, sir, for your valuable presentation. Now we will move towards our next speaker, who is Dr. Khalid Mahmood, and he will join us from UK via online. And the topic will be knowledge-driven farming by Swipe platform. I would like to request him to kindly deliver the topic, sir. Thank you.
Ji, Assalamu alaikum. First of all, thank you very much. Congratulations, Dr. Shah Saab and ICCBS for hosting a wonderful conference event. Thank you very much. I think आज की जो discussion थी, maybe let me unblur myself. There's something coming in the back. Okay, easy. So, my background is I'm agriculture. I would say specialist, a bit of learner. हमारे जो पहले अभी जो स्पीकर थे उन्होंने सॉइल की बात की तो I also wear another hat. I'm involved with in the UK with the British Society of Soil Science, and that was established around the same time when Pakistan was born, so 1947. So that's where the history goes back to in this kind of research. And previously, I was working in a bigger research institute called Rothamsted, and Rothamsted has a experiment on soil and plant back in that started back in 1843. So that's where the history goes. And for science, you need to have the data. So before you reach any evidence, you need to have the data. So, and I think my colleagues said already as well, there's no one size that could fit for everything. So not one size fit for all. So the challenge agriculture is facing the climate change. The Pakistan where we are facing the multiple issues, water availability, climate change is linked with the water availability, heat and drought. Heat is and high humidity causing pests and diseases. And on the top, growing cotton and that condition is becoming a challenge. And my colleague Saqib Saab said here, Pakistan, that if you are producing cotton, which is just a commodity, there is no fancy bit about it. So we're missing the whole trick around the world. We cannot produce cheaper cotton than other countries. We can produce some good stories around the cotton. And this is where Savi been working. I have a short slide deck. If you allow me, I'll share that one. And I will not go in more details about the technology, but just where we try to see a value for Pakistan's cotton in terms of traceability, some stories, the communities they are involved, and how we portray to the world. So that's a challenge. We don't have an answer for that one yet, but we've been working on this concept. I think my colleague Asif Saab mentioned it. Traceability is one buzzword, I would say, but traceability is not the fancy bit. The fancy bit or the important bit is the livelihood of the farmers. Are we able to increase the income of our farmers? My colleague just mentioned earlier as well. So the income of the farmers is the critical. And at the end, cotton farmer has a freedom to operate. If the crop is not making money, he could move to maybe growing sugarcane or growing maize, which is not bad. But in terms of what is the best use of that water and the environment, so Allah has blessed us the region which has the ability to grow cotton. I was looking at some documentary, maybe some kind of a story from our neighboring country. So when they said when Pakistan was born, the whole cotton area moved to Pakistan, and they don't have the cotton area. And they worked really hard, and then they developed those three states, and they are growing cotton, and they are one of those three, second or third largest producer. I think it may be the second largest producer in the world. And also in terms of their access to market, globally, anything that comes out of that country are linked with that country. They have got those associations and relationships. And I think this is where Pakistan is missing. I always say that we need to have those partnerships. We need to have those relationships with the world. But I'll just go back to the farmers where we start our journey. So how do we get our farmers, strengthen our farmer with the knowledge? It's again हदीस शरीफ एज वेल कि इल्म हासिल करें चाहे आपको चाइना क्यों ना जाना पर उसको चाइना बहुत दूर था तो इसलिए इसके बारे में चाइना के बारे में कहा गया तो द पॉइंट आई एम सेइंग इज कि हाउ डू वी get our farmers to understand the myth and the challenge of producing cotton which is sustainable. And for that purpose, we developed a understanding, and still we are learning. We haven't finished yet. So everything starts with the soil. So understanding of the soil, and my colleague mentioned hydrology, soil nutrients, soil biology. So in the past, we were all only to mention about soil fertility, the living part of soil, which is the soil biology that was missed. So if you take a spoon of a teaspoon of soil, it has got more bacteria than humans on this living planet.
So that's where the diversity is. And this is where understanding of the soil is important. We had tried to develop some mechanism, doing maybe field testing a bit, but doing lab test, that's next level. Then we work with the farmers in terms of understanding of which varieties would work in that area. Of course, for organic cotton, there's a limit with I would say single gene varieties, which is grown through this I would say traditional mechanism. So we need to have a maybe twofold system in Pakistan. So, and again, this is where I don't know if you have a colleagues here from Balochistan. So some region we need to isolate. So make them this is a maybe organic cotton region. So the contamination issue is resolved. And then we do a lot of work around weather forecast because again that's a very important as well. So time of sowing is very important, and understanding the diseases as well. Rather than using heavy loaded pesticides, I won't name a big brand who we were just talking together in Europe, and they said they will not buy cotton which is using glyphosate. So glyphosate is used on killing our weeds, pendimethalin, some of other pesticides which are banned in the EU. And this was a bit of a positive note for me from that brand discussion. Look, they are not keen to source cotton from the US because the US is using a lot of chemicals. And our other colleague, I will not mention the other big group in Pakistan who work on the cotton, and they try to promote Brazil as a lord of the cotton. Brazil is killing the environment. So these brands have categorically said they will not source cotton from Brazil because Brazil is using pesticides, Brazil is using everything, and also devastation in the putting the forest down. And I think this is where the opportunity for Pakistan is. So developing a cotton, developing a brand. My colleague said here earlier, the Pima cotton. And so we've been working on a program to, I won't name it maybe at this stage, we still work in process. So this is where how we improve the quality of the cotton, the fiber length, the trash contents. There's a huge issue with our trash content as well. Sometimes it goes up to 10%, 7 to 8%. And the good quality, bad quality cotton is mixed and then sold. And this is where so we've been trying to develop a mechanism. So just as maybe first going on the farm level, so we advise the farmer, you can choose the system which you want. So maybe traditional, organic, region. So we try to help the farmers to develop some biopesticide on the farm. Biopesticide is a hot topic, and bio fertilizers, farm composting, and also we're trying to use the farmers the cotton sticks to make biochar. So this could be an additional income that we can bring to the farmers. So I think my colleague mentioned here Rob from the US. So farmers not only growing the crop, but if they are helping, because on average one cotton plant it's sequester carbon, it's bringing carbon from the atmosphere to the soil. Secondly, it's if you use the stick of the cotton, the waste which is typically used for cooking, and if we can convert them into biochar is a material that's uh, it's like in other words में कह लें कि आपका कोयला है, ठीक है, जो फसल से पैदा करते हैं. So that koila has ability to absorb carbon for million of years, but it also helps to improve the soil I would say biology. सो सरफेस एरिया इंक्रीज हो जाता है, वाटर होल्डिंग कैपेसिटी बढ़ जाती है, ठीक है जी. तो इसके अलावा जो उसके जो एरेशन और अदर बेनिफिट्स हैं वो बढ़ जाते हैं, ठीक है जी. तो इससे जो है एंड उसकी वजह से फार्मर विल अर्न कार्बन क्रेडिट्स, और वी आर वर्किंग ऑन दैट वन एज वेल. सो and then my colleague mentioned cotton uses a lot of water, so how we can use some technology. Of course, technology is expensive, but there could be a way to bring this knowledge to the farmers. And this way, we're using satellite data, machine learning, AI, and also bringing on-farm sensors as well. These we developed together with LUMS in Pakistan. And so for doing everything, we package this thing together. So the farmer's soil health, use of pesticides, even the farmer's income, maybe livestock, so everything is packaged here, and that's come into this data. We call it Savi Trace. So Savi Trace has the management data. So crop using satellite, advisory alerts, AI data from there. I know farmers don't have tools to write down, so we can abstract some data using AI, maybe through some phone, but some information we are using AI and satellite as well. So, but the fiber composition, this would be the physical data that will come from that, I would say, ginning factory. Carbon impact data, soil water data, social and economic data as well. And then everything is our idea, which is what we're trying to bring. And this is a project we're doing with Surti as well, having a QR code on the bale. that you can scan. And my colleague here, Sohail Saab, mentioned the digital product passport. EU is now going to implement in 18 months' time. It's already been announced. So this would give Pakistan's cotton that credibility. So this cotton is grown on an area which is sustainably agriculture being done. It's not grown on area where there was forest. We have cut forest. We're using excessive nutrients or pesticide. So I think this is the story we can tell to the world. So, so how we do it. So then there's a whole technology side. I will not go into the fancy bit of the technology because technology could be evolved. But this is where the whole idea is the data we should segregate the cotton, look after the cotton, don't mix it up. We see when the cotton picking season is there, it's just sitting around the roads. Usme plastic, usme human hairs, everything is going there. We are not protecting its uski quality. तो फिर ये एक विल प्रोवाइड डैशबोर्ड जो कि बाहर के पास भी होगा, तो इंटरनेशनल बायर भी देख सकेगा कि उसकी फसल वहाँ तैयार हो रही है, नहीं हो रही, किस स्टेज पे है, तो रियल टाइम डेटा इसके अंदर होगा. So how we work, so we have a managed models where we help and implement project ourselves as well. We have a little capacity at the moment. We won't be able to do it everywhere in Pakistan, but in the longer run, our interest is hand over this license this. Second model is our platform could be used by anybody. So, and I think this is where we are working at the moment with some of the partners in Pakistan. And this was the impact that we have achieved, and reduction in water. Water is a big thing. So farmers think they need water, but again, managing water is very important. And improving the farmer income, that's the key thing. So yeah, thank you very much. Thank you. I appreciate it. That's our team here. Yeah. Thank you, Dr. Shad.
Thank you so much, sir, for your valuable presentation. Now we have a session of certificate distribution, and for that I would like to request our Honorable Dr. Shahid Mansoor, Dr. Beena, and Dr. Musharraf to kindly come to the stage so they can provide the certificates to the participants. First, we have Dr. Kashif Riaz Khan. Kindly come to the stage to give the certificate. Then we have Dr. Nizam Dipar. Then we have Dr. Arshad Jalal. Then I request Mr. Saqib Suhail. Mr. Saeed Azaz Mehdi. Sayyid Azaz Mehdi. Then we have Mr. Ravi Lasi. Mr. Shahrukh. Ma'am, Dr. Farooq. Mr. Asif Mahmood. Then Dr. Atiya Gohar. Then we have
I must just take 1 minute होगा. I must thank all the organizers, especially जिन लोगों ने बहुत मेहनत से काम किया है. मेरे खास तौर पर मेरी अपनी टीम के जो लोग हैं, जिसमें जो है वलीद और फिर उसके बाद गुलाम हसन, फिर उसके बाद उसके साथ जुहैब हसन और वैसे और उनके दो साथ दोस्त भी आए हैं और बाकी हमारे जो अपनी टीम के मेंबर्स हैं. डॉक्टर अतिया का हर वक्त में इनका सब खाता रहता था. तो ये कि हाँ सबका, और फिर स्पेशली दोस्त जो ट्रैवल करते रहे हैं और आज की महफिल को जान बख्शे. ये सुबह से लेके 4:30 बजे तक बैठना कोई आसान काम नहीं था, लेकिन आई थिंक वी डिड इट. सो clapping for all those who have attended this meeting. हम लोग एक छोटा सा एक सेशन जो है इंशाल्लाह नजदीक जो है उत्तर में करेंगे, क्योंकि हायर एजुकेशन कमीशन की थी, और एक इंशाल्लाह एक वो करेंगे हम लोग जो है मोस्ट लाइकली जो है वो करेंगे बल्तिस्तान में. तो बल्तिस्तान यूनिवर्सिटी में शायद हम करेंगे, तो मैडम फरूक को जरूर उसमें बुलाएंगे. थैंक यू वेरी मच.
Okay, thank you everyone for the participation. And there is a session of group photo. So can everyone please come on the stage?